Chitra Mondol and Professor Snehoshikta Sharnokar. Madam, I will request both of you to come and take your chairs on the dais. Professor Chitra Mondol is an honorary professor at Diamond Harbour Women's University. She has been the former scientist at CIR, Indian Institute of Chemical Biology, and she also served as acting director of the same institute from 2014 to 2015. She is the fellow of all the National Academies of Sciences. She has been the recipient of J.C. Bose National Fellow and ACRB Distinguished Fellow. Professor Snehoshikta Sharnokar is the former chief scientist at the Division of Cancer Biology and Inflammatory Disorder of the Indian Institute of Chemical Biology. Is an elected fellow of the National Academy of Sciences as well as the West Bengal Academy of Science and Technology. She has been the recipient of National Bioscience Award for Career Development and is also a fellow of Royal Society of Chemistry. Ma'am, over to you to start the session.
Good morning, everybody. Can you hear me? So the first talk will be Dr. Venkatesh Balan. So it is my, really, it's an honor to introduce the speaker. After completing BSc and MSc from Madras University, he completed his PhD degree from IIT Madras. And then he moved to Japan, initially Osaka University, and then Tokyo, and then Kansai AIST Center till 2004. Returned to IIT Madras for a very short time, and then moved to Michigan University, State University, and spent almost 12.5 years at the Department of Chemical Engineering and Material Science. He developed producing biofuels, biochemicals, and animal feed, cost-effective microbial fermentation process, and healthy nutritional product from fungus. His groundbreaking work on the patented ammonia fiber expansion process for biomass pretreatment gained national attention, including a 2014 visit from President Barack Obama. Then he joined as an assistant professor at the Department of Engineering Technology, University of Houston, since 2017. And now he is working as an associate professor there. With deep expertise in microbial fermentation, protein genetic engineering, bioprocess optimization, he established the Microbial Product Lab for pioneering research in environmental biotechnology. He worked particularly in engineering microbes such as algae, fungi, and bacteria to convert renewable into sustainable product. He is a leading expert in the use of microorganisms to produce biofuel, chemical, and other research experience include the protein expression systems in microbes, understanding structure and function of protein, producing and identification of small molecule using spectroscopy techniques. Such type of work is really a pivotal role in advancing industrial biotechnology and sustainable production method. Some of his invention has helped to reduce the cost of producing second generation biofuel and produce healthy food material. He has several awards. I will mention only two because it has been just received last year. He received Advanced Material Award from International Association of Advanced Materials, Sweden. And in May 2024, he is a recipient of USDA EKIK De La Garza Fellowship. And he is a distinguished biotechnology researcher and team leader with over 28 years of experience in both industry and academia. He has secured numerous grants, organized sponsor workshops, received multiple awards, as I mentioned. Dr. Balan has authored over 190 peer-reviewed publications, hold 9 issued patents, and manage 13 million US dollar in research finding. So with this short introduction, I would request Dr. Balan, and we will look forward to meet you. Dr. Balan, please proceed. The floor is yours.
Okay.
Thank you.
Thank you. Hello, everyone. Sorry I could not be there in person there because of other family reasons. So today I'm going to, again, thanks for a detailed introduction. Appreciate it. Today I would like to talk about fungus mushrooms. And interestingly, this mushroom was originally discovered in West Bengal. So this is called milky white mushroom. And I was very fascinated almost 25 years ago about this mushroom. I've been working on this particular mushroom to produce value-added products. So today I'm going to share my experience on immunostimulatory properties of this particular mushroom and the role of this extracts, particularly oligosaccharides that are extracted from these mushrooms as adjuvant therapy applications. So one of my students, Sri Nandini, just graduated last week with this thesis work, and Professor Mohan is my other collaborator here. So introduction to about this technology. Mushrooms, many of you know, has got several medicinal remedies from ancient times. It has got anti-cancer, antimicrobial, and anti-diabetic and other kind of properties. It also enhances immunogenicity. Today, I'm going to talk about some of the immunogenicity of this Callosoma indica mushroom, called milky white mushroom. And many of you know about reishi mushroom. It's called Ganoderma lucidum. They are also found to have very highly active biomolecules. So, but not much fundamental understanding has been done on how these mushroom extracts or oligosaccharides has clinical implications, how they can be used as a drug. So it has been used as a traditional drug, or everybody says that consuming mushrooms is healthy, but there is no systematic mechanism how these molecules induce our immune system or activate some of our immune systems to kill cancer cells and also give other broad range of immunity. So this is Callosoma indica. Many of you might have seen this in the market. And this is an edible tropical mushroom. It has got good shelf life. It has got several rich bioactive compounds. And it has got a lot of immune modulating effects. There are preliminary studies showing immune modulation effects and effect on cancer cells, but not detailed mechanism has been done so far. So we tried to attempt to understand how these molecules have an impact on immune cells. So the primary things I'm presenting today is mostly our immunostimulatory properties of Callosoma indica polysaccharides. So many of the mushrooms has got this polysaccharides. I give an example of lentinan. This is a oligosaccharide present in the cell wall of mushrooms. So they act as a shielding mechanism on the cell wall. And these polysaccharides or have some biological activities and have immunomodulatory properties. However, not many studies have been reported about how these have mechanistic, like how these oligosaccharides have impact on immune systems and what are the mechanisms involved. So that is what the primary focus of the studies. So we are trying to show the effect of how this can activate our immune systems. So in terms of molecular characterization, so what we did was we took the mushrooms powder and did a hot water extraction. We filtered, we concentrated, and then we precipitated the polysaccharides by adding some ethanol. And then we did some NMR and GCMS and size expression chromatography analysis. So in terms of oligosaccharides, we did crude oligosaccharides and did this analysis. We found the majority of the sugars were glucose and some amount of the next highest sugar is galactose. We also found minor amount of mannose, fructose, as well as xylose. So overall, we had a mass balance closure of 98%. So though it is oligosaccharides, it has got cross-linked glycans, particularly galactose and mannose, that really gives us immunostimulatory effects. When you compare regular starch or cellulose, they are primarily made up of glucose. Because of presence of these linkages, particularly mannose, galactose, and fructose, they have some kind of immunomodulatory, stimulatory properties. So overall, the carbohydrate accounted for almost 39.4% of the dry mass of Calcium indica. There are other, as I told you, it's a crude extract. There may be other ingredients along with carbohydrates. So we did some size extraction chromatography. We passed through gel filtration column, and we got this kind of a chromatogram with 4 different fractions. We had high molecular weight fraction coming in the beginning and low molecular weight coming in the end. So these are the molecular weight ranges. So high molecular weight around 2000 kilodalton was obtained in the first fraction. And we also found almost like 6 kilodalton was found in fraction 4. We also did some NMR, and we did some using standards. We compared the properties of the NMR of fraction 1, fraction 3, and fraction 4. We found that fraction 1 had alpha 1,4 glycans, and fraction 3 had beta 1,6 and alpha 1,6 linkages, as well as alpha mannans. And fraction 4 had got, this is a 6 kilodalton oligosaccharides, has got alpha 1,3 and alpha 1,6 linkages. The linkages are very, very important. The type of linkages and the type of sugar molecules, in fact, influences the immune modulation. So in summary, we found this calcium indica polysaccharide contains many of these alpha and beta linkages. They are all known to have bioactive properties with immunomodulatory stimulant properties. And also we confirm this structure properties by using 2D NMR and also glycoside linkages composition analysis. So overall, we confirmed that the presence of bioactive molecules present in this hot water extracts that are extracted from calcium indica. So now the question is whether these compounds have got any kind of immunomodulation. So we used mouse spleen cells, and we tested whether this indica crude extract had got any kind of immune modulation. So what we did was we took the B6 mouse, we took the spleen, we extracted the white blood cells, and we added the polysaccharides to the white blood cells, and then we incubated the samples for 6 to 96 hours. And then over a period of time, we measured the what's called flow cytometer with antibody markers. We also did some ELISA analysis. We also did some Luminex analysis. We also did some bulk RNA sequencing to understand the molecular mechanism of what kind of RNA molecules are upregulated, what are the genes that are upregulated, and based on the concentration of various amount of RNA molecules. So for many students, you should have seen this slide when you talk about immune cells. So we have different kind of immune cells. There are innate immune cells, there are adaptive immune cells. So innate immune cells are given on the left, and adaptive immune cells are given on the right. So there is always some kind of cross correlations and connections. So most of the immune cells produce cytokines and chemokines, cytokines, and other signaling molecules. So these signaling molecules, in fact, communicate between the cells. Okay, so you can see all this kind of stuff, all the cells producing different kind of cytokines. And we also have several kind of binding sites in these cells, and these binding sites basically, in fact, communicate and bind and communicate and transfer the signal between the cells. So if you look at this adaptive T cells, we have T cells, helper T cells, and also cytotoxic T cells. So these are the different T cells, and they also produce cytokines. These cytokines stimulate the B cells, and these B cells stimulate the plasma cells, and that plasma cells start producing antibodies. So when you get some infection by foreign bodies, for example, viral infection or any bacterial infection, this immune system kicks in and started producing antibodies. Antibodies goes and binds to the foreign object, and that's how the macrophages identify those antibodies and try to kill them. So for sake of simplicity, in the next few slides, I'm going to use this symbol to make it very simple. So we'll have innate immune system and an adaptive immune system. The nature killer cells fall in both categories. That's what we put that in the center. So moving forward, we can identify the immune cells because it's very difficult to distinguish different immune cells just looking at them in the microscope. So we have to use some specific antibody markers for specific immune cells. So these antibody markers, basically they recognizes antigens. And so basically it gives some kind of signal. So for example, I'll just give one example. B cells has got 3 of these markers, CD19, CD20, CD22, and they also have this surface immunoglobulin and also surface IgG in the surfaces. So likewise, you can see each of the cells has got their own antibody-specific markers. And in this study, if you look at the antibodies, and this is the area where the immune cells bind. And these are the areas where the cancer cells bind, targeted cancer cells bind. So in our studies, we're going to use antibodies and identify, depending upon the type of antibody we use, they bind to specific immune cells. And based on that, we can identify what immune cells are present in that system, what immune cells are activated in the system. So here is another example. Most of you are aware of this, how the helper T cells communicate with the help of the receptors with macrophages or B cells. And so during the process, they also have some kind of a ligand, which comes of the T cells, and also a stimulated receptor, the B cells. That communication happens in both ways. So we are using this almost like this ligand or a stimulated receptor for our binding studies where the antibody bind. And based on that, we can identify what kind of immune cells are present when you add calcium indica oligosaccharides to the mouse spleen cells. So let us go and look into what are the different types of immune cells being produced. So we used CD3 marker in this case, and we vary the concentration of calcium indica from 0 to 50 microgram per ml. And we can see by immune flow cytometer how are these different kind of cells present in different locations. So over a period of time for 24 hours and 96 hours, we monitor using the CD protein complex. It's called T cells, marker for T cells in the immune system. So we can see when you increase the concentration from 0 to 150 to 100 to 500, we can see the impact, how this cells either stimulate. So in the case of control, we use amylose starch as a control. So when compared to the starch, there is a slight increase in some of these T cells at 24 hours. And at 96 hours, though it overall suppresses, but still we see a immunostimulatory effect. And we also did the same thing with the CD4, CD8, DNT cells markers. And we could see when compared to control, there's a slight improvement in stimulation of this immune cells. So we can see the CD3 T subsets. We can see in 24 hours what happens, 96 hours what happens, and we see there is a shift in different kind of cell formations. We also did. We also found that the T T cells get activated by using the CD69 marker. This is a type 2 transmembrane protein expressed on the surface of activated immune cells. So in this case, when you use CD69 T cells with a varying amount of CD4, CD8, and DNT cells, we can now see a distinct jump in the overall stimulatory effect. These are the absolute numbers of the live activated T cells, and we can see the T cells are significantly enhanced when you use a CIP. So starting with 0 and 50, we see a moderate effect, but when you go to 100 and 500 microgram, we see a significant jump compared to the control amylose. And GLP is Ganoderma lucidum. So Ganoderma is a reishi mushroom. When compared to reishi mushroom, Calcium indica has got more stimulatory properties. So overall, we can see this flow cytometer results here. So we can see the amylose, GLP, CIP with varying concentrations. So we can see how low to high modulations is taking place in the presence of different biomarkers. So next, we found that the CIP overall induces DNT cells and activated T cells. That's what we came to know from these kind of studies. Next, let us see what is the effect of CIP on B cells and subsets. So in this case, we use BT20 plus markers. So in this case, again, we saw a moderate increase around 50 microgram, and after increasing the concentration, we didn't see that much impact. And when we added the BT20 220 with CD23, 21, and 23 markers, we saw with 23 markers we saw a jump, whereas with the CD23 we did not see that much jump. So overall, we saw the dynamics of what are the different cells taking place in the two different time points. So we can see that 24 hours there is a jump, and after that, after 96 hours, it slowly declines. So we repeat the same studies with B cells and plasma cells. And so in this case, we used CD69, it's activated immune cells, and CD86 is basically antigen present in cells, particularly dendritic cells and macrophages and B cells. And also CD138 is a transmembrane heparin sulfate proteoglycan. It is present in plasma cells. So when you use these antibodies, cell markers, we figured out we saw a significant boost in the immune cells at 24 hours. And again, after 96 hours, the immune cells drops significantly. So when you combined CD220 and CD138, we saw compared to 24 hours, we saw a jump in the amount of this fluorescent intensity, confirming the presence of the immune cells at 96 hours. So overall, we found from these studies that adding CIP not only activates the T cells and DNT cells, it also activates the plasma cells and activated the B cells. So now let us see how what is the effect of isolated spleen cells on CIP indica. So now we used CD220 plus marker, and this identifies activated B cells. And also we used CD220, B220 low marker. This indicates compared with the typical B cells, and it is usually used for specific development of functional state of the cell. So we can see with CD220 plus alone, we see a decline, whereas with the CB220 low marker, we see an increase compared to the control. So 24 hours, 96 hours, we see an increase in some of these immune cells getting activated. So next, we want to study the activation of this CD220 in the presence of CD69. This is a early activation marker and lymphocytes. So we want to see the effect of these on lymphocytes. So again, in here, 24 hours, we saw a jump, and 96 hours, we saw a drop. Same thing is true when you combine with 86 or plus. The same thing, same effect was seen in both the cases. On the other hand, when you use CD220 low and CD69 plus, we saw a more or less similar kind of impact at 24 hours and 96 hours. We also did similar studies on CD138. This is a heparin sulfate proteoglycan present in plasma cells. So we wanted to see the effect of both lymphocytes and the plasma cells. And we saw similar kind of, we saw a significant activation here at low concentrations without any CIP. With increasing concentration, we see a decrease. Whereas with BT20 low and CD138, we saw a jump with increasing concentration of CIP. So overall, we can see there is some impact, and depending upon the concentration, dose-dependent impact, some of these cells are getting activated by when you see this kind of adding the CIP to this B cells, isolated B cells. So we also identified the IgM total concentrations, the whole sphenokites, and we also identified the total concentration of isolated B cells. Overall, it was increased. And we also identified the total concentration of whole sphenokites and isolated B cells. So overall, we can see with the 0 and 100 microgram, not that much significance was seen with the whole sphenokites and isolated B cells, whereas here we saw a significant increase when you use IgM. So compared to IgM was better than IgG. So overall, we can see the comparison here. Again, CD69, specific for cell surface with immune cells. And we saw with the higher MFA value suggests that that is higher level of 69 expression and often correlating it to immune cells. So overall, we saw untreated, what happens with the CIP treatment, 100 microgram, what happens with the alpha IgM, 5 micrograms, what happens? We can see the impact of different combinations. And overall, we saw a positive impact of immune modulation by adding CIP. So overall, we found that not only activated T cells and DNT cells, it also activate antibody production and activate B cells and plasma cells. So let us see the effect of, so overall the CIP does not have significant effect of myeloid cells. So that's what this study is concluded. We can see this, we used a CD11B marker and CD86 plus marker. And we also used CD11B and F480 plus marker. This is specific for macrophages. And the CD11C is specific for dendritic cells. So by combining these combinations, we saw that not slight improvement or increase in absolute number of live cells was seen 24 hours and 96 hours. But when you use a CD11B and also CD11 with the F480 and CD11B with CD11C, we saw a drop at low for 24 hours and higher at 96 hours. So what we found from these studies are that we found the increase in DNT cells, activated T cells, plasma cells, activated B cells, antibody production. We also found there is an increase in myeloid cells count when you add the CIP. So overall, the immune system is getting triggered by adding this crude calcium indica polysaccharides. So these are some of these observations. CIP elevates cytokine production And we can see the myeloid cells are these kind of cytokines, and T cells has got this kind of cytokines. And these are the common cytokines present in both the P cells and myeloid cells, and T cells and myeloid cells, and T cells and B cells. So eotaxin, IL-6, is present in all of the 3 cells. So we can see that the cytokines from myeloid cells, it recruits the immune cells, and it's basically a chemoattractant and activator of other immune cells. And also in the role of cytokines in the T cells, or it enhances immune response, it stimulates the antigen present in MHCs, and also enhances the proliferation and differentiation of survival, and also it promotes the development of helper T cells. So these are the observations we observed based on the whatever thing we found so far. So overall, we found that not only the myeloid cells increases, we also saw some cytokines being activated because of the presence of CIP. So we also did some molecular level understanding. So when you add CIP, we do see induction or activation of some of the immune cells, but we would like to see what is the impact of upregulation of any of these genes by inducing this CIP. So we did this transcriptomic analysis to figure out what genes are upregulated and what genes are downregulated. We did a volcano plot, and we can see the distinct difference between some of these cells. And also we saw, we compared this differential expression, and based on that, we can see the relative concentration of the cells, the RNAs at varying concentration of CIP, 0 to 100. We can see how this shift happens. So overall, the KIG pathway analysis and gene ontology analysis. So we can see here, we can see some of the activation of cells. We can see as a function of increasing order, we have organized this. And we see some of this TNF signaling pathways is activated, IL-17 signaling pathways is activated. So because of these kind of activation, at genetic level, these overall, there is stimulation of some of these genes. And because of the stimulation of those genes, we see some of these cells are induced or overexpressed in the system. So overall, we could evaluate which of the genes are upregulated and during the process of adding Calcium indica. So this is the overall mechanistic understanding we found so far. So the Calcium indica, when we add, it basically stimulates the B cells. And so the B cells activated and is producing the plasma cell generation and antibody production. And this stimulates the immunity to enhance the pathogens overall. And there is also communication between the B cells and the CD8 T cells. And so because of the activation of the cytokines, some of these myeloid cells are getting activated. And because of the activation of the B cells and the myeloid cells, some of the T cells are getting activated. So because of the activation of these immune cells, we are also seeing increased concentration of these cytokines. So overall, we saw increased concentration of all the immune cells and cytokines. So we can confidently say that the adding CIP do in fact have immunogenicity. That means it modulates the immune system. So what is the implication of this study? So people can ask, okay, it induces immune cells, so what? So while giving some vaccines, for example, we had a big pandemic like COVID infections where people are given vaccines. So vaccines are biological preparations provided to increase immunity against specific infections. In the case of COVID, we had this spike proteins being produced and given to our system to stimulate the body immune system. And typically they use alum, like aluminum salt, in this vaccine. And there are a lot of claims that adding this aluminum salt also causes some kind of autism and other kind of disorders in kids. So that's what there are anti-vaccine movements in many countries because of the presence of adding this aluminum salt in this vaccines. So what we found is based on our studies, we found that adding calcium indica do stimulate the immune cells. So we can use this calcium indica extract as an adjuvant in the vaccines. And more studies we are working on these kind of things on real mice, and also maybe in the future some human trials will be evaluated to see the efficacy. And if it goes very well in the future, this could be used as an adjuvant in vaccines, and that will help many people. It's a low-cost adjuvant, and it may not have much side effect like alum. And if it works out very good, this could be a routine um, adjuvant therapy for which will benefit humans. So in conclusion, we found that C. indica mushroom has immunomodulatory properties, and also we did additional studies using metabolites, and we found that there are some anti-cancer compounds, and we have anti-cancer effects. So overall, immunomodulation also stimulates immune cells and kills cancer cells. There's another studies we are working on right now. And also we found the carbohydrates present in calcium indica are one of the major reasons for immunomodulation, and also boost the immune system and immunocompromised subjects. And also it is a very good potent adjuvant for vaccines. And we also are examining how this calcium indica polysaccharides can be used in the treatment of cancer for immunotherapy, and so that way we can expand the therapeutic usage. With that, I'll stop here. I'll thank the organizers for giving me an opportunity to present our work. Hope some of the work we did, we presented here, stimulate some of the students in the audience, and may help them to take this kind of research to develop some therapies for future generations. Thank you so much.
Thank you, Dr. Balan, for excellent talk. The floor is open for any questions. Slides you have used 500 microgram per ml. Is it the
for laboratory experiments, we use varying concentrations starting from 0 to 1,000 micrograms.
Yeah.
Again, this is a crude polysaccharides. Okay, so out of this crude extract, we found only 38% of that is polysaccharides. There are other kind of compounds or other kind of impurities that could be present.
Yeah.
So to be on the safer side, we use
slides, and they found that these cells are really looking like a small lymphocyte, but they have a very intricate Golgi network. Why do they have this intricate Golgi network? Because these are secretory cells, right? They have to churn out loads of proteins. You'll be surprised to know that they are huge producers of interferon. So if you take 50,000 cells and put in a small culture plate and activate them, I'll tell you why they produced interferon. And you activate them, you will get like 500 picograms of protein, interferon protein, if you just look at one interferon protein, interferon alpha, and that's a huge feat for a mammalian cell. And another cell, this already looks like a lymphocyte, and another lymphocyte has this secretory phenotype. And the initial pathologists got confused, and they thought they were looking at plasma cells. Because plasma cells, again, have secretory phenotype. They have to churn out a lot of proteins, antibodies, etc. And that's why this name came, plasmacytoid dendritic cell. They share the name dendritic cell because from the hematopoietic origin, they share the ontogeny. Now, as I said, so they produce interferon. Why do they do that? Because they have toll-like receptors, not on the surface, but within the endosomal compartment, which are TLR7 and 9 in human. In mouse, it's a little bit wider. Now, and these TLR7 and 9 are toll-like receptors that will recognize nucleic acid ligands. Now, where from these nucleic acid ligands come? They will come from either pathogens, and also they can actually come from self-origin, self cells, host cells. And it was long back during my PhD, so our group was the first to show that yes, self-origin nucleic acids can actually activate PDCs. And because self-origin nucleic acids are activating in a sort of sterile context, they are really crucial, or they are really very important in a lot of pathogenic contexts like autoimmune diseases. And we found, and other groups also confirmed, that self-DNA, RNA, they can actually bind to different endogenous proteins which work like alarmins, and they are taken up by these cells, goes to the endosome, activates toll-like receptors, produces type 1 interferons. Now, so our lab has been, I have been working on PDCs for last 2 decades. The first decade was learning the craft from the masters, and the second decade was, doing my stuff. Contributing myself. And so I have been working on this for last more than 20 years. And we have been interested in looking at role of PDCs in a lot of chronic inflammatory contexts, including lupus that was just mentioned. By the way, before I forget, I don't think alum is associated with autism, so for the students. So just don't become anti-vaxxer. It just came to my mind while I was mentioning to the previous speaker. Anyway, coming back to our work, and we did look at how PDCs and interferon induction by PDCs can play a role in autoinflammatory diseases. But also a part of the lab was very much interested in the cell biology of the PDCs, right? Because these PDCs are really a dedicated cell for producing type 1 interferon in response to cellular activation. Naturally, it has, we always hypothesize that it will have its special machinery to drive the secretory phenotype through cell biological mechanisms. And we have been looking at that. For example, we have been looking at what are the extrinsic regulators that affect PDC function. We are looking at endosomal trafficking. The reason for looking at endosomal trafficking control in PDCs is because these TLRs are present in the endolysosomal compartment, and they come through this path. I don't think this is working. Anyway, through this path, and they actually reach the surface and gets into the endosomal compartment, right? And then one may surmise that perhaps these TLRs, when they are reaching the surface, they will also look at the nucleic acids and they will activate the PDCs. That doesn't happen. Why? Because these TLRs, when they are en route to the endolysosomal compartment, they are not functional, because they will get functional when they reach the endolysosomal compartment, reaches a specific pH window where other enzymes like cathepsins will cleave it and make it active. And that's a very interesting natural mechanism of protecting from responding to self-origin nucleic acid all the time. Because if it's responding to nucleic acids on the surface, our body is always having a lot of free nucleic acids because cells are dying, nucleic acids are going out, and they're getting disposed of, they're getting cleaved off by DNases and RNases present in our body, things like that, right? So it's a very interesting phenomenon. And naturally, because it operates in a very kind of narrow window of pH, the control of TLR trafficking as well as the endolysosomal maturation also needs to be really tightly controlled, right? Now, that's why we are really interested in endosomal trafficking and endosomal pH regulation as well. Now, starting with extrinsic regulation, a student called Deblina, who is now in MD Anderson doing her postdoc, she found that lactic acid present in inflammatory tissues can actually affect PDCs, and in this case, they actually inhibit interferon alpha induction and drive a T regulatory response. That's a control part of it. And also another student found another extrinsic regulators, again, really not known very much in the context of immune system, called endocannabinoids, the endogenous ligands of cannabinoid receptors that are circulating in the blood. And she found that endocannabinoids play a very important role, again, in stopping the PDCs from producing type 1 interferon. That again tells you that PDC-induced type 1 interferon has this huge potential for driving autoinflammatory diseases. And that's why body is trying all the way to kind of stop it from getting overactivated or aberrantly activated, right? Now, coming to the next part, endosomal trafficking, again, different students took up different regulators, which are, now I should point out, in most cases, these regulators are actually unique for PDCs among all the circulating immune cells. And that's where we are actually interested. We want to find out what are the specific regulators in human PDCs. Another student, Ronit, he found that a protein previously known to actually help in the transport of melanosomes within melanocytes, called melanoregulin, is actually expressed in PDCs, and he found that it actually helps PDCs to decide whether a TLR ligand cargo will go to the autophagosome or to the lap phagocytosis, right? L6 associated phagocytosis. Another very interesting area that recently came up in our lab is looking at this mechanical cue in immune cells called, involving this mechanosensor called piezo1. And these students together actually, so it actually took a long time in our lab. We have been working on this for last 8 years. And 3 students took part in this project, and they found that a long-standing question in the field was that TLR9, if it gets signaled from early endosome versus if it gets signaled from late endosome, it has different functional outcomes. And they found that this differentiation is largely driven by structure of the cargo. That means how big the cargo of TLR9 ligand is. And that's really interesting, and we found that it is actually giving a mechanical cue to the membrane, how big the endocytic pit is, and that's how the cell biological mechanisms are controlling where from TLR9 signaling will start. Okay, now coming to endosomal pH regulation, recently Purvita showed that ATP13A2 or PARC9, it's a PARC protein. Again, it's previously reported in neurons, mostly in dopaminergic neurons. And she found that this PARC9 protein is highly enriched in PDCs among all circulating immune cells. And again, it plays a very important role, crucial role in controlling the pH. So just for the students, so endolysosomal pH is controlled in all phagocytic cells, right? Because cathepsins have to be activated in all phagocytic cells so that it can actually digest the cargo. Now, because PDCs are so crucial and they have this really incredible ability to ramp up interferon production really fast within 4 hours it will start churning out interferons, its control is even more stringent, and that's why additional pH regulators are actually expressed in this cell. Now, I'll just briefly talk about a small story. Again, it involves the endosomal pH regulation, and this work was done by Jafar Sharif. Jafar is on his way to NIH at this point. I don't think he has reached the destination. So Jafar just completed this work, and he found out that another family of proteins called SLC4A family of proteins, which are anion exchange channels. Again, it's not really expressed to a great extent in other circulating immune cells, but it's actually very enriched in PDCs. And when he found that these anion exchange channels are really enriched in PDCs, we got into this. So this is, you can actually see there are at least 3 members of this channel, AE1, A3, or SLC4 A1, A2, and A3. And we found that A3 is in a steady state, human PDCs, A3 is really highly enriched, and which is not present in other immune cells. And this is the recently solved cryo-EM structure of this channel. And again, this has been previously reported in certain neurons in the central nervous system. Now, when we looked at immune cells, so we did a very simple experiment. You just take PBMCs from human donors and then isolate PDCs from them, right? So if your gene is enriched in expression in one immune cell subset, then its expression level should be higher than the PBMC bulk expression level, right? So that tells you it's enriched. A very simple experiment, and that shows that PDCs are really enriched in the expression. And you can actually see we looked at different immune cells which are circulating in the blood and compared their level. And it's moreover, when PDCs are, you can actually use this one, right? So when PDCs are activated, so if you give a TLR9 ligand to PDCs, TLR will be activated and interferon production will start. So when you are activating PDCs, again, expression of this gene is really heightened. So then we wanted to find out, because in neurons, it's mostly reported on the cell surface. It's a chloride bicarbonate exchange channel, right? It gets out chloride, gets in bicarbonate, and modulates the intercellular pH. So if there is free bicarbonate inside the cytosol, the acidification will be retarded, right? But when we looked at PDCs, we found that this channel is definitely present some amount on the surface, but also rather predominantly it's giving a vesicular staining. A lot of endolysosomal compartments are actually expressing it. And if you look at this, it seems the late endosomal compartment or lysosomal compartment, which is essentially more acidic compartment, by the way, in the endolysosomal path, expresses this much more, right? And then we tried to see whether, because there is an inhibitor available for this family of channels, which is called DIDS, and it's diisothiocyanate, stilbene disulfonic acid, anyway. So DIDS I just mentioned. So DIDS doesn't have any specificity towards SLC4A3. It inhibits A1, A2, A3, all of them. And then we tried to look at what happens in PDCs when they're getting activated in the presence of the inhibitor. And we used a pH determining dye, right? So you can actually have a cargo which is tagged with something called pH rhodo that will give fluorescence in different pH. So we actually use 2 different methods to confirm. So in one case, when there is low pH, fluorescence intensity decreases, and in another case, when there is low pH, fluorescence intensity increases. And so in pH rhododextran, it's the low pH leads to higher intensity, and you can actually see when you give DIDS, when you are stimulating PDCs with CpG-A, with CpG oligonucleotide, TLR9 ligand, there will be some amount of pH in the endolysosomal compartment. If you give DIDS, it decreases. And you can also use CpG, which is tagged with FITC. FITC is also pH sensitive. And in that case, low pH will actually reduce the fluorescence of FITC, and that you can also measure and then find out that yes, the pH is, so the endosomal compartments, endolysosomal compartments are getting alkalinized. That means its TLRs will be less efficiently processed to be active receptor, although we haven't looked at that. Now, we wanted to see whether that actually affects interferon induction. It does really severely. And there is, so you use CpGA for looking at the interferon alpha induction. There are other kinds of oligonucleotides called CpGB, which will largely drive TNF alpha induction. Again, I didn't talk about that project, so I already mentioned, right? When TLR9 is getting activated, will actually differentiate the functional outcomes. So this CpGA and CpGB actually does that. CpGA forms a bigger cargo. It signals from the early endosome and drives interferon alpha induction. And when CpGB is used, which is mostly a monomeric ODN, and that will activate TLR9 in the late endosomal compartment. And because the cargo goes fast to the late endosome lysosome part of the compartment pathway, and then activates TLR there, right? And so you can actually see both CpGA and CpGB induced interferon alpha and TNF alpha induction respectively are inhibited. Then we wanted to do a knockdown experiment because we always thought that SLC4A3 is doing it, right? So we knocked down SLC4A3 because you can't really do a lot of CRISPR in these primary PDCs. These are primary circulating PDCs collected from the buffy coats of donated blood, right? And so, but siRNA works nice. And so when we did that, we found that yes, there is some decrease in the interferon induction when you knock down specifically SLC4A3, but it's not really quite there, right? So that tells you there is something else going on. And another way we actually confirm these experiments is by looking at the level of protein reduction or expression reduction of the target gene and how it is correlated with the functional outcome. So that's the graph here, right? So we actually found there is no correlation between the interferon % reduction and the SLC4A3 knockdown efficiency. So that was interesting. So then we thought maybe other family members are compensating for it when you are knocking it down. So we actually looked at both SLC4A1 and SLC4A2. SLC4A1 doesn't really change much, but SLC4A2 expression increases. And we found that SLC4A2 is also expressed in the steady state to some extent. And again, if you activate these cells, the SLC4A2 expression further increases. That means SLC4A3 and A2, they actually work together in PDCs. And when we look at the confocal microscopy to find out what is the localization, you can actually see in the unstimulated cells, there is a very strong surface expression of SLC4A2. But when you activate these cells, there is more vesicular staining, and so these are actually playing role in the endolysosomal compartment. Now you can do a dual knockdown. Now you can knock down both SLC4A3 and A2 because A1 was not really changed in response to the knockdown of A3 alone. And so then you actually look at what is like happening with the pH. Yes, so the pH is increased really high. So this is CpG FITC experiment. And then our hypothesis is that there is bicarbonate flux and there will be a more chloride intake. And so we actually looked at the chloride content of the endolysosomal compartment. We isolated the endosomes and we measured the chloride content of the endosomes. And we actually found, yes, when you knock down both these proteins, then you actually find a very strong kind of stopping of chloride influx as well as pH decrease. Now, if you go back to the interferon alpha induction phenotype, again, you find that this is now really solid knockdown or solid deficiency of interferon induction in response to this dual knockdown. And also, if you look at now this correlation of what is the % of interferon alpha reduction and what is the average knockdown efficiency of both these genes, you actually see a a fantastic correlation, right? So that gives us more kind of confidence. So that was interferon alpha. This is TNF alpha in response to CpGB. Again, you find the same thing. So we found that SLC4A3 and A2, SLC4A3 is PDC enriched, SLC4A2 is more ubiquitous, but still both of them work together in PDCs to tightly regulate the endolysosomal pH and help in the control of TLR9 induced interferon alpha induction. Now, because you have one inhibitor, and this inhibitor has been used in murine models for other contexts, we wanted to see whether you can actually affect in vivo inhibition of the PDCs. Now, we did a pharmacodynamics model, right? So you just trying to activate PDCs in vivo and then giving this small molecule inhibitor. Well, this is not a drug, right? So this is a non-specific SLC4A family inhibitor. This is just proof of principle inhibitor study. Now, when you give that small molecule inhibitor 1 hour before intraperitoneally in mouse, and then after 1 hour, you come back with loads of CpGA and CpGB mixture, which is essentially TLR9 ligand. And you inject again in the peritoneal cavity. What happens? There will be a peritoneal infiltration of different cells because you are inducing peritoneal inflammation, and you can actually isolate the peritoneal fluid. You can actually get out the peritoneal cells. You can measure whether interferon was being produced because we already see that there are PDC infiltration inside the peritoneal cavity. That means whenever you are giving TLR9 ligands inside the peritoneal cavity, PDCs are coming, they're producing type 1 interferons. Now, when you give this inhibitor, interferon induction in peritoneal fluid goes down. Also, if you look at interferon signature genes, so interferon signature genes will be expressed if interferon is around and host cells are seeing interferon through their interferon receptor. And we actually looked at several interferon signature gene expression in the peritoneal infiltrate cells, right? So you take out the cells, get the RNA out, look at the expression of these genes. And then when you do that, you find again the inhibitor is reducing. More stark effect is found in the systemic circulation. Now you are looking at interferon induction in the serum. Again, the inhibitor almost abolishes the whatever induction is there. In peritoneal fluid, still you have some interferon because it's the local inflammation that is going on because the TLR9 ligands are there. But it's not that much that this will leak out to the systemic circulation. And we also looked at circulating immune cells, whether they are seeing interferon, and you can actually see that interferon signature gene expressions are much less. Then the question was, can you actually do it in a disease model? So there are at least 2 disease models that we work in our lab, which are known for having crucial effect of PDC function in their pathogenesis. One is psoriasis. In the initial stage of psoriasis, PDC activation is very crucial. Interferon activation is interferon induction is very crucial. So you can actually do TLR7 ligand-induced psoriasis on the mouse skin in wild type mouse. We frequently use that for several experiments. And if you now inject the mice with this DIDS, you can actually see the psoriasis lesions are not showing the characteristic morphology. We actually did a proper histopathologic assessment of the different features or different histopathological features because there are classical histopathological features associated with the psoriasis phenotype, and all of these get reduced. And if you now go to the circulating immune cells, look at the interferon signature genes. To save space, I'm showing average, median, not average, median of 5 different genes. We call it ISG index or interferon signature gene index. They are also reduced in the peripheral circulation as well as in the cutaneous bulk. If you now take the skin out and isolate RNA from that, again, the interferon induction is much less. Let's not get into this. So this is, you can actually see the PDC, circulating PDC frequency is not affected. So cells are not dying, right? So PDCs are there. They're inhibited from producing type 1 interferons. Now, if you look at the T cell aspect of this disease, it's mostly a Th17 driven disease. Also Th1 cells play a role. And we found that yes, the Th17 and Th1 cells are also reduced both in skin and also in blood. Now, we also went to another model, that second model where interferon is so crucial, right? Systemic lupus. Again, how do you do systemic lupus in a wild type mouse? Because we didn't have access to a genetic model of lupus. So there is a induced model of lupus developed by Mariana Kaplan in NIH, and which is actually very severe SLE model. So again, you take a TLR7 ligand and paint the TLR7 ligand imiquimod on the ears of the mice for 42 weeks. And they develop severe systemic inflammation and produce autoantibodies. Yes, you are looking at anti-Smith autoantibody, anti-dsDNA autoantibodies, right? And also, this is a very interesting phenomenon, especially in mouse. In humans, I have not seen it. In most murine SLE models, you actually see a macrophage population expansion. You also see that in this imiquimod-induced model. And now, so for 42 weeks, they have developed disease. Now we started injecting DIDS up to 60 days, right? So it's kind of a therapeutic model. And then you see that the autoantibody response goes down. Even the macrophage population, this inflammatory macrophage population accumulation is also going down. If you go to the glomerulonephritis scores, if you just take out the kidney, take sections, look at the histology of this. So you can actually see these are classical glomerulonephritis picture. The major thing that for students, look at the size of the glomerulus. It's actually expanded like anything because it's incorporating a lot of immune infiltration. Now, when you give this inhibitor for these SLC4A family channels, again, the glomerular size is preserved. Also, proteinuria is reduced. So this is the vehicle. You are comparing 42 days, disease has developed, and 61 days, you have stopped your therapy, and you're looking at proteinuria. Now you go to the inhibitor, again, 42 days and 61 days. You can actually see proteinuria is reduced. So it's very interesting therapeutic target. I don't think DIDS is the drug. It's a small molecule inhibitor that proves the point. But I think this is a very interesting target where like drugs can be developed or lead molecules can be developed against. And because it needs to be a selective targeting, you need to know that it's actually targeting SLC4A2 or A3, things like that. Otherwise, there will be other problems because SLC4A2, by the way, is kind of ubiquitous. It is present in a lot of cells. So I'll just end here. I discussed work by Jafar. And so Jafar is now headed to Sylvia Boland's lab. So she has been in NIH. She has been one of our idols in the SLE research because she has been really phenomenal in systemic lupus research. I have to acknowledge, so I have recently moved to Ashoka University in the Department of Biology. And this work was done in CSIR-IICB by Jafar. Jafar did his PhD from ACSIR, and our work has been really supported by different Government of India funds, also CEFIPRA, which is an Indo-French organization, as well as a very recent philanthropy foundation, which is Ignite Life Science Foundation, which is actually helping us in another aspect of autoreactive inflammation. Now, let me just take 2 minutes to introduce to you this university where I belong. This is Ashoka University, very interesting experimental model. I think one of its first kind in India, a philanthropy initiated liberal arts university where you find all kind of aspects of human knowledge being taught and being studied all together. And it's a fantastic setting in terms of its campus. And so these are so we have quite a big department at this point. You can actually see all of us are here. And there are international advisors, Jack Shostak, Ron Vell, Jim Collins and Maria Leptine. So they are closely engaged with the department's growth and planning. And also there are visiting fellows like Hiroshi, Shahid, and Shashi, who are always giving us inputs, right? By the way, within the Ashoka University, there is now it has come up as Trivedi School of Bioscience, which is a dedicated building here. And the Trivedi School of Bioscience is composed of Department of Biology, where I belong, but also there are different focus-oriented centers like Digital Health, Quieter Center for Digital Health, and Health Analytics Research Center, which is called CHART. New Human Immunome Center is coming up, hopefully in this year only. We have already got this Ashoka Zeiss core imaging facility, and Ashoka Immunocore is coming up really soon. This is the facade for Trivedi School of Bioscience. I'll just point out to the students and PhD scholars, Ashoka has a very fantastic postdoctoral program called Simon Ashoka Early Career Fellowship. It's quite a good salary and fantastic kind of infrastructure that will be at your disposal. And also there will be research funds. So if you are really thinking of staying back in India, you are still have this fire in your belly and you want to do science and you do it in India, come over, talk to us, okay? Ashoka PhD fellowships, these are university fellowships that will be supported. This is, so there are different aspects of biology you can actually work on. And so look at that, all the master students do consider. It's a fantastic place to be. This is the Ashoka Zeiss core imaging facility we have. This is perhaps one of its kind in India at this point, at least at this moment. This is at the inaugural, inauguration. Vijay Shomak Rai Chaudhuri, our VC, and Jack Sostak, who inaugurated this facility. We have the most state-of-the-art microscopy facilities from Zeiss. And I'll end here. Thank you.
We have listened such a great lecture from him. He described so well plasma, cytoid, dendritic cell, but he loves to say professional DC. So PDC is genesis, its function, and functional impact. Everything you have heard so well. So I wish that students or
any questions, there is no pressure. You don't have to ask questions. If you have any question, just grab me. I'm around. Please ask anytime it appears to your mind.
But I have one question.
Yeah, please. Yeah. Yeah. No. Yes. I'm not aware of. So a lot of IHG, because IHG biology is something of your domain. So we usually stop at looking at IHG, because IHG gives us a very dependable signature of interferon induction in the vicinity of the cells, right? So that's where we stop. So our index is for signature of interferon induction, because that becomes really easy for assessing whether in a tissue interferon has been induced. Otherwise, measuring interferon in tissue is really difficult. So but the downstream pathways, I'm pretty sure when interferon alpha is getting churned out by PDCs, and it's such a high amount, all the IHGs will be affected. Now, certain cells perhaps will actually express subset of IHGs versus other subsets. And so in that case, the biological outcomes may vary, but I'm not really an expert in this. I don't know.
Thank you. Very nice talk.
Yeah, one question from Kaushik.
Dipoman, fantastic talk, fascinating talk. I have a question. Maybe I missed it, but just for clarification. So the involvement of this anion channel that you are working, one question is it working only in the endolysosomal compartment, or can it also be activated on the plasma membrane itself?
Of course, yes.
So in that case, is it the outcome of some other response? It is triggering the first initiator response.
So the pH down, the outcome of pH down regulation?
Right.
I don't think so, because there are other ATPases that will actually start the pH fall in the endolysosomal compartment. I think these additional regulators are controlling it at different times, like compartments of endolysosomal pathway. For example, this regulator is actually restricted in the lysosomal compartment, where pH needs to fall further, right? Things like that. So I don't think the pH fall is initiated by them, but these are additional regulators that PDC have that can actually help it regulate pH more stringently compared to, say, macrophages, where pH will fall like this. And in PDCs, perhaps it like balances it so that it doesn't get activated too much. And also it gets
So another quick follow-up question, not follow-up question, quick separate question. So PDCs are mostly involved in innate immune response generation, or they can also be involved in adaptive triggering as well?
So these are innate immune cells because the cytokines they produce are innate cytokines. But definitely they will link to it because interferons are very crucial for T cell expansion. For example, if you have interferon around, DCs will talk to T cells in more fluently.
But they don't possibly have any antigen presenting cell ability as such, right?
They do present antigens. Interestingly, although I don't work on that area, a lot of people who work on antigen presentation of PDCs, they show that antigen presentation of PDCs become important in tolerogenic contexts. For example, oral tolerance and things like that, even in the intratumor immune suppression, things like that. But in a ongoing immune response, PDC antigen presentation is not that much. I mean, you will have nice antigen presentation without PDC.
Any other question from audience? Okay, last question from me.
Sure.
In cystic fibrosis, they do have this problem of ion channel. So, what is your?
I don't think that's linked to this. That's a different different ion channel which is affected. But definitely there are
PDCs are definitely.
PDCs. I don't think there is any report that in CF PDCs are involved. But there are indeed reports because CF was largely thought to be channelopathy. But now people are slowly getting into the inflammatory context of cystic fibrosis. Because initially, I mean, for doctors, cystic fibrosis inflammatory process is largely outcome of the disease. But now we think, I think there are data showing that peripheral immune cells do participate in the pathogenic process as well. It's not only the disease progression phase of the disease. But I don't think PDCs play a role. I mean, there is any data. I don't know.
No data.
No data.
Because
Yeah, but even interferons, I don't think.
Drug will be somehow helpful for those patients.
But in CF, I think a lot of drug candidates are already in clinical trial, Well, I mean,
Yeah. Okay.
that doesn't mean that there will be drugs, but they are in clinical trial.
Thank you very much.
Thank you, sir.
I think you have participation from the organizer.
Yes, we will request the chairpersons to felicitate Dr. Ganguly. So I would like to repeat that there is an exhibition going on just behind the curtains at the back of the seminar hall. You can take your time and visit the scientific models displayed there. And there is also a stall given by our vendors and our co-sponsors on the other side of the wing. And our volunteers will guide you there. You can have your time and visit the stalls also. Now we will start our next session. I call upon stage the honorable chairpersons for the session, Professor Sushanta Roy Chaudhuri and Professor Mitali Chatterjee, Madam. Please take your seats on the dais. Dr. Sushanta Roy Chaudhuri is an ICMR emeritus scientist at the CSIR Indian Institute of Chemical Biology. He is also the honorary advisor of research at Dashgupta Cancer Center and Research Institute. He has been the recipient of J.C. Bose National Fellow, as well as he is the fellow of all the Indian National Academies of Sciences, and he is also being the member of Guha Research Conference, and he's also a governing body member of the Dashgupta Cancer Center and Research Institute. Madam Dr. Mitali Chatterjee is a professor of pharmacology in the Institute of Postgraduate Medical Education and Research, IPGMER, and also a J.C. Bose National Fellowship recipient. After completing her medical MBBS degree, and then she also did her MSc, and then she did her MD, as well as also a PhD. So she's in true sense a clinician scientist. Her research interest is in redox biology and immunopharmacology. With that, I will hand over the session to Madam and Sir. Over to you.
Hello, everybody. Very good afternoon. Are you able to hear me at the back? Micটা চলছে? A very good afternoon to all of you, and welcome to this session, which has a number of interesting presentations. It is my proud privilege to introduce Dr. Joyashree Das Sharma, who is a professor at the Indian Institute of Science, Education and Research. She is a very distinguished neuroscientist. And just to give you a very brief overview about Joyashree's work, she completed her PhD in collaboration at the Indian Statistical Institute along with Indian Institute of Chemical Biology in Kolkata, and then moved on to do her postdoctoral work at the University of Pennsylvania. And later she joined Thomas Jefferson University Neurology Department as an assistant professor. She then moved to ICER, where she is presently a professor and also is an adjunct associate professor at the Department of Ophthalmology at University of Pennsylvania in the US. She is an elected fellow of several academies. It is with great pride that I tell you she is an elected fellow of the Indian Academy of Sciences, of the National Academy of Sciences, elected fellow of the Indian Academy of Neurosciences, as also the West Bengal Academy of Science and Technology. That is no mean feat and is in itself a testimony to the excellent work that she does in neurosciences. I will leave it to her to tell us about her science. So it's over to you. You have 15 minutes. Just to keep time, we will at 13 minutes tell you that you have another 2 minutes to go. And hopefully if you tell us within 15 minutes and then keep 5 minutes for discussion. So thank you, Joyce, and over to you.
Sincere thanks goes to the Honorable Vice Chancellor, Professor Dhruva-Jyoti Chattopadhyay, and all the organizers for giving me an opportunity to present my work here. I'm going to talk about the gap junction protein connexin 43 and its chaperone ERP29 mediated cellular signaling in beta coronavirus induced neuroinflammatory demyelination. So first I would like to introduce what is gap junction. Gap junction formed by connexin molecules. 6 connexin molecules form hexameric hemichannel, and this hemichannel goes to the surface and docks or associate with another connexin and form gap junction channel. And these channels are very important for maintaining the potassium buffering in the CNS and allowing small metabolites to gap junction intercellular communication and allow the small molecule to go through. As we all know that CNS is a very complicated panglial system, and it has astrocytes, oligodendrocytes, neuron and microglia, as well as ependymal cells and blood vessels, and they communicate with each other through metabolic coupling through gap junction protein. And among these, the focus of my talk will be connexin 43 in the astrocyte and connexin 43 and 47 in the oligodendrocytes. So with that, I would like to say that there are several evidences that there are altered gap junction protein in the one of the abundant connexin, which is the connexin 43 in several viral infections like coronavirus infection, bovine papillomavirus infection, Rous sarcoma virus infection, and HIV infection. But there is not much information is available in murine beta coronavirus, and now beta coronavirus is one of the major thrusts in the neurovirology field as well as in the virology field, because SARS-CoV-2 belongs to the beta coronaviridis family. And my talk will be focused on how murine coronavirus induced alteration of cell-cell communication may affect the viral replication, spread, and pathogenesis. And neuroinflammatory demyelination could be due to alteration of astrocytic 43 and oligodendrocytic connexin 47 mediated gap junction intercellular communication, and also to understand the viral induced like modulation of connexins and its regulatory protein that may help in designing therapeutic targets. So first I would like to introduce you that murine beta coronavirus belongs to the family Coronaviridae and genus beta coronavirus, and beta coronavirus are the much more threat to the human society because it belongs to the SARS-CoV, SARS-CoV-2, MERS-CoV. As you know, SARS-CoV-2 causes the trauma for last 3-4 years, and murine beta coronavirus belong to the same Coronaviridae family and genus beta coronavirus. And murine coronavirus is a prototype beta coronavirus, which we can use it in BSL-2 facility. We don't need any BSL-3 facility, and it causes several diseases. Different strains causes hepatic, enteric, neurological manifestation. That's what it is, a very good model system to study. And that's what we use this. And for last 2 decades, we are using the in vivo and in vitro approaches to understand the viral-induced neuroglial dystrophy and how individual neural cells actually contribute to the demyelination phenotype, which mimics certain pathology of human neurological disease, multiple sclerosis, and now it is a model to understand the SARS-CoV-induced neuro-COVID. So while we do in vivo studies, to study neuroinflammation denoted by meningitis, encephalitis, microglial nodule, and we also take the neuropathology to understand the demyelination, which happens due to macrophage-mediated myelin stripping. At the same time, we are interested to understand individual glial cells like astrocytes, oligodendrocytes, and neuron, how they contribute to the axonal loss and concurrent demyelination. So here, first thing I wanted to say, from the large number of studies, we came to a conclusion that the virus infects both neuron, astrocytes, oligodendrocytes, and they all can be victims of this viral-induced neuroinflammation. And what we know, that oligodendrocytes are dying, and this damage actually causes the demyelination, astrocytes from glial scars, and in some cases, axon degenerates. But how it happens, that is not very much clear. So to understand that, we know both astrocytes, oligodendrocytes, microglia, and endothelial cells, they secrete cytokine and chemokines, and they actually invite the peripheral immune cells. So again, a summary slide showing that when we inject the virus into the brain, it has a cause-effect relationship. Because in myeloid coronavirus infection, we can know that that is causing the cause. And that's what we show, that astrocytes and microglia get infected, cytokine, chemokine get secreted, leukocyte infiltrates, causes meningitis, encephalitis, microglial nodule, and inflammatory responses if the virus spread to the white matter. And that time viral RNA reaches its peaks, infectious virus particle also reaches its peak. But after that, there is a due to the interaction of microglia monocyte macrophage with T cells, clear the virus and they establish the homeostasis. And in that homeostasis, the viral particle is completely gone and viral RNA present in low abundance. But in low abundance, the virus causes the demyelination and axonal loss. But how it causes the demyelination and axonal loss when virus is not there is a major question. So here that focus is, as I said, the astrocyte oligodendrocyte metabolic coupling through gap junction communication is very important for myelination. So that is one of the target of my studies, what I am going to describe here. So astrocyte and oligodendrocytes, they form gap junction channel and they get destroyed during viral infection, which I will show you in successive slide. And in another thing, when virus infects neuron, they actually releases ATP, which uses unopened hemichannel and activates astrocytes, and astrocytes releases leaf. Leaf actually helps oligodendrocytes to form myelin, but the leaf secretion gets destroyed during viral infection. So taking this, what we did in our lab, we have a astrocyte-enriched culture from the mixed glial culture, and which shows that connexin 43 forms very nice puncta at the cell surface, and they dock or associate with another connexin to form gap junction channel that actually allows the metabolites to go through. So what Rahul, one of the very talented PhD students who graduated, he showed that when viral infection is there, connexin 43 retains in the GIC compartment, and there is a continuous loss of 43 in a different MOI post-infection as well as in astrocyte culture. So he said that mouse hepatitis virus infection remodels connexin 43 mediated gap junction intercellular communication in vitro and in vivo. Then Dr. Abhishek Bose, he wanted to ask whether that is a common phenomenon for other cells which also expresses connexin 43. So he took meningeal fibroblast and he also showed that 43 mediated functional gap junction communication also get lost during viral infection. So these two together, they wanted to understand what are the mechanistic standpoint, why it happens. So we know connexin 43 uses the microtubule to go to cell surface, but when virus also uses microtubule by tough microscopy, Rahul showed very nice association of beta tubulin and the viral particle. But this association get lost when there is a viral infection. So from the mechanistic standpoint, we know the microtubule disruption could be one of the way to control the connexin 43 trafficking. So here you can see virus connexin 43 uses microtubule to go to cell surface and dock or associate with another connexin. But when virus is going through the microtubule, connexin 43 cannot go through. So then the question is whether that is true, that connexin 43-47 association get lost during viral infection, and connexin 43 communication may come back because astrocytes is very important cells for cellular homeostasis, but oligodendrocytic connexin 43 may not come back. So with that hypothesis, what Rahul showed that connexin 43, when it comes back at day 30, but in connexin 47, there is a continuous decrease at acute and chronic stage. So he completed that study saying microtubule assisted altered trafficking of astrocytic gap junction protein. Connexin 43 is associated with depletion of 47 during mouse hepatitis virus infection. And he also showed that there is a dissociation of connexin 47. You can see in corpus callosum, anterior commissure, and cerebellum, where there is a loss of myelin sheath, where connexin 47 forms string on bead structures to communicate with each other. But you can see there is a significant loss upon viral infection. So this study actually led to understand that connexin 43 connection is very important during acute stage, and their depletion actually destabilizes oligodendrocytic connexin 47, which is very important for myelination, and loss of 47 is associated with loss of proteolipid protein, which forms the myelin in the neuron and which further leads to demyelination. So then we wanted to see from one aspect, we know microtubular dissociation is one of the factor, but why then connexin 43 retained in the GIC compartment when there is an infection? So what Abhishek did, Abhishek wanted to see whether there is a alteration of the ear stress, because we know virus induces ear stress. So he looked at both ear stress beep marker and UPR response pathway ATF6, and at the same time he showed there is a significant downregulation of endoplasmic reticular protein ERP29. So then we tried to understand, we know 4-PBA is a chemical chaperone, and we know it increases ERP29 expression. So we wanted to see whether 4-PBA treatment can increase the ERP29 and increases the connexin 43, but there are no significant changes in the BIP. So that said that 4-PBA can rescue ERP29 expression. And is ERP29 expression actually help in trafficking of connexin 43 to the cell surface? If this is true, that's what we did. We transfected the cells with ERP29 and make a stable transfected cell line. And it showed that there is a significant upregulation of connexin 43 and they form gap junction channel. But there is much less ear stress, which is denoted by XBP splicing and BIP. So that together said that is one of the things like which is controlling the viral regulation. So that's what we did. We tried to see whether 4-PBA treatment and ERP29 expression can significantly reduce the viral replication. And our answer is yes. That 4-PBA treatment, there is a much significant syncytia formation and different time post-infection, which is much more evident in 12 hours post-infection. Same thing we see when we use the ERP29 transfected cells compared to DBT wild type cells. So these together, we say ERP29 expression reduces MHVA59 infectivity and replication, and that actually is helping us to design the therapeutic targets. Then Grishma wanted to, reviewer asked the question, how do you know that is due to connexin 43? So what Grishma did, Grishma used 2 peptides which actually blocks gap junction channel formation. And she showed when gap junction channel is blocked, then there is a much more syncytia formation, and that says infectivity was significantly increased when connexin 43 channels are blocked using connexin specific mimetic inhibitors. And this study together, we say infectivity was significantly increased when 43 channels were blocked using connexin specific mimetic inhibitor. And we published that paper in JBC saying that the endoplasmic reticular resident chaperone protein regulates ERP29 in murine beta coronavirus host cell response. Then we wanted to see it, we did all the study in vitro. So again, the reviewer asked, can you show in vivo? So what Grishma did, she did lots of immunofluorescence as well as biochemical studies. But in summary, you can see here that when there is no PBA upon viral infection, she treated the cells with until day 5 for acute stage and day 10 for chronic stage. And she showed that without PBA treatment, there is a demyelination and there is a loss of connexin 43-47 communication. But when she injected with the 4-PBA, there is a much significant less of demyelination formation and 43-47 is restored. So these together, we can say 4-PBA has a rescuing effect on 43 post-murine coronavirus infection in primary and astrocytes and reduced viral infectivity. So this study, when we did, we said that from last 2 decades, long studies help in understanding the regulatory role of gap junction protein, connexin 43, and gap junction intercellular communication in murine coronavirus infection, both in vitro and in vivo. And we published a large number of papers and book chapters and invited reviews. And then we also showed in a human coronavirus, when SARS-CoV-2 came, we cannot use SARS-CoV-2 because it is a BSL-3 agent and we cannot use it in our laboratory. But we showed, actually Shovik showed in human coronavirus, which is OC43, it is much less lethal and it causes only mild cold. it infection demodels connexin 43 mediated gap junction intercellular communication. So overall conclusion is that connexin 43 can be a potential therapeutic target for treatment of pan-beta coronavirus infection-induced disease. And though I showed the work, it is just a presentation from mine, but it is a work of my very talented students who graduated for last 16 years in my stay at ISER Kolkata, and now 14 PhD students are working in different aspects. Among these, Grishma and Shovik are directly involved in this project, and I'm thankful to a large number of collaborators, both international as well as in India, and I'm thankful to a large body of funding who helped me to do this work. And thank you all for your patience.
Thank you very much. Absolutely spot on time and a lovely story that you have put together. So it's open for questions, please. So can I set the ball
Sure.
rolling by asking a very naive question? Sorry. Yes, please.
Great work and lots of work. Connexin 43. So if you use that peptide, small peptide, how big I don't know. So that peptide basically can be used as a drug, right? Later on. Whether it also helps for connexin 43 and 47 interaction as well?
Absolutely. Connexin, if the connexin that we showed indirectly, if connexin 43 is lost or connexin 43 cannot come to the surface, they destabilizes connexin 47.
Okay.
So-
So you need a particular stoichiometry.
Exactly, because connexin 47, the phosphorylation requires connexin 43 help. So that's what connexin 47 get destroyed, and they cannot come back even in the chronic stage.
So while you are using peptide, whether any drug that are in market, whether any testing with this drug for connexin 43?
Yeah, one of them is 4-PBA, which is FDA approved. And also there are other beta-glycerotonic acid, which we are working on it. And due to time constraint, I didn't talk about it, but there are several components which can actually block either connexin 43 communication or can block gap junction channel formation.
So in case of influenza also, that can be used.
Yes, that's what OC43 maybe closely resembles SARS-CoV-2, but it can also be done in influenza and other like dengue viruses.
Thank you very much.
Thank you. Sure.
This is just really a very general question. Is you showed the association between the connexins and neuroinflammation and demyelination. So is this unique Is it unique to an infectious organism? So are bacteria also able to, would they be able to also initiate the same? So it could be involved in many other diseases because you see meningitis. And the other is, what about demyelinating diseases? There also is it similar? Because
Yeah.
what you have shown here as a therapeutic target, is that extrapolatable to other conditions also?
Yeah. Very good question. Actually, FDA drug people use in urea cycle, but connexin 43-47 association, which gets disassembled, is also shown in human neurological disease, which is a demyelinating disease in multiple sclerosis, and its experimental model EAE, and as well as neuromyelitis optica, which is also a demyelinating disease. But nobody treated with, they said that there is a downregulation of connexin 43, and connexin 47 is not very well studied because this is very tough to do because human antibodies are not working very well, but there are a large number of studies for 43. And another question, that viral infection alters? Yes, some of the bacterial infection also alters connexin 43 communication. So what we showed in other studies, like, in other neurodegenerative disease, which happens due to bacterial infection, that bacteria can also binds to the extracellular loop of connexin 43. So they cannot bind to each other to form a hexameric hemichannels, and that's why they cannot dock or associate with another connexin to form channels.
Yes, please.
Nice talk, ma'am.
Thank you.
Ma'am, I have two questions. First, have you checked the tight junctions region also?
Very good question. In murine coronavirus, we didn't look at it, but in OC43, we started look at it tight junction protein and cell adhesion molecules also, and we observed that yes, there is a alteration because tight junction and gap junction share this common PDZ domain.
Yes.
So there are alteration in OC43. We didn't check it in MHV yet.
And my second question is, in SARS-CoV-2, there are a lot of reports on the syncytium formation.
Yes.
Have you seen such type of in this murine coronavirus?
There are predictions. Clinicians said that there could be alteration in cell-cell communication, and that's what reviewer asked us to do, but unfortunately we don't have BSL-3. We showed it in OC43, which is a human coronavirus. But we are trying to see whether we can do this work in collaboration with the United States. Lots of lab has good BSL-3 facility, and I have collaborators. But the problem is to do this high-end microscopy in BSL-3 is a challenge.
No, ma'am. In the virus that you were working with, did you find any kind of syncytium formation?
Yeah, absolutely. That's
Yes,
what we showed now.
Oh, I missed it maybe.
I can show you quickly. You can see here.
Oh,
This
right.
is the syncytia, cell-to-cell fusion.
Yes,
And you
yes.
can see this is restricted, but when in ERP29 expressing cell, and that is much evident in this one, like you can see in DBT cells where ERP29 is not there, syncytia is much more large, even in DBT cells which is not treated with 4-PBA. But that is getting reduced when ERP29 is there.
Okay, ma'am. Thank you.
You are most welcome.
Thank you,
Yeah.
Jayashree. I think let's move on to the next speaker.
Hello, ma'am.
I think there is any further.
Ma'am, I have a question. This peptide treatment can this peptide can treat the acute dementia.
Yes, we did it in acute stage, but in dementia, there are some evidences that gap junction channel get lost, especially HIV-induced dementia, but we didn't try, or there are no literature yet. Most of the studies, people said there is an alteration, but the mechanism is not known.
But there is another type of dementia in Parkinson disease and
Yes.
Parkinson disease. But what is the type of treatment in that of disease with the help of this peptide?
These peptides may not work for Parkinson because cell-cell communication, nobody showed that there is a MTP model, MPTP model, which is a Parkinsonian model. There is no evidence that connexin 43 gap junction channel is lost. They said there is an increase of connexin 43, which is opposite to our story.
Thank you, ma'am.
Thank you.
Yeah.
Let's move to the next speaker. Next speaker is Dr. Gopal Shaha. He is a scientist at National Institute of Plant Genome Research, New Delhi. Dr. Jha significantly contributed towards understanding molecular intricacies of blight disease in rice and established that silencing of pathogenicity factors host susceptibility genes is effective in disease control. His work on the isolation of rice-associated bacteria that feeds on fungi has received wide appreciation. And you can see that because of his outstanding work, actually he has been recognized by his peers. He's a fellow of the National Academy of Science. He received Ramachandran National Bioscience Award. He was elected for the Gore Research Conference and also received the Sharma Fellowship from BST. He is also an INSA and NASI Young Faculty Associates. So may I now invite Dr. Jha, please. So, Dr. Jha, as we said that the talk is for 15 minutes and 13 minutes.
Sure. Sure, thank you. It's fine? Okay.
Okay, so
Yeah.
15 minutes time, 30 minutes.
Sure. So thank you so much for giving me this opportunity, and I'm thankful to the organizer to give me this. and the students who have been taking care of me nicely. So I am supposed to take a gear and shift your focus from animal and immune responses, animal pathogenesis and immune responses, to plant pathogenesis and the plant immune response. Okay, so my title says that how to harness a plant-associated bacteria for sustainable agriculture. So, plants are susceptible to many diseases, and we see plants green, happy, but it's not the case. Many times plants get disease. So one of the important disease of rice is sheath blight disease. You can see the rice being necrotic, and this is caused by a fungal pathogen called Rhizoctonia solani. And the challenge is there is no resistance gene known, so it's very difficult to have rice tolerant to this disease. So our lab at NIPGR has been working on various aspects of host pathogen interaction studies. We try to study how this pathogen cause disease in rice and how the plant defends itself and how to modulate this for imparting tolerance. And in the process, I identified a very novel bacteria which has ability to forage over fungi. So in today's talk, I'm going to tell you a story how we are harnessing the bacterial ability to forage over fungi for sustainable agriculture. So I will briefly tell you how this bacterium was isolated, and then I will tell you that this bacteria has deployed a bacteriophage protein to forage over fungi. And I will tell you that this protein has broad spectrum antifungal activity so that you can utilize it for disease control. And another interesting aspect of my talk will be that the plant end up recognizing this protein as an inducer of as a foreign particle, and it will mount a very strong defense response. And that can now not only protect fungal, but also give protection against the bacterial disease. So let's first talk about how we isolated it. As I said you before that I was working on a fungal pathogen of rice, and we were trying to understand the pathosystem. So I was observing whenever a yellow colored bacteria was oozing out from the rice, it was giving protection to the fungal infection. So we isolated the pure culture of this bacteria. You can see this is yellow in color, and this is the pathogen which we are working. Okay, this is Rhizoctonia solani, and initially when this bacteria was interacting, there is a zone of inhibition. You can see this is an antifungal zone. And interestingly, this bacteria subsequently will ultimately forage over the entire fungal biomass. You can see the bacteria has spread over the entire fungal mass. So most of the bacteria which has antifungal ability will have restriction. There will be no man zone, it will prevent each other and will be restricted. But this bacteria has a unique property to spread over it. So this property is called macrophage, and till now there was nothing known how the bacteria can forage over fungi. For the students, I know you know bacteria. Bacteria is a tiny prokaryotic organism, and the fungus is multicellular organism. So it's very interesting how a tiny organism which you cannot see through your naked eye can be able to forage over the fungi. So we started first teasing out the process one by one, and what we observed that this bacterium has ability to grow over to the fungal biomass and macerate it. You can see the bacteria infected fungi is totally macerated, and bacteria induces cell death in the fungi. So we tried to study the mechanism behind this, and what we did RNA-seq analysis, and what we observed that during interaction, the bacteria upregulates one of the particular secretion system, which we call it type 3 secretion system. So this type 3 secretion system is a needle-like apparatus through which the bacterium sends its virulence function directly into the host cell. You can see the bacteria directly injecting into the host cell. So the role of type 3 secretion system during bacterial pathogenesis in animals and plants has been well known, but its role during bacterial fungal interaction had not been explored. So we thought that let's see how this happens. So being a geneticist, we tried to mutate the gene to study its function. So we obtained the mutant defective in type 3 secretion system, and you can see this wild type bacteria is able to completely forage over the fungi, but the mutant which is defective in type 3 secretion system is not able to forage over it. So this showed that the type 3 secretion system is important for bacterial ability to forage over the fungi. So as I said, the type 3 secretion system is a needle-like apparatus through which bacteria delivers its effector protein. So we try to predict what are the potential effector proteins which are secreted through type 3 secretion system. And we identified around 30 such effector protein, and one of them was showing homology to a bacteriophage protein. So for students, the bacteriophage is the bacterial predator which can lyse the bacterial cells. So it was surprising how a bacteriophage protein is being predicted as a effector. So we thought that let's characterize this protein. So again, we disrupted this gene and we observed that this is the wild type, it has completely foraged over the fungi, and the mutant which is defective in type 3, this effector protein is not able to forage it. It's restricted at the center, and when we put back the wild type copy of the gene, it was fully complemented. So this shows that this bacteriophage protein is important for mycophagy. So we have been doing a structural modeling and crystallography, and what we observed that this protein has a pore-forming ability, and it can oligomerize and make a barrel into the fungal cells and lyse the fungal cells. To summarize this, what we have thought that this is a very potent antifungal protein which can be deployed. So here I am showing you that this is the Rhizoctonia solani. You can see the buffer-treated Rhizoctonia is growing happily. And when we treated with the protein, it completely prevented this. And eventually, the protein treatment is able to lyse the fungal membrane. So this protein is disintegrating the fungal mycelia and is showing broad spectrum antifungal activity, but it does not have antibacterial activity. So sorry. So to summarize what till now I told you, that this bacterium during interaction has ability to sense fungi and upregulate type 3 secretion system through which it delivers effector protein into the fungal membrane. One of them I identified to be a prophyceal protein that which lyse the fungal membrane and releases its nutrient into extracellular milieu, and that bacterium is using as a source of nutrition. So this work is published in Nature.com, and you can have the detail of this. So we thought to exploit this for controlling the fungal infection. So now let's talk about how we can bioprospect this protein for controlling the fungal diseases. So we started using this protein as a fungal spray, and you can see this is the buffer treated one. Fungus is infecting, but the protein treated one, the disease is totally controlled. And now we can also use it as a transgene. We can make transgenic plants. We made transgenic rice plants, tomato plants, and all of them is resistant to this fungal infection. So to the best of our knowledge, controlling sheath blight by mere deployment of one gene is not reported. And we are able to demonstrate this, that this gene can be used as a protein or a transgene to control the fungal infection. But the challenge is how to use this for controlling infection in the field, because there are million hectares of rice plant fields and it's very difficult to bulk produce. So that bulk production of the protein had been a roadblock. So we have started thinking, can we have a smaller variant of this protein that we can chemically synthesize and use it for a spray? So what we identified is that there is an N-terminal peptide of this protein that is just 34 amino acid long, and that is good enough for its antifungal activity. You can see this is the buffer treated fungus. It is growing happily. This is the full-length protein-treated Rhizoctonia. It is completely prevented. And just the 34 amino acid peptide is good enough to control the infection. And I told you that the peptide can be chemically synthesized, so you can upscale to the any level what you want, and it can be deployed. So we have started deploying this for the disease control. Then we come another interesting aspect of the plant pathogen interaction, that we figure it out that the plant is able to recognize this peptide as a MAMP. MAMP is microbes associated molecular pattern, and it is well known that when a bacteria infects plants and the pathogen, they will have an immune response against it. And one of the classical known example of MAMP in microbes is flagellin epitope, FLG22 epitope of the bacterium. And it is known that the plants has a well-developed receptor called FLH2, which recognizes this flagellin peptide. And then there is a co-receptor back one, and this interaction ends up triggering a immune response, and that is potent enough for the control. So what we observed that this 34 amino acid peptide of our 9562 protein is having sufficient similarity with the flagellin peptide of this bacterium. You can see out of this 22 amino acid, which is a classical PAMP, 15 is conserved in our peptide. So we tried to test this by synthesizing, altering the sequences alteration in this peptide, and testing it is for antifungal and defense ability. So what we observe that the full length protein or the peptide is able to mount a defense response, and the way we study the defense response is through induction of the callose or through induction of MAP kinase. You can see the protein treatment is able to induce the defense response. This peptide 1 treatment is able to defense response, but peptide 2 peptide, which is we alter the sequence, is not able to induce the defense response. So this tells us that the plant is able to recognize this protein as a defense elicitors. And once the defense response is induced, that is very potent and that can give you protection. So what I am going to show you this, there is a very important bacterial pathogen of tomato called Ralstonia solanacearum, which is causing wilt disease. This is the wild type plant infected with bacterium. It is completely wilted. This is the protein treated tomato plants, and you can see it's completely protected. Similarly, the peptide 1 treated plants is completely protected, but peptide 2 treated plant is not able to be protected. So this shows that this protein treatment is able to induce a defense response, and that is mounting sufficient immunity to not only protect fungal infection, but also the bacterial infection. So our working model is that plants should have some receptor to recognize this peptide, and I told you that flagellin is the well-characterized PAMP molecule, and there is FLS2, which is supposed to be the receptor of FLG22. So using lots of in silico and the wet lab study, we figured it out that the plants have this flagellin FLS2 receptor, and that can bind to this peptide, which I've talked about it. You can see this peptide of this is able to be interacting with FLS2 of the tomato, but not with the another homologs of the FLS2. So our working model is that plant is able to recognize this protein as a MAMP using the signal receptor called FLS2. And BAC1 is important because when I silence this BAC1, which is a co-receptor for the immune response, the plant is able to lose its immunity. You can see the BAC1 silence plant is completely wilted. So whenever the bacteria is colonizing into the rice plant, it is sending this protein into the apoplastic space using type 3 secretion system. And at the apoplastic space, it is recognized by one of the receptor protein, plasma membrane localized receptor protein, FLS2, and thereafter a BAC1, which is a co-receptor, binds to it and induces a series of signaling response that is mediated through MAP kinases, and defense genes are induced. That gives us protection. So the plant has a very strong defense response, and similar to animal immune responses, plants do have a strong immune response, and if we can induce that, it will be good enough to impart tolerance. So to summarize my talk, I talked to you about a very interesting bacterium which we isolated from rice. This is Burkholderia glandula strain NGJ1. And I told you this has a broad spectrum antifungal activity, and it is an inducer of the defense response. So it is like a double-edged function. At one hand, it is directly killing the fungal pathogen, and on the other hand, it is inducing the defense response, and thereby it is giving us protection for the bacterial diseases. So now we are in collaboration with academia and industries. We are developing this peptide-based nanoformulations that can be used for deployment of the fungal and bacterial disease management in plants. So we have MOA signed between Tata Rallies and other companies. They are developing formulation for field level testing. So with this, I come to my end, and I must acknowledge the hard work of my students. They are really dedicated and hardworking students, and they make my life simpler. And due to their hard work, I've been recognized and I've been generously funded. And I'm thankful to all my collaborators. Dr. Sonti has been my mentor, and Dr. Sundaram, who is director of IIRR, ICR Institute, he is helping us in testing and developing this formulation. And also I have interaction with IIT Delhi to develop this nano formulation. So thank you.
Thank you.
Fantastic talk. Use it here. So why is this process called mycophagy, not mycolysis?
Phage is eating actually. So because the bacteria is able to eventually metabolize the fungal biomass.
Okay, okay. So,
So
that's what
because,
was
yeah, so
wondering.
that's why. So imagine during the plant is a very hostile dish. The plant has to grow itself, and then these microbes has to colonize into the plant. So if they start harming plants, it will not be good. They will become pathogenic. So this kind of bacteria has evolved a very unique way of surviving in the plant. So they are not harming plants, but they started using the plant-associated fungi as a source of nutrition to survive there.
Okay.
Actually.
And another thing is, are there, is there any need for other bacteria to be present for these bacteria to survive, whether there is any?
Yes, so that's another interesting aspect, and my time was limited,
Okay.
so I did not talk to it much. So basically imagine what I told you, that the bacteria is lysing fungus. So it is like releasing sugars into the environment. You are making sweets available, opening the sweet shop. So there will be lots of other things which will go, and the bacteria has a huge social ecology. So what subsequently we figured it out. I have another paper in EMBO where we figured it out that bacteria is able to prevent the attack of the other bacteria using another kind of secretion system called type 6 secretion system. So bacteria using type 3 secretion system to forage our fungi, and bacteria using type 6 secretion system to use it as a gatekeeper and protect from the other invader. But at the same time, bacteria is able to have socializers and good neighbors. It is trying to recruit it. So I feel that this kinds of bacteria is really hub bacteria. They can be a good socializer and prevent the intruders, introduce the beneficial ones, and then give protection by immunizing immune responses. So that's our long-term goal and the hypothesis which we have generated. Sir, I've tested that it is preventing the cohabiting bacteria, but it is not killing all of them actually. So it is, imagine rice will have hundreds of endophytic microbes which is growing. So maybe 80% of them it is killing. 20% of them it is not killing. It is killing the pathogenic fungi, but many of the beneficial fungi is not being killed. For example, trichoderma. If you now try to study this association with trichoderma, this bacteria is not killing trichoderma. So we feel that it is able to modulate the microbiome, killing the pathogenic ones and introducing the good socializers. So we need to now increase our level and do whole metagenomic based approaches. So we are on the way, sir. That's another aspect, sir. Yes, I agree to it. We need to study that. That's another complication. That's another variation, sir, actually. Another aspect is the bacteria will have its own flagellin. So why flagellin is not perceived? So because during bacterial interaction, because bacteria knows that the plant will recognize flagellin, so they will shut it up actually during interaction. Yeah, similar sequences. So they shuts up its original flagellin so that it avoids the induction of the defense response. But this protein is important to kill the fungi and ensure nutrition. So somehow they kept it. So that's why these kinds of bacteria does not have very high copy number in plants. So generally, if it is pathogenic, it will tend to have 10 to the power 9 cells and things like that. But these kinds of bacteria will only grow to 10 to the power 3 to 4 cells because they keep their growth restricted into the plants actually.
Gopal,
Yeah.
pretty excellent work here.
Hi.
So that particular peptide that you found out and in collaboration, so you are going to use that as a spray, right?
Yes.
And now the treatment with the protein, how, directly you were
Protein
spraying?
treatment is very effective, ma'am. It is able to fully control it. But the only challenge is bulk production, actually,
Okay.
because there the upscaling fermenters and things like that we don't have, and the process becomes very costly. So that's why we have been thinking to reduce these sequences to be minimal, and we figured it out a smaller peptide that is good enough for doing this job actually. It retains antifungal activity, it's able to retain its ability to induce defense.
And this T3SS system
Because-
is just like Shigella is having another, it's pretty much similar
So
kind.
it's well known in animal pathogens like
Okay.
Salmonella and
Shigella.
things like that.
Yeah.
They use this to deliver into
Right.
directly into the host cell. So similarly,
So
this
any counterpart protein in animal is there, like BG particular number?
ma'am, actually, this bacteriophage protein is conserved. As I said, as you know, bacteriophage is a common bacterial predator. So the most of the bacteria has this bacteriophage protein. The interesting evolution, again, due to lack of time, I did not talk, is the evolution of some 9 amino acid at the N-terminal region of this BG9562. So this bacteria and its relatives has evolved some 9 amino acid at the N-terminal, and due to that only it is learned to be secreted by T3SS. Otherwise, in general, in other bacteria, it is not a T3SS effect.
Yeah, quick question. So you mentioned about the function of BG protein
Yes.
or peptide, and you mentioned it forms spore in the target membrane.
Yes.
Is it based on only AlphaFold prediction, or you have really tested pore formation mechanism?
So actually, we have been trying to crystallize this protein in collaboration with researcher at RCB, the Jain, you may know. So we were trying to crystallize and we failed to crystallize this protein. So till now, we don't have the structure of it. But the AlphaFold prediction says that cryo-EM, if you put this protein, it makes a fiber structure. And when we do microscopy, we see pore into the membrane.
Okay.
So that's why we call it a pore forming structure.
Thank you very much. We will now move on to the next speaker. We now invite Dr. Somdev Dashgupta, who is based in IIT Kharagpur. Somdev is a young upcoming scientist who began his career with a very dynamic career. He got an EMBO long-term fellowship for postdoctoral research in 2009. He was awarded the INSA Young Scientist Award in 2011, and he received the DBT Ramalinga Swamy Fellowship in 2015. His interests are focused on Mycobacterium tuberculosis and Leishmania donovani, both of which are intromacrophagic pathogens, and he uses the CRISPR-Cas technology to explore host factors which are involved in parasite pathogenesis, specifically focusing on the role of phosphatases and kinases. And the work is aimed at identifying host-directed immunomodulatory therapies to restore macrophage function by killing the parasite/bacteria. So you have 15 minutes to tell us your story.
Okay, thank you, ma'am.
And you can see 15 is showing up here, so
Okay.
it's right there. All yours.
Yeah, thank you, ma'am. So to thank SNU and especially Chudipta for giving me inviting me for this talk, and DGC sir, and I'm really honored that I'm presenting in front of the chairs who actually idled during my PhD days as a Leishmania work, as well as Shuzansu sir when he was in IICB. So now I am at IIT Kharagpur working on mycobacterial pathogenesis. So today I'll be stating this work where actually mycobacterial infection triggered S-nitrosylation of KNG facilitates secretion and phosphorylation of macrophage substrates. Now TB. So TB, we are sometimes complacent and thinking that we have cured it, but then it always resurges. And in the post-pandemic, what we observe is that the global burden of TB has increased, and we share the 2/3 of global burden. And in 1 minute, one life is lost in India. Now, the major problem is that 30% of the India's TB-affected population are affected with extreme and total drug-resistant TB. So we need to have, total drug resistance means this is to 17 drugs. So antimycobacterials will not work. So we have to alternative avenues on which we are working on, but then today we will be focusing on the fundamental research that we are doing and not the host-directed therapeutics that we will be working on. But another problem is latent TB. It is believed that 1/4 of the Indian population, I don't know what is the population here, maybe 1/4 here may be affected with latent TB because we are in a healthy atmosphere, living healthy, eating healthy, do not exhibit, but as soon as we get immunocompromised, the TB comes up. So the thorough knowledge of the secreted virulence factors is important. and how they modulate host factors to promote pathogenesis is very crucial. So in context of mycobacterial pathogenesis, what was is a continuously changing paradigm. I say so is because initially it was thought that mycobacteria, when it enters the macrophage, it actually prevents phagosome maturation block, and by blocking the phagosome maturation and its transfer to the lysosome, the mycobacteria proliferates in the phagosome, and thereafter it actually disseminates. But then if actually it finds the phagosome hostile, then it does not divide and stays in a latent condition. These were the two conditions that were initially thought. Next, what was observed is that when the mycobacteria sees that the phagosome is hostile, then it escapes into the cytosol, and then inside the cytosol it can actually replicate and thereafter get disseminated. That's why we can see both CD4 and CD8 presentation with respect to mycobacterial infection. Next, this particular cytosol translocated mycobacteria has also seen to induce autophagy, and all this is actually a process to buy time. Why it buys time? Because the present particular paradigm states that actually it's not a phagosome maturation block, but a prolonged phagosome maturation. So normally phagosome maturation generally takes from 2 hours to 3 hours, but what mycobacteria does is that it prolongs the pathway from 2 hours to 3 hours to 20 to 24 hours. And in doing so actually, it actually acid tolerates itself such that it can later go and fuse with the lysosome, and inside the phagolysosome now this particular inactive TB, the particular mycobacteria thrives and thereafter disseminates, and also this latency still is there, okay, if cannot actually do any of these. Now the question is that what is this? So this is actually quite simplified, but it is not so simplified, but I don't want you to go into all these details, but I just focus again on this particular, the initial phagolysosome fusion or the phagosome maturation process is 2 to 3 hours, whereas what mycobacteria does is that by overexpressing these acid tolerant proteins, it actually it increases this particular phagosome maturation process to 20 hours. Now, why are all these important, and what is this particular factors that are actually doing it? Now, in context of a mycobacteria, an important virulence factor that is expressed is PKNG. But then all these pathogenetic processes, which I call the mycobacterial Olympics, you can say, would only be possible if the macrophage altruism is inhibited. What is macrophage altruism? Altruism is an inherent property of macrophages which are defined to die or die along with the microbes. So if the macrophage altruism is not inhibited, then any of these processes would not happen. So how does mycobacteria prevent this altruistic phenomenon of macrophages? For it, what we have found is that it uses this particular secreted virulence factor called protein kinase G. Now, looking into this protein kinase G, we have seen that this is the domain architecture where the particular kinase domain sits in the middle, and there is an important domain called the rubridoxin domain, which actually facilitates its kinase activity. Inside mycobacteria, this PKNG is known to regulate the TCA cycle and the glutamate metabolism, and also phosphorylate certain factors. But till date, until this research was carried out, it was not known what this mycobacteria secreted PKNG does inside the macrophage. What are the macrophage substrate? Why does it not phosphorylate any particular macrophage substrate, and what does it do? For that, what we found is that initially when we generated a PKNG knockout mycobacteria and infected cells, what we observed is that there was a dampened TNF-alpha response compared to for the particularly when infected with the wild type. So RV, for simplicity, is the wild type TB, whereas the RV delta PG is the PKNG mutant TB. So what we observe is that compared to this particular wild type, the particular PKNG mutant exhibits an increased TNF-alpha response. That means the PKNG secretion dampens the TNF-alpha secretion upon infection of macrophages. And when we infected mice, the alveolar macrophages, and when we observe, what we observe is that for the PKNG knockout macrophages, there was an increased apoptotic phenotype that was observed, as you can see, for the annexin 5 PE positive cells, indicating that this PKNG could be involved somehow in inhibiting the macrophage apoptosis, because the apoptosis was not present with respect to the particular wild type infected mycobacteria, but was only present when this mice was infected with the PKNG mutant mycobacteria. Now, time course study shows that although there was an increase in initial pro-inflammatory response and TNF-alpha production in this particular wild type, but that gradually decreased. But with respect to the wild type, there is an increase in TNF-alpha response, indicating conclusively that this TNF-alpha secretion was dampened, and the dampened TNF-alpha secretion was due to reduced expression of TNF-alpha. Now, what caused this reduced TNF-alpha expression? For that, what we did was a chromatin immunoprecipitation with using the two important transcription factors that regulate TNF-alpha expression, that is ATF2 and NF81. What does use, and what do we see? That the particular sequence to which NF81 binds to the TNF-alpha promoter, that sequence was equally present for both the wild type and the mutant infected cells. But with respect to the ATF2 binding sequence for the particular wild type infected was much less compared to the infected or the PKNG inhibitor treated cells, indicating PKNG inhibitor in pre-treated cells, indicating that this particular ATF2 transcription factor was somehow not able to bind. So this particular ATF2 not being able to bind could be because of two reasons: either a decreased expression of ATF2 or inability, or in the particular ATF2 getting degraded by itself. So what we observe is that the levels, and when the cells were pretreated with the proteasomal inhibitor MG132, we could observe ubiquitinated bands, indicating that this particular ATF2 was getting with increasing time of infection was getting degraded, and thereby it was not able to express TNF-alpha to that level in the wild type macrophages, and hence the dampening of this particular response. Now, what does TNF-alpha do? In an autocrine fashion, TNF-alpha is known to bind to its receptor and induce the this particular activation of this particular procaspase 8 by degrading caspase 8, and thus this one induce extrinsic apoptotic pathway. Now, what we observed is that in case of this particular cells where we actually observe for caspase 8 cleavage assay, we observed that for the particular cells where protein kinase G was expressed, there the particular caspase 8 activity was, cleavage activity was not present, whereas the cleaved caspase 8 was obtained for the PKNG mutant infected cells, indicating thereby that this particular TNF-alpha, decrease of TNF-alpha was important for this dampening of this PKNG cleavage and preventing apoptosis of the cells. Next, what we did was to identify the particular substrates of the particular protein kinase G inside the macrophage, for which we did a phosphoproteomic screen. And using this phosphoproteomic screen, what we did was we identified this as this. We were using this particular approach, which I am not getting into the details. We identified this set of particular proteins, which through scaffold and mascot, and then what we identified at the list listed in the top was PS79 and SOD. Now, what were these proteins? PS79 is a regulator of proteasomal subunit, so or proteasomal degradation. So this hinted that it could be involved in the particular ATF2 degradation, whereas SOD is a protein which is actually known to reversibly bind to the TNF-alpha receptor and prevent the constitutive activation of the TNF-alpha receptor. If I go back to this cartoon, what happens in normal situation is that when TNF-alpha is not bound here, that SOD actually occupies this place, and this death-inducing silencing complex or the procaspase cleavage complex that is being formed by TRAD and FAD is not formed. So when this TNF-alpha binds to this receptor, it opens up and the structural change actually dislodges SOD, and thus allowing this to form. Now, what we hypothesize is that, okay, next, what happened is that when we actually did an in vitro kinase assay, we were at actually missed because this particular PKNG was not phosphorylating my purified SOD and PS79. What could have happened? This is a particular problem that we were as initial setback. Next, we went back to the structure of PKNG and saw that the rubridoxin domain actually harbors 4 cysteine molecules, 4 cysteine amino acids, which actually forms iron sulfur cluster, and it is this iron sulfur cluster actually that is prone to S-nitrosylation. So what is the S-nitrosylation? S-nitrosylation is a post-translational modification which actually controls lot of protein functionality. So we hypothesize, could this any one of these cysteines get S-nitrosylated? When we analyzed in silico, we found that cysteine 109 actually had a very high nitrosylation score. To analyze this further, we did iodide TMT switch assay, where actually we found that only where the particular macrophages where this particular 109 was not mutated, in only those cells the particular nitrosylated cysteine was modified to the TMT, which was detected by anti-TMT, and it is only on those cells that the particular phosphosod and phospho-PSMD9 was observed. And then what we did was we repeated the particular autoradiograph, but in this time we included GSNO. What is GSNO? GSNO is actually a particular chemical which can induce in situ S-nitrosylation of pKNG. So inclusion of GSNO now in the assay buffer of kinase assay buffer now allowed my particular substrate SOD and PSMD9 to be phosphorylated. So this indicated that yes, indeed, this particular SOD and PSMD9 that we identified as the substrates of pKNG were actually the substrates of pKNG. Now, further what we did was that we saw that this particular pKNG, that phosphorylated SOD, and this phosphorylation was dampened if we had treated the cells with this LNMA. What is LNMA? It is actually a inhibitor of INOS. INOS is a particular molecule that upon a pro-inflammatory response generates reactive nitrogen intermediates, and it is this reactive nitrogen intermediates that was responsible for the nitrosylation of pKNG. So this this particular pretreatment with LNMMA prevented the INOS activation, and thereafter prevented nitrosylation of pKNG, thereafter reduced phosphorylation of SOD and PSMD9 was observed. And the same thing was observed for the other cell types as well. Now, moving on to how was this S-nitrosylation happening with respect to RNI or the reactive nitrogen intermediates? What we did is that we took mycobacteria with different, like the wild type, mutant, and the mutant now expressing the wild type pKNG or the kinase dead pKNG or the nitrosylation deficient pKNG. And then we treated this mycobacteria with DANO. What this particular chemical does, it actually induces nitrosylation inside this particular cell type irrespective of infection. And now what we observe is that this particular pKNG was only S-nitrosylated as detected via using this S-nitrocysteine antibody in these IP fractions, where this was this wild type as well as for the wild type expressing mutant as the kinase dead mutant, but not for the particular cysteine 109 mutant pKNG, indicating that the S-nitrosylation was happening on cysteine 109 of pKNG, and that actually induced its secretion into the particular macrophages. Okay, moving further. Sorry. So what we observed is that inside this particular macrophages as well, particular S-nitrosylation of particular pKNG was involved for this phosphorylation of SOD and PSMD9, as you can see here. Now, moving further, what does SOD do? So, functionality of SOD and PSMD9 that we identified. SOD, as I stated, it reversibly normally binds to the particular TNF alpha receptor and prevents the constitutive activation of your constitutive activation of caspase 8. Now, when we actually did a binding study in silico, what we observed is that compared to SOD, phosphosod binds to altered residues and more increasingly, as you can see with the delta G values with respect to TNF alpha receptor 1. And upon infection with wild type, what we observe is that there is an increasing interaction of the TNF alpha receptor 1 with phosphosod for the wild type infected cells, whereas there was no interaction or as there was no phosphorylation of SOD here for the knockout. And on contrary, there was an increase in the particular TRAD interaction with TNF alpha receptor 1. This indicated that for the mutant, this particular interaction with TRAD caused this caspase 8 activation, whereas phosphosod, by binding irreversibly with this TNF alpha receptor 1, prevented the activation of TRAD and thereafter caspase 8, which thereafter prevented apoptosis of the cells. Now, what was PSMD9 doing? Now, I'll not go into these details. okay. Now PSMD9, the other identified molecules, as I stated, was a proteasomal regulator subunit. What does it do is that when we actually observed this particular ATF2 degradation, it was observed only when this particular PSMD9 was phosphorylated. And similarly, in in literature, it was already stated that PSMD9 could actually prevent or could cause the degradation of I kappa beta. What is I kappa beta? If you know, I kappa beta is actually a negative regulator of NF kappa beta. When I kappa beta gets degraded, NF kappa beta gets activated. So if you see here in this microscopic images that with increasing time of infection, the I kappa beta gets degraded in the wild type infected cells and not in the mutant cells. And this particular degradation of I kappa beta causes nuclear translocation of this 65 or the active subunit of NF kappa beta. What does this P65 do? This P65 causes overexpression of the anti-apoptotic protein A1. Well, as you can see in these microscopic images, P65 with increasing time with infection with RV exhibits this particular thingy, the nuclear translocation, and there is overexpression of protein A1. So to sum this up, what we can see is that this particular upon mycobacterial infection, when this mycobacteria enters, PKNG gets phosphorylated, S-nitrosylated. S-nitrosylated PKNG is like secreted into the cytosol. There it actually phosphorylates either PSMD9 or SOD. SOD prevents the extrinsic macrophage apoptosis, whereas PSMD9 prevents the intrinsic macrophage apoptosis by inducing activation of NF kappa beta. As you can see, protein A1 sequesters backs and bead and prevents this particular pathway. So these studies were recently published in PNS and MBio, and yeah, so these are acknowledge the work of Sharadindu and Sadhana, who actually majorly did most of this work, and Arnab and Devan helped them for this. I have for the last 10 years been moving. First, I started my journey at Biomedical Engineering, IIT BHU. Then I moved to Department of, so this was a inter-migration from IIT BHU to IIT Kharagpur. And then at IIT Kharagpur, again, we moved in 2000, during the pandemic period, to our new building that has come up. And we are now there. So I'm indebted greatly to all my collaborators, especially those of JALMA and NIRT, where I use most of my BSL-3 work. And I am also indebted to the funding agencies of Government of India. All the different funding agencies have been handsomely funding me, and that supports me to carry out all this research, which actually involves a lot of funds, obviously, everyone knows about it. So thank you, and I move to questions.
Okay, so this is now open for questions. It's interesting that you work. So when you look at intramacrophages, so what did you infect the macrophages? Where were they sourced from?
The macrophages were, this is what like the normally all the cell culture work that we do is THP1.
Okay.
So and then on the in vivo work was mice infection, and then we isolated bronchoalveolar lavage, and then isolated the alveolar macrophages from there.
So in the clinical scenario, if you have two intramacrophage pathogens, suppose you have Leishmania and you have Mycobacterium, co-infections, it doesn't happen. Do you think that you could link it with this?
No. So co-infection does happen.
They compete with each other, and then you will only get one infection.
The thing is that as far as the present paradigm of Leishmania infection goes, is that Leishmania halts in between. Like the particular the lysosomal transfer does not happen, and it is the in between acid stress that actually facilitates Leishmania to go from chromastigote to amastigote stage. So it does not have to go ultimately to the lysosome, but with respect to mycobacterial pathogenesis, it has to go to the lysosome, because thereafter that's the present paradigm actually, that the particular, and that so co-infection. So we want to say why one hinders the infection of the other. So why Leishmania infection will not allow?
Because you speak about macrophage altruism.
Yeah. So exactly. So the question that whether if the infection of Leishmania is happening in that situation, whether for the same microbe, same macrophage, it will not happen. This is because this particular once one particular microbe has already hijacked this particular, exactly. So the particular microbe thereafter, also their pathogenesis is different and their hijack procedure is different. So since one set of hijack procedure has already begun, that could be in maybe.
I have one query, Somdev.
Yeah.
Here.
Yes. Yeah.
So that particular domain where you have 4 cysteine, right?
The rubidoxine domain.
Yeah, whether that particular cysteine where interaction with iron is there, instead of cysteine if histidine is there, whether it works similarly or not, instead of cysteine if histidine is there.
Histidine?
Yeah.
No, we tried to mute, so the tried to mutate to glycine as well as histidine. If it like, then the particular S-nitrosylation obviously will not be there. With respect to activity also, we have.
So inos activity will
The activity
differ.
is compromised. The kinase activity is compromised to some extent.
Okay, so what is the correlation with that domain structure and inos activity?
Yeah, so what happens is that the inos activity is just irrespective of what inos is prior to. So initially, when any infection happens, there is a proliferative response. So that's M1 macrophage polarization. So what microbe good is that it actually converts this M1 to M2. So that's what's happening. So with respect to the wild type, the initial M1 is required for this RNA production, where inos is there. But thereafter, mycobacteria actually using other activates arginase 1 and but this initial inos that's why when we did a time course of TNF alpha, there was an initial surge and then decrease. Initial surge actually is required for this particular to generate this ROS and RNA, and this RNA then actually goes and induces S-nitrosylation inside mycobacteria, and that S-nitrosylation. So what we believe is that, what we also have a upcoming manuscript on that as well, what we believe is that this S-nitrosylation, like infection-induced S-nitrosylation of PKNG, is giving it a secretory impetus, because this particular PKNG is well involved in regulating glutamate metabolism, TCA cycle, and latency, and all these things inside the mycobacteria. Now, how does it get the impetus that now I have to get secreted and do something inside the macrophage? That S-nitrosylation gives it a trigger. Okay, now that S-nitrosylation has happened on the cysteine, this particular secretory impetus is there. Then it actually binds to a chaperone called SATase, which I've identified. If I can actually quickly show you. Oh, sorry. Yeah. Exactly. So that S-nitrosylation is actually regulating the exit of this secretion impetus is provided to it. Yeah.
Thank you, Sundar.
Thank you.
I think let's move on to our next speaker. So we now invite Dr. Shaha from Presidency University. For many, more than a decade, actually, Avik is working on Epstein-Barr virus associated B-cells lymphoma. And also recently he got interested, which is very important area in cancer, the microbial signature associated with development of several human cancers. It's becoming a new area of cancer research. Avik has actually was awarded the Wellcome Trust DBT Alliance Intermediate Fellowship, as well as he received Ramanujan Fellowship by PhD Government. So Avik,
You have a pointer? Just moving this one and this laser pointer I'm setting in the studio. From this you can notice.
thank you. So Avik, you have 15 minutes and you can see the library.
Thank you. Thank you, Sushantada. Thank you, Mitaledi, for the nice introduction. Thank you, SNU. Thank you, SNU, Amida particularly, and DJC sir for inviting me for sharing our work in front of you. So the title of my talk. Okay, so title of my talk is cellular control of Epstein-Barr virus lytic cycle application. As Sushantada mentioned, that I work on cancer viruses, and Epstein-Barr virus, is one of them. So 20% of total cancers worldwide that are associated with various infectious agents, According to International Association of Cancer Registry, there are 11 such infectious agents that are considered as a group 1 carcinogen, which includes 3 parasites, 1 bacterium that you all know of, that Helicobacterium pylori, and 7 human tumor virus, and this red marked one that my lab has been working on for a long time, that is known as EBV or Epstein-Barr virus or human herpes virus 4. That virus was the first human tumor virus identified in 1965 by Anthony Epstein and Evan Barr from a patient sample sent by Dr. Dennis P. Barkit from an equatorial region of Africa. So Dennis actually thought like an infectious agent because he collected the sample from the malarial belt. But instead, these 2 guys, they found a viral agent that is responsible for this cancer development, now we known as Burkitt lymphoma. So this virus is a double-stranded DNA virus. This belongs to gamma herpes virus family. This virus transmitted through saliva and is also causes or has been associated with multiple oral disorders. 95% of the world population is asymptomatically infected with this virus. That means 9 out of 10, as we are sitting here, we all are actually infected with this virus. However, asymptomatic. But in some conditions, like particularly immunocompromised conditions, this virus has been shown to be associated with multiple cancers, like as I mentioned, that Burkitt lymphoma, Hodgkin's lymphoma, diffuse, a subset of diffuse large B-cell lymphoma, and various post-transplant lymphoproliferative disorders in immunocompromised population, as well as epithelial cancer. As well as this virus also associated with neurobiological disorder or autoimmune disorder like multiple sclerosis, as Joyce Riddhi mentioned earlier in the morning. This virus, as similar like any other herpes virus, has a biphasic life cycle, a latent phase and a lytic phase. So what does it mean by these 2 phases? So latent phase, that means the virus is hiding from the immune system and it is expressing a very small subset of genes, mostly are oncoproteins required for the cancer development. And on the other hand, the lytic phase is actually required for the full replication of the virus or the egression of the virus. So after initial replication in the oropharyngeal epithelial cells, it infects the B cells are growth transformed, through the germinal center, it reaches to the memory B cells, and this memory B cell serves as a reservoir for the lifelong viral infection. Intermittently, during the B cell to plasma cell differentiation, this virus goes to the lytic replication, as I said, and this lytic replication is required for the viral transmission from one cell to the another and from one host to another. So in vitro, we and others developed a model to study the EBV-induced B-cell lymphomagenesis, where after infection, this resting or metabolically quiescent B lymphocyte, they are growth transformed to make or to generate these lymphoblastoid cell lines called LCLs within a period of 28 days. And these LCLs, as well as many EBV positive Burkitt lymphoma cells or many other patient-derived cell lines that can be triggered to lytic replication by several strategies. Like for example, sodium butyrate and TPA combinations, where TPA is a protein kinase C activator and sodium butyrate is an HDAC inhibitor that activates the lytic replication. Hypoxia is also induced viral lytic replication. As I already mentioned, differentiation to B cell to the plasma cell by antibody cross-linking, which triggers the B cell receptor signaling pathway, as well as we and others showed that chemotherapeutic treatments such as proteasomal inhibition by bortezomib, that actually activates the lytic cycle replication. So interestingly, so far there is no specific treatment for the EBV positive B-cell lymphomas. However, the EBV positive B-cell lymphomas are much more aggressive than the EBV negative counterparts. And the treatments are used called as R-CHOP, where R stands for rituximab, that is the anti-CD20 antibody, and CHOP stands for the combination of chemotherapeutic agents. And because, as I mentioned, the EBV positive lymphomas are much more aggressive, a lytic induction therapy has been suggested in which the chemical induced viral lytic replication is actually leads to either direct killing of the virus, or it allows the immunodominant lytic epitopes to be recognized by the cytotoxic T cells, or it allows the nucleoside analogs such as ganciclovir, which is a prodrug, and that become a drug after successive kinesing activities both by viral kinase and cellular kinase that ultimately kills the viral or cellular replication. So from here, it is imperative that to understand that what triggers the viral lytic replication. Okay, what are the cellular? So we asked the question in the lab that what are the cellular factors that are responsible for this switching of this latent to the lytic replication for a therapeutic intervention. So for that, we started our work by looking at the RNA-seq data of latent infection in the B lymphocyte as well as the lytic cycle reactivation by TPS sodium butyrate treatment. But we, in the RNA-seq data, we found a set of genes or pathways that are significantly upregulated during the latent infection, while the same set of genes are downregulated in the lytic cycle reactivation. So for example, like in some E2F isoforms like E2F1, 2, 7, 8, that are actually shown to be upregulated in the latent infection and downregulated in the lytic reactivation. So what are these E2Fs? So from your textbook, you know that E2F isoforms, or particularly E2F1, are responsible for controlling the cell cycle, particularly at the G1 to S phase transition. And among all these E2F isoforms, E2F1, so they have an oncogenic activity, but E2F1 also, in response to a DNA damage stimuli, it can behave as a tumor suppressor protein too. So my work, we actually focus only the E2F1. I will actually skip the parts of the other E2Fs, and let's start with the E2F1. So we hypothesize a very simple hypothesis that E2F1 transcription factor that may contribute to the EBV-induced B-cell lymphomagenesis by controlling this latent lytic switch. So we first confirmed the RNA-seq data by qPCR analysis as well as by reanalysis of several RNA-seq data available in the public domain. And that confirms that, okay, E2F1 significantly upregulated during the latent infection. But at this point, we do not know that which factors that are responsible for this upregulation of the E2F1. So again, I will skip that part and I will switch gear from the latent infection to the lytic replication, and where we showed, I will not go through in details of my the data here. If you have any question, you can ask me later after ending of my talk. So what we showed, as a whole, that EBV reactivation not only in the B lymphocyte but as well as the epithelial model that triggers the E2F1 downregulation. So we used for the epithelial model, we used HEC293 T cells stably harboring the back mid of GFP, and also we used several strategies of EBV reactivation that I mentioned in the chart. That TPS sodium butyrate treatment or MG132 proteasomal inhibition, as well as the IgG cross-linking. So all of this showed that E2F1 is down-regulating. So how it is it happens, right? What are if there is any viral factor that is responsible for this down-regulation. So for that, we need to understand first the lytic replication, which is a temporally regulated. It starts with the BZLF1, which is an immediate early protein, and that triggers the approximately 30 to 40 other lytic genes are involved in the lytic replication. And it starts with the, as I said, It starts with the immediate early and then late gene expression, and then ultimately it forms the encapsulation of the virus and then egression of the virus. BCLF1 particularly is a transcription factor and also belongs to the AP1 homologue, cellular homologue of cellular transcription factor family. So ChIP-seq analysis found that BCLF1 strongly occupied on the E2F1 promoter region. We also found there are 3 AP1 binding sites. We confirmed the data by ChIP-qPCR analysis. After the heterologous expression of BCLF1 in HEC293 cells, it also downregulates the E2F1 expression. And subsequently, we cloned this region into a luciferase-based promoter vector, and our data showed that, okay, E2F1 significantly downregulates BCLF1 promoter activity in a dose-dependent fashion. And by mutational analysis, we confirmed that all 3 binding sites are actually responsible for this transcriptional repression. Interestingly, we did not find from the ChIP-seq analysis, none of the viral latent proteins actually occupy into the E2F1 promoter region. So our conclusion so far is like that during the lytic replication, BCLF1 binds to the E2F1 promoter region and represses its expression. So automatically the question comes to the mind whether E2F1 expression level that can controls the EBV lytic cycle replication. So for that, we first checked several EBV positive lines. We found one cell line, GOE, which has a very negligible E2F1 expression. And in compared to the other EBV positive lines. So we took these 2 cell lines, P3HR1 and GOE, for the successive studies, and we showed that upon EBV lytic cycle replication, GOE cells with a negligible E2F1 expression had a higher BCLF1 expression both at the protein and transcription level, as well as the viral lytic cycle replication that we counted as both an intercellular as well as extracellular level as compared to the P3HR1 cells, which has a higher E2F1 expression level. So definitely this prompted us to take other 2 simple strategy that we knock down E2F1 in the P3HR1 cells, P3HR1 cells, which has a high elevated E2F1 expression, and we overexpressed E2F1 in the GOE cells. So E2F1 knockdown in P3HR1 cells significantly upregulated BCLF1 expression as well as viral lytic cycle replication. This has been triggered by in the presence of the viral lytic cycle inducer. The data was also confirmed by the CRISPR-Cas9 study. And whereas in contrast, the E2F1 expression in HEC293 stably expressing EBV significantly repressed the BCLF1 expression as well as the viral lytic cycle replication, even in the presence of lytic cycle inducer, the TPA sodium butyrate. So this tells us that, okay, E2F1 is responsible for controlling the BCLF1 expression as well as the viral lytic cycle replication. So as the E2F1 is a transcription factor, we wanted to know what are the genome-wide regulation of E2F1 in controlling the viral lytic cycle replication. So for that, we did a ChIP-seq analysis. We found that E2F1 bound in the several promoters of EBV. We confirmed by ChIP-qPCR that it bound to all the viral latent promoters. However, it only bound to the BCLF1 promoter, but not at the other lytic regions. And we cloned this region again in the luciferase-based promoter vector, and the promoter assay showed that E2F1 expression significantly downregulated the BCLF1 expression in a dose-dependent fashion. Time is up. Okay, I need two more minutes. Okay. So also we found mutational analysis that the site 2 is actually responsible for this transcriptional inhibition. So what we conclude so far that E2F1 expression down-regulates the BCL-F1 expression in the latent infection in contrast to the lytic cycle replication. So these two proteins are actually acting as a negative transcriptional loop that controlling each other expression. We also found that cell cycle, E2F1 mediated cell cycle regulation does not affect the E2F1 expression as well as the lytic cycle replication. Also, we found, it's a fantastic study, please give me 2 minutes more. So what we found that, we asked that whether E2F1 or any other E2F isoforms like also responsible for controlling this, the BCL-F1 expression. And the sequence homology study showed that E2F2 has a highest sequence homology, more than 50% among all the E2F transcription factor. And the sequence homology actually are concentrated in the DNA binding domain and the dimerization domain, but it differs in the transactivation domain. So we found that E2F2 is unable to downregulate the BCL-F1 expression, and if you actually delete the transactivation domain of E2F1, it's again unable to downregulate the E2F1 expression. But if you swap this the transactivation domain from E2F1 to E2F2, then it is actually able to downregulate the BCL-F1 expression. That tells that yes, that E2F1 specifically through its transactivation domain is responsible for the downregulation of BCL-F1 expression and controlling the viral lytic cycle replication. So next we wanted to find what are the downstream regulators of the E2F1. We did a RNA-seq analysis, and as expected, several pathways that related to the cell cycle, the B cell activation are significantly downregulated. This is very expected. But our eyes stuck to one gene, that is c-MYC, that is significantly downregulated in response to the E2F1 knockdown. Why we chose this gene? Because recently a paper showed that MYC can control this viral latent to lytic switch. But what we found that these two proteins are significantly correlated in EBV positive lines, that E2F1 knockdown depletes the MYC expression, E2F1 overexpression, upregulates MYC expression. However, in the EBV negative and solid cancer, these two proteins are not correlated in deep map analysis as well as in the TCGA cell line model. E2F1 binds to the MYC promoter at the first exon of the MYC gene. We've confirmed by the chip qPCR, confirmed by the promoter analysis. However, unlike the E2F1, the c-MYC does not regulate the E2F1 expression. But together with the c-MYC and E2F1, that is responsible for the downregulating the BCL-F1 expression as well as the controlling the viral lytic cycle replication. So this is our working model. So we found three important points. As one, I already mentioned that E2F1 and BCL-F1 form a negative transcriptional feedback loop that tightly controls the lytic cycle replication. E2F1 positively regulates the c-MYC expression, which collectively replace BCL-F1, which where is not required, and maintains the latency. And c-MYC does not influence the E2F1 expression, nor does the BCL-F modulates the c-MYC transcription, underlining a unidirectional regulatory hierarchy. So this study is unpublished. I'm going to actually submit in 2 days. And this is my community. Shamoresh actually started this work a long time back, 4 years back. Jayanto took over, almost single-handedly finished this work. Asif, actually our DBT project fellow, worked hard for this project. I also thank Piali for critically reviewing our work. And I also like to thank my funders, DBT India Alliance, DBT, DST, SARP, West Bengal DST, and CSI for funding. Thank you.
Hello. Thank you, Avik. Of course, we want to discuss our data fresh from the oven, right? But we have some we have some lunch is calling, so just pressing one question.
Yes.
The E2F and the c-MYC interaction, what you see here, is it also present in other herpes viruses, or it is only with the related to cancer?
Haven't checked. So as this is a cancer virus, I check other cancers. In most of the solid cancers, somewhere we found a negative regulation, but no positive regulation we found.
So it is special to Burkitt lymphoma and
And EBV positive lymphomas.
EBV positive. Very nice work.
Yeah.
Thank you.
Thank you, Obik. We'll move on to the next speaker.
Thank you.
May I please invite Dr. Siddik Sharkar, who is based at the Indian Institute of Chemical Biology. Siddik did his PhD from IIT Kharagpur, and he is actively involved in developing next generation immunotherapies with a view towards identifying novel cancer biomarkers which can be used for immuno/chemotherapeutics. So he has been focusing on chimeric antigen receptor therapies that can target neoantigens or cancer markers by bypassing the MHC expression. So his focus is on solid tumors like breast and ovarian cancers.
From this point, you can move the slides. For the next slide. This was the next slide.
Yeah, I'm the last speaker, I think,
Yeah.
before the lunch, so please bear me. Good afternoon, everybody, especially the organizing committee, chancellor, students, chair who is sessioning this, and the other delegates, and media personnel, if there are any. So I have joined in ISCB in 2018. It's really a challenge if you are doing some sort of challenging research. So developing biologics, it's a new concept, not a new concept. We have been working in my postdoc for the last 15 years. So for this, it's a old concept. So, but in India, there you will find very restricted number of people working here. So what is biologics? These therapeutics products derived from the living organism, like monoclonal antibodies, vaccine, cell therapies, gene therapies. So this is more a personalized sort of therapy, and there should be like a less side effect compared to the chemotherapy. So if you look about the cancer, we are trying to basically, our achieved goal is to basically treat the patient with have the limited off-side effects, but till we are not achieving that. So whether this sort of new, it will shape the new sort of treatment, whether they will change the paradigm of treatment or not, let's see. So these are the different type of biologics. Our lab focus also with antibody. We also focused with cell therapy as well as the oncolytic virus. So during my postdoc, so let's a glimpse of cell therapy before I will tell about CAR-T therapy. Cell therapy doesn't mean only T-cell therapy. It can be NK cells therapy or any adipose-derived cell therapy also. There are some cells which has a tropism towards the tumor, so like TILs or adipose-derived stem cell can be engineered, and they can be used to basically carry the gene of interest. Like a lot of suicidal gene therapy is based on this cell therapy. So here just a cartoonic picture. This is a genetic modified stem cell. So here you can basically put your gene of interest, which can convert your product into active drug. So in this way, you are basically increasing the active drug in the region of interest. So you are not basically having that off-side effects. So this is not just like a concept. We have demonstrated in the in vivo preclinical model, where we have implanted the luciferase cells into the ovarian cancers, where you can see these are the luciferase cells. Then these are the active tumor targeting. We have modified stem cells. So when you co-register the image, like what you've seen in the confocal, you can basically using the IVIS machines. Okay, you can co-register and see they are going towards the tumor tropism because these cells are corrected from them only. Then whatever, they will be converting your active drug into product. We have basically, so most of the time cancer who are doing this, they see just like immediate effect. Like after 28 days, they dissect the tumor and they say it's working. But what about the post effect? So we have chased up to 6 months. and we didn't find, most of the mice didn't come back. So that is what like the treatment should be focused on, because treatment is not the problem. It's like the relapse is the problem in the cancer. Okay, so we have done that. It has been published. So we have also shown that these are the necrotic cells. These are the stem cells where you can find these regions both by MRI and the other images. So this cell therapy has a like a very targeted effect. So can we do also like using the T cell therapy or not? So before that, a previous speaker had told about the virus. So here, we also in our lab do work with the oncolytic virus. So I will give a glimpse of like biologics, then I will go to the, especially the CAR-T therapy, which I will focus with today. So what we do here, like, do you know, you can basically use, like, it's like the viral-like particles. You can basically make them replicating into the cancer cell. This concept have been there, and it has been also used in the clinics since 2001-2. Okay, so what you can do, you can put your replicating gene, HTAD or hypoxia or any promoter driven their genes that will get replicated. And then you can put your, it's like armed virus. So you can put the secondary genes to have a bystander effect. Not only it will have an effect on the infected cell, but the other cell. That's what we want to have. So here we made that, and we found that way. And also during the time of this carboxyl esterase is mainly a, do you know, these are basically intracellular enzyme. You want to make it secretory. So we engineered them to make it secretory. And this is the graph showing the secretory form and their activity. And we use different type of signaling peptide to make the base peptide. So a lot of engineering work has been done here. So after that, we found that when you are giving the product, it is converted into active drugs. And this is basically 0 MI. Then if people are working in virus, you know the titer. So 0 MI is something like the control. So this is when you are giving, as the titer is increased, the cell is getting lysed, and we don't do in the, do you know, just the 2D model. Because 2D model, anything can kill, we use the 3D model. Okay, so basically this will also give you one step to say whether they are entering into the cell or not. Okay, so you can easily see that when you are giving with the virus, they are lighting the cell, but in combination with CPT11 alone, they are not doing, but when you are giving with the virus, they are lighting. And this is basically one of my talented students is working on this. So next we go, because one of the problem is the infiltration. Solid tumor generally infiltrate less amount of the cells. So whether, because now one of the problem in CAR-T therapy is the infiltration of the T cells, especially considering the solid tumor. We have a lot of success in the liquid-like tumor. So here we show that whenever you are giving the virus, you are able to basically increase the number of the T cells. So it's like the immune cells are getting activated when you are giving the virus. CD8 is getting active, more amount of CD8 is there. And this is just the, so what we do, we give the virus, we co-culture with the 3D culture. Then after 2 days, we just, means we remove all the cells, only take the spheroids, because this and the loose particles will not be there. Then we basically make it a single cell and do a shorting. So this shows that your cells are basically getting inside the tumor. Okay, so this is, so here I am not going this we are soon going to publish. Here we found that one of the problem is the cold tumor, especially the ovarian cancer. How to convert the cold tumor into the hot tumor, that we achieve by using the virus mediation. Then basically, this is a SN38, is a topoisomerase inhibitor, where since I will not talk much. So we also find that they also increase the NK cells activity and also decrease the PD-L1 expression. So it's more this combination process of oncolytic with using the suicidal genes has been used basically to boost the immune system, as well as basically take care of the immunosuppression. So this will be communicating soon. In vivo work is lacked. So whenever we look, whether this is in the clinics or not, okay, you find that a lot of virus of these has been ongoing. Some have been safety has been accessed and using GBM and the other studies. So now the main story comes. So that's why, like, if you see the cancer, now cancer it's a very heterogeneous cancer and molecular subtyping is also there. So they generally go for molecular subtyping, then according the treatment is given. So there are 4 types of subtypes, mainly differentiate, immunoreactive, mesenchymal, and proliferative. So we take the data from TCGA and do our own analysis and see the subtype 3, which is more a immunoreactive subtype, has a better prognosis. What does that mean? So if you can convert the cancer from the immunosuppression to immunoreactive, it have a better prognosis. So we use the same model when you do a data to see that what the cancer looks like in ovarian cancer. We have used almost like 7,000 patient data here. Then we do AI/ML approach, artificial machine learning approach, to bring the prognostic genes. And then these prognostic genes, we do the feature selection using a lot of regression. Mostly validated is the Lusher regression is used. Then these predicted genes give you the value. This value is called molecular prognostic score. This is not like the, and this is just, they will give you the value. And this value, then you basically used it to basically train as well as basically validate your data. And what we come that, we found that the molecular prognostic score, this paper has already been published this year. We found that the molecular prognostic score basically tell you the high risk group. And these high risk group, why I'm focusing on the immunoreactive. If you see this one, anti-presentation is low. That means basically they are immunosuppressive. Most of the molecules are downregulated, MHCs and everything. If there will be not MHC, then who is going to present the tumor-associated antigens? So even there will be the T cells. So you have to know the mechanism. Then, so with this, our approach is to make a antibody or a CAR-T therapy. What we have done, so whenever, if you know, if you are doing cloning, you know size of the plasmid or the lentivirus particle matter. So generally, a antibody having a heavy chain and a light chain. But due to camelid, you have a single chain antibody. So you can basically reduce the size into half. That's why they are also called nanobodies. Now people are using that. So we use the concept of nanobody because at a time you can put 2 antigen recognizing site. That's why the bispecific comes. So one antibody which is giving you the result, actually you can, of the same size, you can have two antibody site. So this is the concept we use for making our CAR-Ts. So we make the CAR-T using antibody. One of the valid marker is mesothelin. So we use against the mesothelin. And the other co-stimulatory signals and the persistent signals we have been used. Why we have used is we didn't start from the scratch because we want to bring this technology to India. So we have to go, what are the markers, where they are going currently. So mesothelin is one of the marker. It is basically found in the ovarian cancer. And this marker is used for CAR-T therapies for phase 1, phase 2. What is the difference what people have used and we have used? What is the nanobody sequence, not the single chain variable region of heavy and light chain. So that's the difference. And also we use another step ahead because recently people are showing that when you are using with the immuno checkpoint, they have a better response. So in a one go, we make the same thing. So that's the difference from others with the, so these are some of the literature showing our concept. The dual CAR-T targeting PD-L1 and MUC16, it is also one of the antigen, have a better response. So these are already in the clinics or in the preclinical phase. So according to the success and whatever the stories, we bring the best out of them. So if you see mesothelin, so the main problem of solid tumor is the heterogeneity, and they basically escape the treatment. It is because, so how do you, and also like T cell because of the persistence and infiltration, these are the issues. So we had tried to basically solve it. First, the infiltration, I had shown you how we can do it by oncolytic virus and the others. Okay, so there are a lot of clinical trials going on CAR-T mesothelin, but ours is the unique. Okay, so why immunocheckpoint is important? If you see, we have generated same intraperitoneal model of high-grade serous ovarian cancer in the mouse model, syngeneic mouse model. Then we collect the cell, then we stain with CD45 positive, CD3 staining, and we found that the cells which are CD45 means the lymphocytes, and those cells are PD-1. They express high amount of PD-1. Okay, so there will be always a PD-1, PD-L signaling axis is there if there will be a T cells presence. So you need to block it. Then what we find that whenever there will be, so this is the ovarian cancer cell. So untreated cell, then we took the naive 2 cells, so you find the PD-L spaces. But when you take the activated T cell supernatant, there is a high amount of PD-L. This says that if there will be a T cell and if they will be activated, cancer cell will basically evade it by counter expressing PD-L1. So this needs to be one of the question or to be addressed when you are using CAR T therapies. That's why we make PD-L1 and mesothelin CAR T. So now if you see when you are able, so most of the time people don't believe about the media and those. So what are the factors? Is it exactly IFN gamma? And IFN gamma is one of the marker for taking when there will be activated T cells, there will be IFN gamma secretion, and IFN gamma secretion basically leads to more amount of PD-L1 expression. Now, this is the current status. So ovarian cancer basically evade the peritoneum, and you find them directly into the in the peritoneum during the time of diagnosis itself, because mesothelin react with CA125, and this basically have a means there will be like a interaction which allow it to come through. And it was also found when there will be high amount of mesothelin, there is a poor prognosis. So we took a cell line with a high expression of mesothelin, okay, this is MD, and a low expression of mesothelin to basically validate a construct. Now how we made a construct? 5 minutes? Oh, sorry. Okay, so we had made the construct, and I will basically show you, this is the construct. We have expressed it into the T cells. This is how we basically schematic make the lentiviral particles in our lab itself. And this is about the T cell. You can see that this is an activated T cell. This is a bispecific T cell. Like if it is C 10 to the power 8, it is going below 10 to the power 6. So I'll say it is below than 0.1%. Do you know? You are killing all the cells. So normally we didn't get more 5 to 10% of the cells. So we had done, because of the lack of time, I will not show much of the data. So we have done bispecific cells. So what happened? We found that in activated T cell, this is the means amount of IL-2 and IFN gamma. You can, because these are the markers for T cell activation, and you can say in the bispecific they are activated more, and same is seen in the lytic activity also. So this is what we have proved it. So this is the 3D culture. And we also stained by doing LAMP1 and the other cytokines and show that bispecific has the much more expression. So this is some of the persistence study. Lack of time, I will not show. So what I will go. So this is one of the slide where in T cells, CAR T cells, you need the memory stem cell to be much more. So because the T cells get exhausted, so we are using anti-PD-L1. So if you see, with antigen and without antigen, the memory stem cell is much more than this. So we basically also find by using the single cell analysis of the FACS result using couple of markers, and found that you are basically increase the stem cell memory by using counter using the anti-PD-L1. So this is where we are landing exhaustion, and this thing I will not go in. So the one thing what I was telling that, that tumor penetration, you can see in the activated cell, much amount of the cells are there. This is then we basically dissect that multicellular, you can say organoid or the spheroid, because these are multicellular. Then you dissect it. Then you feel that dissected region where the lighting is. This is the T cells stained with CFEC or the cell tracking dye, because we want to do very earlier whether they are getting entered, and you find that they are entering inside. So this basically proves the concept of persistence as well as infiltration, which is a major hurdles in the solid tumor. So I am not going to say much. So this, we are focusing with breast cancer, ongoing labs in breast cancer and ovarian cancer. And so this is what we have in our research lab to address these problems. We have the sequencing facility to study new markers and the other associated things. And engineered cells are propagated in the BSL2. We have a chimerics of the antibodies which can be used for CAR-T therapies. This project will be soon funded because do you know if CSI doesn't take the step, who are the next? Because CSI, one good thing is they has the, as far as the infrastructure is concerned, with respect to other institutes, CSIR has that advantage over it. So if we don't take this sort of work which can be translated, I think, so in that regard, this is one of the, recently we get reviewed. So we will have a clinical partner with this, as well as the industry and this mission, and hopefully this will be translated or come into the market very soon. So that we can basically able to give the app, because the cost of CAR-T therapy is almost like 40 lakhs. And if it is like Indian made, and we don't know the other associated things, but we are trying to hope it below than 10 lakhs and to make it affordable to the Indian patients. With this, thank you for your time and attention.
So thank you very much for the benefit of time, and you understand that our blood sugar levels are dropping very rapidly. So I think if all of you agree, your work has so much of translational potential. It obviously there will be many questions that will be asked. I would request that we continue the discussion over lunch. And with that, we come to an end of this session, which was really an excellent session and a fine example of translational research, from neurosciences to cancer biology to infectious diseases. So thank you all for the excellent work that you have done. Thank you for keeping to time, and thank you for giving us a wonderful perspective on your work.
Yeah, but
Hello. Okay, so with this, we proceed towards the felicitation ceremony of our invited speakers and our respected chairs. May I call upon stage our 5 invited speakers, Dr. Sharma, Dr. Jha, Dr. Dashgupta, Dr. Shaha, and Dr. Sarkar. And may I request our chairs for the session, Dr. Chaudhuri and Dr. Chatterjee, to felicitate.
Sorry, we rushed you.
Now, may I call upon stage Professor N. Lahiri Majumdar, Emeritus INSA scientist and mentor of the Microbiology and the Biotechnology Department, to felicitate our chairs for this session. May I request all our delegates to please come up on the stage for a group picture. Please come up on the stage. Yes, all the delegates, please. Now we break for lunch and we come back here in an hour. Please proceed towards the ground floor. And those who have not yet received their lunch coupons, please collect them from the
এখানে খালি লেজারটা তুলে দিতে
Signaling innate and adaptive immune.
লেজারটা ব্যাস
Hello, a very good afternoon. Really, we are, myself and Shamala, Professor Sharar, we are feeling so much delighted to be here today. And we are really grateful for this to our beloved Dhruvoda, Professor Dhruvoda Chakrabarti, Vice Chairman of this university. And
ইয়েস ঠিক আছে 25 probably
in biochemistry. And then he joined same department to carry out his doctoral research under the guidance of Professor Dhruvoda Chattopadhyay, and subsequently received PhD degree in 2003. And then he joined laboratory of Professor Alexander Hoffman and University of California, San Diego. And on return in 2010, he joined National Institute of Immunology at New Delhi. So his basic interest mostly centering around immune response in health and diseases, basically host pathogen interaction. And he's basically investigating how immune surveillance and signaling network can be modulated to alleviate disease, such as cancer and metabolic regulation. Somen is a recipient of Shanti Bhatnagar. Then he's a fellow of the National Academy of Sciences, Indian Academy of Sciences, and Indian National Science Academy. And he's also a member of the Society of Inflammation Research in India. With this brief introduction, I invite Somen to present his talk.
Thank you very much, and it's really a pleasure, and thank you for inviting me here in this Immunotherapy University. This is for me the first visit, and thank you for kind introduction. And today I'll be talking about this title, non-canonical NF-kappa signaling calibrates DC functions in the colon, and this is another part of the introduction, right? So I had the privilege to do PhD under Professor Dhruvoda Chattopadhyay in Calcutta University. I finished my PhD in 2003, then I moved out to UCSD and all. But it's a really nice homecoming in a way, but not in Calcutta University, but in Srinagar per se. And this is my presence. That was my past. This is my present. This is the systems immunology laboratory. Fondly call it the SEAL family. And past and present members, particularly very happy to inform that one of my second PhD student, she will be next month joining IISc Bangalore as the assistant professor. And today I'll be talking about stories that are coming up in my laboratory, recent stories. Naveen and Alvina, they're the two prime mover of this story that I'll be discussing here today. And this would have not been possible. As you see, we do systems immunology. We collaborate with a cohort of individuals. It's important. And particularly for today's work, I think Amit Avasti and Bhabatosh Das and Devarka, they have made some important contribution. And the funding was from Department of Biotechnology, Government of India. So before I go into what we are trying to talk here today, make a simple question. Why study immunology? And these are the numbers that are coming up. Mind-blowing, actually. So if you think of total number of viruses on Earth, that's 10 to the power 31. Someone actually tried to have an estimation. If you put them next to each other, it will be 100 million light years. If you think about number of bacteria in ocean, it's 100 million times more than the number of stars you see in the sky. You take a tablespoon of soil, number of microbial entities in that tablespoon of soil would be more than the number of human beings living in continent Africa. So no question asked, we are living in a very hostile world from outside, full of microbes. Some of them, around 1400 of them, are considered to be pathogen. And guess what? We don't get sick every day. And then the threat from within, something I have heard some talks here today, is connecting to it. It's cancer immunity. So number of mutations from the time you are born and the time you are dying, you will have close to 100 new mutations in your genome. I'm not saying that all of them are necessarily going to drive cancer, but there will be 100 new mutations in each of us. And statistically, some of them would be oncogenic in nature. And this study was done particularly in breast tissues. And you see lot more prevalence of pre-neoplastic lesions that are not cancer yet, pre-neoplastic lesions. And that rate is far surpasses the rate of breast cancer incidence, suggesting that those never became cancer. So someone is also protecting us from within. So in a hostile world outside and a hostile world inside. And this is what it is, that immunology, right? The immune system is protecting us. You have this innate immune systems and adaptive immune system. And today I will discuss how they crosstalk. Now, when I say that the immune system is protecting, it's a very powerful system. Any power has an underpinning that it can abuse. So, this is what you have in the immune system. While it can be protective, it can cause damage too. And I'm giving you a small cartoon to explain what I'm meant to say. What is immune system? Think about cellularly, when you have dangers, you have pathogen-associated molecular patterns, you have cytokines that are coming out because you have sensed dangers. They will activate intracellular pathways with certain dynamics, the green one. And that would orchestrate immune gene expressions. Some transcription factor would be activated in the nucleus in a dynamically controlled manner. That would orchestrate a very controlled immune gene expression program. But if you give a little overt activity, or if you show insufficient activity, both ways it would cause disease. When you overtly activate these immune pathways, cell signaling pathways that are linked to immune response, you would cause immune-related pathologies. For example, that you see in inflammatory diseases, including inflammatory bowel disease, something that I will be discussing here today. But if you have insufficient activity of those pathways, you also pose threat because you will have immune deficiency. So the question that we have asked, how this is so tightly controlled? How do you achieve about the amount you want from a biological system? People have their hypothesis. We, in our lab, has been testing a hypothesis for last 15 years. Something that I'm going to explain it to you. So this is the immune signaling pathway, this magenta pathway. I'll refer to it as magenta pathway, I'll refer to it as a canonical NF-κB pathway. So let's keep it simple, say magenta pathway. This is the pathogen sensing pathway, immune signaling pathway, that would activate immune response genes. An instructive pathway responding to microbial signals. But when you think of cells in their anatomic niche, they are not only exposed to microbial signals. They are receiving signals from dozens of different homeostatic cues, tissue differentiating cues that are activating other pathways. Let's call it green pathway. And last few years, an ancillary pathway. that may not be directly relevant for immune response, but they are activated nonetheless. And last few decades or so, we learned that these pathways are not exactly insulated from each other. They have shared biological entities. They have feed-forward feedback mechanisms that connects them. So one hypothesis that we have developed, nurtured, and began to establish is that these ancillary pathways modify the activity of the immunoregulatory transcription factors, providing for immune control and also may have ramification for disease. So there are pathways, one instructive pathway, and there are not so relevant ancillary pathways. Because these pathways are interconnected, we are proposing that crosstalk between these pathways is how you tune inflammation, how you tune immune response. And when you have a deregulated crosstalk, it leads to disease. And today, we will actually show you what we have shown so far on this aspect, this hypothesis in relation to dendritic cells. So, this is your intestinal inflammation control. And one interesting aspects of those areas, intestine, they have mucosal surface. And we perhaps all know now, we have gut microbiome. So this is a very delicate challenge that intestine faces. They have to be tolerant to those gut microbiome because they are resident microbiome. But also they have to be alert enough so as to elicit a robust response when a pathogen infects. And that's a mucosal immune system that you see in intestine, and dendritic cells plays an important role. So this is your dendritic cells. So in normal courses, they will instruct these regulatory T cells through certain cytokines such as IL-10, TGF-beta, so on and so forth. That's not important. The important part is these regulatory T cells, they suppress inflammation. On the other hand, these dendritic cells would also instruct effector T cells. They would express interferon gamma and so on and so forth. They are very good at combating pathogens. And in a homeostatic gut, these green cells, regulatory T cells, will balance this overt activity of the effector T cells that you may suspect. And that's how you maintain gut homeostasis. More so, these dendritic cells will also orchestrate local IgA in the gut lumen, immunoglobulin A, which would coat gut microbiome, and that is important for eubiosis. That is important to maintain a healthy gut microbiome. Any alteration in the coating would lead to dysbiosis, meaning you will begin to have homeostatically poor gut microbiome that may lead to disease. So coming back, dendritic cells are instructing regulatory T cells or also instructing effector T cells, and that's a healthy gut. In the disease, what happens? This dendritic cell mediated instruction of regulatory T cell weakens. And you have an overt reaction of these effector T cells. And that would lead to inflammatory tissue damage. That would lead to intestinal destruction. And that would percolate into inflammatory bowel disease. Because you have no control over the inflammatory cascades anymore. And this is where a dendritic cell has indeed been implicated. This is a couple of pointers. The point is that people have noticed in inflammatory bowel disease patients, there is a marked reduction of dendritic cells, certain dendritic cells that are known to actually have tolerogenic ability. There are retinoic acid that dendritic cell produce to enforce tolerogenic gut. And people have shown that dendritic cell might be having some impairment in retinoic acid production as well in the diseased gut. And so on and so forth. So the point I'm making that intestinal inflammation would be uncontrolled if you have poor regulatory T cells and more strengthened effector T cell compartment, and people have linked such disbalance to dendritic cell malfunction. And this is where I'm coming with this NF-kappa B system. This has been, it's a signaling pathway. This is an immune signaling pathway. This pathway has been implicated in dendritic cell biology. So here again, coming back to what I said earlier, magenta. This is the magenta NF-kappa B pathway. We call it canonical NF-kappa B pathway. What happens that when you have microbial cues or cytokines, this pathway is activated. It leads to nuclear activation of relay serial containing transcription factor. And they're very good at inducing expression of immunogenic genes. For example, IL-12, IL-6, IL-1 beta, and even interferon gamma. So this pathway is the instructive pathway when it comes to pathogen response. Within the NF-kappa B system, however, there is also a green path. To nucleus as RelB NF-kappa B dimer. This pathway as such mediates expression of lymphokines. So the green pathway, the non-canonical NF-kappa B pathway, that activates RelB NF-kappa B activity, important for immune differentiation. People have not thought that this pathway would be directly relevant for inflammation. However, we and others have shown that these pathways crosstalk with each other. Particularly with the non-canonical NF-kappa B pathway, people have shown that this pathway is important for dendritic cell function. People have shown that NIC in dendritic cell aggravates effector T cell function, particularly pathogenic TH17 response. They have shown that this pathway in DC could also limit inflammation suppressive regulatory T cells. And more so, recent GWAS studies have shown that some component of this green pathway, they have identified them to be associated with human IBD. So we are talking about a green pathway which may not be directly relevant for inflammation. However, it is involved in dendritic cell function. Dendritic cells has been shown to be important for controlling intestinal inflammation, and GWAS study has shown that this green pathway could be involved in human IBD. So we asked if this pathway in DC plays a role in intestinal inflammation control. And this is where we are starting. And this is something we have done. Just single cell RNA sequencing data analysis. You must have heard these days, a lot of those things are coming up. So this is intestine from a mouse. And these green guys, they are dendritic cells. Each spot is one dendritic cell, and we know about their transcription, entire transcriptome. And this is macrophages. So look into the dendritic cell. What we see, as you can see, that these green pathway genes, RelB and NF-kappa B, remember, they were the important mediator of non-canonical signaling. They are expressed mostly in dendritic cell, less so in macrophages. Second, as the this is an animal model of human IBD. What you can do in animal, you can give them in drinking water a chemical called DSS. Intestine, you have this mucin coating. DSS would erode this coating, exposing intestinal epithelial cells and immune cells to gut microbial luminal content. Thereby, it will incite inflammation. So the mucin is gone. Now your gut is exposed to its own microbiota and it will trigger inflammation. This is called DSS-induced colitis studies, and I have seen some poster here also yesterday. This is how you study aberrant intestinal inflammation. It is a rather imperfect model of human IBD, but a model is always an imperfect one, right? So what we see that during this progression of DSS colitis, day 0, day 3, and day 6, in mice, in their gut, we look into the dendritic cell, we see increased expression of so-called RelB important target genes. So not only green pathway components are present in the intestinal dendritic cell, their target gene expression is also elevated as you progress through aberrant intestinal inflammation in mice. So we've established that this signaling is intensified in intestinal disease. during the progression of experimental colitis. Question is it doing something with respect to apparent intestinal inflammation? Yes, we generated a knockout mice strain which specifically lack this RELB, the green pathway transcription factor in dendritic cell. This is done using flox and tree mice by crossbreeding them. You generated this RELB delta CD11C Cree. These mice, they lack expression of RELB specifically in CD11C cells that are dendritic cells. And what we see when you challenge these mice with DSS, you see the blue line, this is wild type mice. DSS aggravate intestinal inflammation. This would lead to tissue damage and then mice will lose water. There will be diarrhea, there will be rectal bleeding, and you will see profound body weight loss. That's the symptom, that's the attribute, macroscopic attribute of DSS colitis in mice. And you see these knockouts were rather resistant. They show far less obvious body weight decrease during the progression of DSS colitis. So we are saying that this green pathway is activated in DC during the progression of DSS colitis, and if you knock out the green pathway in DC, you are protected from DSS colitis, right? Then the question is why? How is it connected to experimental colitis? So we looked into RNA sequencing again. We compared bone marrow derived dendritic cells. I mean, it's again, every model has its limitations, and Dipiman is sitting there and staring at me. So, bone marrow derived dendritic cells, we compared wild type and knockout dendritic cells, meaning RELB knockout dendritic cells and wild type, and we performed a transcriptomic analysis to see what genes their expressions are altered if you delete this pathway in dendritic cell. And this is a geobiological pathway enrichment analysis. What we did, these are differentially expressed genes, and you are asking what pathways are enriched in these genes. What you see is that retinol metabolism is one of the most affected pathway when you knock out this RELB in dendritic cell. And this is the retinoic acid metabolism and dendritic cell. Retinols are converted into retinoic acid very efficiently by dendritic cell, and these are the enzymes, RDH and RALDH group of enzymes. They help dendritic cell to convert retinol into retinoic acid. In the intestine, dendritic cell secreted retinoic acid is very important because it promotes, in one hand, these regulatory T cells. Remember, they were suppressing inflammation. So dendritic cells secreted retinoic acid would promote regulatory T cells, the guardian of the gut, in the intestinal niche. Second, this retinoic acid will also support IgA positive plasma cells. They support plasma cells to make IgA in the gut. So this is what we actually then asked, if this enzyme and their expressions were dependent on this RELB transcription factor in dendritic cell. And the result was very clear, that we found out that in absence of this RELB or NF capability of the green pathway component, there is an elevated expression of RALDH2, an enzyme that converts retinol into retinoic acid. So we reasoned that in our knockout DC, maybe they are making a lot more retinoic acid and thereby supporting regulatory T cells in the intestine. So looked into the intestine. And this is the intestine data. Coming directly from the intestine, what we see that even in the intestine, this is an enzymatic activity assay. We found out that the knockout dendritic cells, or in that knockout mice, dendritic cells had lot more RALDH2 activity. And again, consistently we found they had lot more regulatory T cells. This is lamina propria, this is mesenteric lymph node. These are the two important component of the intestinal immune system, or niche of the intestinal immune system. What we see that in our knockouts, there are a lot of regulatory T cells that suppresses inflammation, presumably thereby mitigating DSS-induced inflammatory pathologies. How about the IgA secretion? And this is the data. We found out again in our knockout, there is a lot more IgA positive cells, and so as to that have an increase in the fecal IgA level. And as I said, this IgA, immunoglobulin A in the fecal, in the lumen, would code gut microbiome, and it has an important role to instruct the composition of the gut microbiome. And here you see that in our knockout, there is an increase in the firmicutes. They're inflammation suppressive. They are health promoting gut microbiome. So taken together, that in your knockout, you have more retinoic acid synthesis that is promoting more regulatory T cells and more good gut microbiome overall, leading to less pathology in DSS colitis model. The question is, what links this non-canonical signaling to retinoic acid? There are 3 transcription factors that have been implicated to control RALDH2 synthesis in dendritic cell. One is NOTCH2, another is IRF4, and the third one is beta-catenin. We just simply compared them by Western blot, and the data was very, very instructive. That in our knockout, you have a lot more beta-catenin that may have led to a lot more RALDH2 expression. And indeed, by using inhibitor to show that beta-catenin, heightened beta-catenin expression is what was driving high RALDH2 synthesis in our knockout. The question again comes, obviously, what connects this non-canonical NF-kappa B to beta-catenin? To cut a long story short, this is what we found out, that this green pathway, which would otherwise mediate immune differentiation gene expression, controls the production of auxin protein. Auxin promotes beta-catenin degradation. So in absence of the non-canonical pathway, you have lesser auxin, more beta-catenin, that then drives in this tolerogenic beta-catenin RALDH2 axis in dendritic cell, leading to less colon pathology in mice. But animal studies, is it relevant for human? We looked into this before that, but this is very correlative. We even performed a causal study, because this is our model that this pathway through beta-catenin controlling tolerogenic property of the DC in the gut. Because in knockout, we have more beta-catenin. Was it causally important to give us the phenotype? And this is the data I should be very proud of. Alvin and Navin, they spent a whole 1 year. They got another mouse strain. This is beta-catenin flox mice, and introduce an haploinsufficiency of beta-catenin in their knockouts. And in this knockout strain, you see again RALDH2 activity is falling down. Once you remove at least one allele of beta-catenin, you see less beta-catenin in the knockout. RALDH2 expression is falling down. Regulatory T cell frequency is falling down. Fecal IG is falling down. And yes, animals are showing again as much cholitogenic sensitivity as you see in wild type. So causally linking beta-catenin to this. And then the final is this, we are showing it in IBD patients. Again, that you see the signature of the non-canonical NF-kappa pathway in IBD patient in their dendritic cells derived from their colon, and also a signature of, this is evident from their non-canonical NF-kappa pathway target gene expressions. So overall, what we say, that this green pathway, which is not directly involved for inflammation, crosstalks with the beta-catenin RALDH2 axis and modulate intestinal inflammation. This pathway is aggravated in the dendritic cell, suppresses tolerogenic beta-catenin RALDH2 axis, leading to more inflammation. Conversely, when you knock out this pathway in mice, you see more strengthened beta-catenin RALDH2 axis, which would lead to lot more regulatory T cells and eubiosis, and thereby prevent disease in animal. And obviously, immediately you think that this is a very interesting discovery, because green pathway is not pleiotropic, not ubiquitous. So it can be therapeutically targeted. You have shown that this pathway is even relevant for human IBD. So why not think about targeting this pathway to alleviate inflammation in human patients? Well, before you do that, you want to ask the question if this pathway is otherwise involved in other immune functions of the gut, right? And this is the experiment we did. We examined if this green pathway in dendritic cell, while important for aberrant intestinal inflammation, promote gut immunity or not. And this is the last few data that we got. I'm going to show you. This is the first shocker. You got these knockouts, and then what we do to understand gut immunity, you infect them with a gut enteric pathogen called Citrobacter rodnicium. Its human analog is enteropathogenic E. coli. But this is more of an academic interest to understand gut immunity. You infect mice with Citrobacter rodnicium and then study them. What we see that in these knockouts, In the wild type, you see this is fecal bacterial count. If the count drops down, that means you have performed a good immune function so as to limit local infection. If you don't limit local infection, those bacteria will fall colony and will be excreted in the fecal matter, right? So in wild type, you see these blue bars, there is a gradual decrease in the bacterial count in the fecal matter. In the knockout, they didn't go down. When you look into the body weight changes again, wild type, this Citrobacter infection typically causes a self-limiting symptom. So these blue guys, they are recovering very well. And these knockouts, they show progressive deterioration. So this pathway, RELB, or the green pathway in dendritic cells, seemingly directs gut immunity against pathogens. How? And this is again trying to summarize it for you. These are the few important players when it comes to gut immunity. That in dendritic cell, when they perceive pathogen infection, they secrete this cytokine. Remember IL-23. Very important, this is secreted cytokine that instruct Th17 cells, this is an effector T cell, to produce IL-22. So IL-23, Th17, IL-22 axis. These will instruct antimicrobial gene expression in the epithelial cell and they will eventually get rid of the pathogens. Okay? Now, this is what we saw in our knockout, a reduction in the IL-23, a reduction in the Th17, a reduction in IL-22 in the colon. We performed measurement of these cytokines in the colon explants. We see they are reduced in our knockouts. So this axis that is so important for gut immunity, IL-23, Th17, IL-22, this axis is somehow weakened. And that led to impaired or reduced epithelial antimicrobial gene expression. These REG proteins are very important antimicrobial protein. They are important, epithelial cells express them to fight pathogens. So while this non-canonical pathway provokes inflammation, it seems to play an important role in orchestrating gut immunity. So why in our dendritic cell knockout you have a reduction in IL-23? And this is a very exciting data that I'm sharing. We performed this RNA sequencing study, again, single cell RNA sequencing study analysis. So these are dendritic cells in the colon. Particularly, these are CDC2, a particular dendritic cell subset. You see, there are within the dendritic cell, there are 3 or 4 compartments, right? And in RELB knockout, this guy is gone. And turns out, this is the subset that only makes IL-23. You see, these are the 4 compartments, 4 subpopulation, C1, C2, C3, C4. Clearly, C3 is absent when you don't have RELB in dendritic cell. And when you look into these subpopulations and measure IL-23 expression, only C3 makes IL-23. And this is called another thing, this is called velocity analysis. What they are trying to show you here is that these red guys, these guys, they are differentiating into C3 with a precursor product relationship, suggesting that in the intestine, these guys become a specialized dendritic subset. RELB supports this differentiation to produce or to maintain a specialized dendritic cell subset that would make IL-23 upon infection. And we have now performed some experiment to supplement IL-23 in our knockout, and they fend off pathogen very well. So this is where I'm going to conclude with a significant statement. The significant statement being that the pathways do not act alone. Actually, on the contrary, crosstalk between different pathways are perhaps a dominant immunoregulatory mechanism. Here today we show that the non-canonical NF-kappa pathway, I'm alluding to as a green pathway, crosstalks with beta-catenin in RLDH2 axis to modulate intestinal inflammation. And that raised the hope that you can target this pathway using pharmacological inhibitor to alleviate gut inflammation. But then you wondered if this pathway could also be important for immunity. And we found out that this pathway is actually important for gut immunity as it instructs and particular specialized disease subset that is important for IL-23 production. So this is where we are. You can put RELB, you can put name of any other factor, X, Y, Z, it doesn't matter really. Magenta green is okay. Canonical, non-canonical, NF-kappa B is not important. The concept stays that you have a factor that is important, that is driving deleterious inflammation, and you thought of inhibiting it, but that is also important for protective immunity. And this is where I'm going to say, finalize my talk saying that this is the precise drug dilemma in the era of system medicine. That interconnected pathways and a network that guides phenotype poses extraordinary challenge for therapeutic targeting, because there is always another side of the coin. When you think that this pathway is deleterious, you find its role that could be protective. When you think that this pathway is protective, you see too much of an activity of that pathway could lead to some disease. So in a way, this again compels us to think that more studies are to be done so as to get to a finer detail. For example, the one I'm showing is a particular IL-23 producing subset. Can you target this pathway barring that subset? Can this be achieved? So this is where I'm going to stop here today. But thank you very much for your attention, and I will take up questions.
Thank you, Soman, for your wonderful presentation. It's a kind of story. So now the floor is open for discussion. So let's start with Kaushik.
Soman, as usual, fascinating talk.
Oh, sorry, yes.
One question. So the canonical and non-canonical pathways that you are describing, so maybe I missed that, but is it like this, that a same cell, if we talk about one single cell, is getting the signal and evoking the or selecting choice between canonical and non-canonical pathway, is the choice open for a particular cell, or that choice is restricted when the subsets are decided? Because ultimately signal is decided based on the cell surface receptor.
Very good question. Yes, we have, first of all, we have been discussing cell intrinsic crosstalk. So let's assume that a pathway where both the pathways are activated in the same cell, because they are biochemically interlinked, the outcome of response A and response B, there will be somewhere in middle, right? But we have seen that there are cell types which would foster this crosstalk because of the expression of those mediators of the crosstalks are different. Vis-a-vis, there are cell line which would evolve mechanism, sometimes I would say kinetic insulation mechanism, to keep these pathways separate. For example, our earlier studies which looked into canonical and non-canonical NF-kappa B crosstalk, we found that that's not relevant for macrophages. And here also we looked into that crosstalk first, but there is no crosstalk evidence in dendritic cell between canonical and non-canonical NF-kappa B. What we found instead, that the non-canonical NF-kappa B is cross-talking with the beta-catenin axis. But yes, there could be cell type specific and anatomic niche, the receptor that you're getting at. I would get into the ligand actually. That anatomic niche would have different kind of ligand reporter that can influence the crosstalk. Yes.
It's very nice talk, sir. So I have a question starting the last slide, the drug dilemma. So we know a lot of metabolites, they can activate. So why don't we try with the metabolites that are produced from the gut microbiome? And have you tasted those microbiome, the metabolites that they produce, enhancing this pathway to cure the disease?
Yeah, this is again good point. We have not done anything so far. Pharmaceuticals, this would require a bit different degree of different interventions. But in my laboratory, last 1 year, we have been putting a lot of emphasis on it. The reason being, as you have said, that metabolites provide a lot more opportunity for intervention than pathways itself, right? So, for example, when you say retinoic acids, right? Would you think of suboptimal use of retinoic acid and non-canonical pathway inhibitor to have some intervention? So when I began to think of those combinations, particularly involving metabolites, there are possibilities to move it forward. We are doing something in that direction. Currently, we know that this non-canonical NF-kappa pathway could have implications for, I think, lipidemia. So the pathway in gut may provoke high degree of lipid absorption. So we are looking into this. We are getting there. What you are, the other part that you are asking, gut microbiome derived metabolite, it is an excellent question. People are working on it. We have not done so far. It's an excellent question.
So I have done some work that I'm going to present. I
Sure,
hope.
sure. I'll look
Thank
forward to it. Thank you very much. Deeply.
Yeah.
So,
Okay, Joyce
yeah.
is there?
Oh,
okay.
Please go ahead. Please go ahead.
I'll give Joyce three.
So we were just discussing. So Dipanjan and me. So there must be studies that have looked at canonical NF-kappa in IBD models. So what was the results? Because I don't know. I'm just asking.
Oh, the genome scale.
So if you get rid of the relay.
Oh, this is it. So by the way, this is a good
disease.
question. I'm going to address it. I just need 1.5 minutes. Jiva study surprisingly did not show anything from the canonical compartment, show only thing from the non-canonical compartment. Because I think this is going to be too fatal, I believe, if you have some jivas which is going to give a very strong canonical activity. In animal, when you take relay from the epithelial cell, you might suspect some spontaneous colitis because this pathway is also important for cell survival. This study has been done.
That is true.
So this study has been done. I think I forgot his name, but yeah, this is done. We have the flux mice with us. We know this is true. So the problem again, coming back to ubiquitous and pleiotropic, this pathway is doing too many important things to be targeted.
That's why you can't really tinker with
Yes,
it.
that's why we're going to the green one. Anyway, if you have some hope there.
Okay, Joyce Sri.
That NF-kappa B has a pleiotropic role, right? So now you are telling it is for protective immunity. But how the cross between innate immunity to the adaptive, or it is only for the protection of the gut?
No, I think that there is two NF-kappa B. The red one is, it is everywhere, right?
Everywhere.
It is everywhere. It is important in the inflammation. It is important for proliferation, survival, so on and so forth. For the non-canonical, if I can narrow down the question or the scope of it, the green pathway is particularly involved in lymph node development, lymphocyte trafficking, some part of B cell maturation. Now we know particularly a very specific subset of dendritic cell. And that is what the appeal is, that you can neutralize this pathway for a span of time to get some disease alleviation. But the question that you were asking, if I think, what is the question again? Sorry.
My question is that inflammation is there, right? There are lots of innate. Yeah.
So the crosstalk between this, the one actually I'm showing, that inflammation is driven by NF-kappa B activity. That would bring those affected T cells at the site of damage, right? And that is true for IBD also. We have done this in earlier work published in 2021. Epithelial NF-kappa B activity would drive those affected T cells, and if you do too much of it, you will have too much of those affected T cells, and that will cause a vicious cycle. That will trigger a vicious cycle, right? And also in these affected T cells, TCR activates NF-kappa B, not only TLRs, right? BCR activates NF-kappa B, and people have shown genetically that these activities are important for those affected functions as well. And this is where I was saying this is a hopeless thing. If you think about intervention, if you think about academic interest, that's a bit different.
No, thank you.
So any student from the back? No?
No, I'll grab them.
There is one from back.
Good for you, because otherwise I'll talk to you in the tea session.
Sir, it was very interesting talk.
Thank
I
you.
have only one question. According to theory, retinoic acid also inhibits T helper 17 production, like proliferation. So, it also inhibits ROR gamma T cells. So, if whatever you said, like retinoic acid increase is happening, is it relating to this?
Yes. I got your question. So yes, retinoic acid broadly changes the balance between Th17 and regulatory T cells, right? One being inflammatory, then latter being the anti-inflammatory. And yes, people have proposed retinoic acid would directly impact Th17 cell. Vis-a-vis the other one, that they are saying retinoic acid would promote regulatory T cell, which will make tons of IL-10. That would be detrimental for Th17. That will skew the Th17 compartment. And we have studied in our model, and I think we have pretty a fair amount of confidence to say that independent of TH17 contribution or this crosstalk between TH17 Treg, DC-derived retinoic acid directly impacts regulatory T cells, independent of what the compartment size of TH17 may have to inform, right? Thank you.
Okay. I think there are no other questions. So, Showman, would you
Thank
give
you.
me an opportunity?
Yes.
Okay. My question is that REL-B. Okay. So if you knock down REL-B, so the IBD apparently, if the gut junction is also, those proteins are also somehow
Very good.
dysregulated. So that also cause you IBD, apart from whatever you have shown.
Yes, this is a good question. I think the story, I think this is why sometimes I feel reluctant to go into the disease and commenting on it. I have in this presented work today narrowly focused on the role of REL-B in dendritic cells. There are other compartments. Our previous work suggested NF-kappa to the other part of the green pathway. When you delete it in the IECs, intestinal epithelial cells, that improves experimental colitis. So they are both in the same direction, that you delete REL-B, you improve experimental colitis. You delete NF-kappa between epithelial cell, you improve experimental colitis. So it gives you a very strong appeal that if I inhibit this pathway, I'll improve experimental colitis. However, last year we deleted REL-B, not NF-kappa2, in epithelial cells, and it is exactly looking like what you're saying. We have reason to believe it is sort of disrupting the gut barrier, even and worsening DSS colitis in mice. So it's a lot more complicated at this point, and this could be for different functions that we need to fully understand.
Okay. So thank you, Showman.
To my postdoctoral research. Our understanding of infection biology and immunity. She has received several floor is yours, Ornika.
Hello. Okay. Thank you, Tanyadi, for the nice introduction. And thank you, Shamulda. And thank you, thanks to the organizing committee for inviting me here. And thank you, sir, Professor Dhrubajyoti Chattopadhyay. And good afternoon to all of you, all the distinguished scientists and the students. And My lab is majorly focused on looking at how innate receptors, they coordinate to modulate inflammatory outcome. So towards that, a very recent work that we have done. So I'm going to present that. And there also, like, we have looked at the innate receptor coordination, and we have taken gram-negative enteric bacterial ligand, Vibrio cholerae cytolysine, for the study. So this work is majorly done by Shraddha, and this manuscript we just submitted the final revision. And these two students, Sindhura and Akanksha, they have also done part of this study. And this is in collaboration with Kaushik's lab, Professor Kaushik Chattopadhyay from ISER Mohali. So they have done a lot of work on the structural and biochemical aspect of VCC. And we were interested to see how VCC can modulate the inflammation. So VCC, or Vibrio cholerae cytolysine, this is a pore-forming toxin, exotoxin, and this is secreted as in the pro form. And upon chopped up of the pro domain, the VCC monomer, and when it gets attached or it sees a mammalian membrane, so it attaches and make the and upon oligomerization, it forms a pore. And upon forming the pore, it can cause the membrane damage and killing of the host cell. And this Vibrio cholerae, it causes cholera, and the major causal or agent for the cholera is cholera toxin. So in cholera toxin deficient strain, this VCC, this play a very important role. It causes inflammatory gastroenteritis. So we, as I mentioned, that we want to know how VCC modulate immune response or the inflammatory outcome. So we have taken dendritic cell, and now, like, after listening to the talk from the from the morning, you know how dendritic cell is very important. So dendritic cell, these are sentinels of the immune system, and they contribute both in the innate immunity and shape up of the adaptive immunity as well. And it is in the body, in absence of any infection or inflammation, it remains in mature condition, and upon activation, it forms the mature dendritic cell, which is a professional antigen presenting cell. And so what we have done, we have differentiated dendritic cell from bone marrow, and then we have treated the bone marrow dendritic cell with VCC. And we first looked at the cell viability, and we saw that different doses like 10 nanogram per ml and 100 nanogram per ml. And so around 10 nanogram per ml, there is around 40% viability. And using that concentration, we observed that there is also IL-6 is a pro-inflammatory cytokine, and there is a significant amount of IL-6 production, suggesting that VCC can activate dendritic cell in terms of production of pro-inflammatory cytokine. Now, as Shaumin was mentioning, that there are this PAM, pathogen-associated molecular patterns, which activates the receptors, and there is transduction of the signaling, and ultimately there is activation of NF-kappa B and production of pro-inflammatory cytokines. So as I also mentioned, that I am more interested in looking at like how the innate receptors they coordinate. So what is the receptor which is causing this pro-inflammatory cytokine production? So it could be TLR or toll-like receptor. Now toll-like receptor is very well-known classical pattern recognition receptors. And these toll-like receptors, they are either present on the cell surface and also present on the endoplasm. So as VCC is a secretory toxin, we were thinking that it could be the surface TLRs which are involved. So let's see like whether TLRs are involved. Now TLR mediates their signaling via mediated adapter molecule or TRIF. Now, if MyD88 is involved, then for sure the pattern recognition receptor is the TLR. So at first we check like whether MyD88 is involved, and we observed that yes, in the MyD88 knockout DC, so there is a significant decrease of IL-6 production, suggesting that TLR is a PRR here. So TLR, they generally form hetero or homodimer, and they have this leucine-rich repeat domain by which they bind to the ligand, and then they have this TIR domain and by which they recruit MyD88. This TIR homotypic in that in TLR2 deficient dendritic cell, there is no decrease in IL-6 production. However, in TLR1 deficient case, there is significant decrease of IL-6 production, suggesting that TLR2 is not involved, but TLR1 is involved. Now, it's okay, but it's not okay. Why? Because as I mentioned, the TIR domain is important for recruitment of MyD88. Then only the induction of signaling can happen. Now, TLR1, it does not have a functional TIR domain. So that suggests that TLR1 cannot recruit MyD88. So that is why TLR1 and TLR6 also has the same case. It does not have a functional TIR domain. So that is why TLR1 and TLR6, they can bind the ligand, but they need this TLR2 to form this TLR2 heterodimer, so that with TLR2 TIR domain, this MyD88 can be recruited. So here, interestingly, TLR1 is there, but TLR2 is not there. So we wanted to know that some other TLR which can lend this TIR domain, or let's see like if some other TLR is involved. So we have used TLR4 mutant. So this C3HEJ, say this is not TLR4 deficient, but it has a mutation in the TLR4 signaling region. So it cannot induce the TLR4 signaling. So when we used this C3HEJ mice, then we observed that there is a significant reduction of IL-6. Almost IL-6 is not forming. So suggesting that TLR4 is very crucially involved in the signaling mechanism. So now this we found that TLR1 and TLR4 both are important, but TLR2 not. So now we wanted to know that TLR1, which is deficient in the TIR domain, so is it that TLR1 and TLR4, they are forming the heterodimer and TLR4 is lending its TIR domain to induce the signaling? So to address that question, we used HEC293 overexpression system. So HEC293, it does not have surface expression of any of these TLRs. So we can use HEC293 cell and we can overexpress any of the receptors and we can check the response. So what we have done, we have cloned the receptor genes in pcDNA 3.1 vector and we have transfected, and we have then treated the cell with VCC monomer and looked at the NF-kappa B receptor assay. So what we observed that when we have transfected with the TLRs individually, there is almost no response upon VCC activation, but wherever like we have transfected both TLR1 and TLR4 together, there is a huge signaling response. So suggesting that yes, TLR1, 4 together only can induce the signaling. So probably TLR1 and 4 is making a heterodimer. Now, as I mentioned earlier, that this monomer can form the oligomeric pore, and there is this disintegration of the cell is killed and the disintegration of the membrane. But this disintegrated membrane, they are housing this preformed oligomers in it. Now this transmembrane oligomeric, this VCC, they can act as a dam and can induce some cellular responses. So to know this, what we have done, we have used this artificial like lipid membrane, like liposomes. We made it and we have incorporated the VCC in it, and it forms a transmembrane oligomeric structure. Now these oligomers are very stable, very resistant, that we have seen. And then what we have done, we have treated this in macrophage, and this is a study we have done in 2014. And we have seen that with this transmembrane oligomer, in case of macrophage, the responses is generated via TLR26. Now, in dendritic cell with the monomer, we are seeing the response is getting generated via TLR1 and 4. So we thought, like, what is happening? Let's say, like, maybe the oligomer is can induce the signaling via both TLR26 and TLR14, and what it does in case of dendritic cell. So we have treated the dendritic cell with the preformed oligomer, and we observed that in case of dendritic cell, the preformed oligomer only induces signaling via TLR14 heterodimer, not by TLR26. Then we have done again this receptor coordination assay, and we observed that in case of oligomer, so both TLR14 and TLR26, so here you see that TLR26 is co-transfected together, and here TLR14 is co-transfected together. So oligomer can induce signaling via both TLR14 and TLR26. Now we are thinking this is probably the characteristics of the oligomer only, or like oligomer, that means like it is inducing the signaling via both TLR26 and TLR14. So then we have checked in case of VCC monomer in macrophage, and we observed that in case of macrophage, VCC monomer can induces its signaling via TLR4, TLR1, but both via TLR2 also. So that means in case of macrophage, both VCC monomer and oligomer is inducing its signaling via TLR26 and TLR14. And in case of VCC oligomer, it is inducing its signaling in macrophage both via TLR26. See, here in TLR2 knockout background, there is decrease in cytokine response, and in both TLR4 and TLR1 deficient background, it is also there is a decrease in response. So suggesting that in case of macrophages, both monomer and oligomer is inducing signaling via TLR26 and TLR14, but in case of dendritic cell, it is like both the preformed oligomer and the monomer is inducing signaling via TLR14. So in case of dendritic cell, the preferred heterodimer is TLR14. In case of macrophages, it both TLR14 and TLR26. So why it is happening? Why this discrepancy? Why this single ligand is getting recognized by different different receptors in two different cell types? So is it because of the basal level availability of the receptors? So what we observed that in case of this red is the dendritic cell and the black is the macrophage. So in case of this dendritic cell, the basal level availability of TLR1 is more compared to the macrophage. And in case of macrophage, the basal level availability of TLR2 is more compared to the dendritic cell. So TLR1 is more available in case of dendritic cell, TLR2 is more available in case of macrophage, TLR4 is more available in case of dendritic cell, and TLR6 is more available in case of macrophage. And then we have also checked like what is happening in case of ligand treatment, and we observed that TLR2 further is decreased, and TLR1 is further increased in disease following VCC treatment. So that is why probably in case of dendritic cell, VCC, both the form is recognized by TLR14 and inducing signal via it, and probably in case of macrophage, the basal level availability of the TLR26 also high. So that is why in macrophage, both TLR14 and TLR TLR26 are playing a major role in induction of inflammatory responses. Now, as I have shown that VCC plays a very important role in induction of the cell death. So now we want to know that why this TLR has any role in the cell death. So what we observed that yes, in case of, so this is a cytotoxicity assay, and in the TLR knockout background and TLR4 knockout background and mediated knockout background, the cell cytotoxicity is significantly less. Only in TLR2 background, the cell cytotoxicity there is no change in the cell cytotoxicity. So suggesting this TLR14 heterodimer not only is probably playing a very major role in inflammatory responses, but also probably could be involved in VCC-mediated cell death. Further, in case of BMDM, we observe that there is a decrease, though it's non-significant, but there is a decrease in cell viability in all the knockout mice. So, knockout cells. So then what we have done, we have injected the VCC intraperitoneally in wild type mice as well as this TLR knockout and mediated knockout mice, and checked for the survival and observed that in case of mediated knockout mice, the survival is much more. In case of this red is for TLR1 and green is for TLR4. So in case of TLR1 knockout mice and TLR4 knockout mice is also survival is much higher. And this magenta is TLR2 knockout. In case of TLR2 knockout, it is almost same as the wild type. So suggesting that this TLR14 and the mediated dependent pathway is very important and play a crucial role in mortality of mice. So then also we have looked at the serum cytokine response, and we observed that all the different TLRs, so far we mentioned TLR1, TLR2, TLR4, and all of them are play important role in the serum cytokine. So this is regarding the VCC. Now, what happens in case of V. cholerae? So, what we have used V. cholerae wild type and delta HLYA. This means this VCC deletion mutant. And and then we have actually this, and this is an enteric bacteria, so it has to be like colonized in the gut infection should be there, so we have fed them orally and looked at the colonization and also at the serum cytokine response. So after the colonization, you can see here this is the wild type and this is the HLYA, this is a deletion mutant, and both of them the colonization is same. So, however, when we looked at the serum cytokine response, so in case of delta HLYA, that is VCC deficient mutant, the serum cytokine response, inflammatory cytokine response is much less, suggesting that in case of Vibrio cholerae infection, VCC play a major role in inflammation. And further, we checked that in this serum cytokine response, how much contribution is for TLR14 and the mediated dependent pathway. And we observed that in case of mediated deficient, TLR1 deficient and TLR4 deficient mutant, the serum cytokine response is decreased. So suggesting that TLR14 mediated dependent pathway play a very major role in serum cytokine response upon V. cholerae treatment. Now, a study came up around 2023. So there they have shown that V. cholerae can make biofilm surrounding the immune cell. And during the biofilm, first there is an attachment of the bacteria, then the biofilm formation, and following the biofilm formation, there is actually killing of the cell, and that killing of the cell happens by secreting the VCC. And after some time, there is dispersal of the biofilm. So we have checked that in our case also, in the early time point, there is attachment of the bacteria, and then there is the formation of the biofilm, and then there is the dispersal. And there we have checked in case of bone marrow dendritic cell and also macrophages. So now we wanted to check that whether cell death happens upon this biofilm formation. And what we have done, we have co-cultured the immune cells with the V. cholerae and observed that in case of the V. cholerae, the cell death is much higher, but when there is VCC deletion, so the cell death is almost negligible. So this is the red is the PI and this green is the cell trace. So the dead cells will accumulate the PI in the nucleus. So the same happens in case of macrophages as well. Now, what is the role of this different TLR signaling in it? So we observed that in case of TLR1 and the mediated deficient, there is a decrease in cell death in bone marrow derived dendritic cells, but not much in case of TLR1 knockout in case of macrophages. However, in TLR2 and mediated, probably there is a decrease in the cell death. So this suggests that TLR14 play a very major role in cell cytotoxicity upon V. cholerae infection and pro-inflammatory responses upon V. cholerae infection. So with that, I like to end it. This is Aisar Mohali, and this is my group. And of course, Aisar Mohali and my granting agency, I like to thank, and thank you.
Thank you, Urmila, for your excellent talk. Very interesting work. So the lecture is open for discussion. Any question? Shovel.
Arunika, here, yeah,
Yeah,
very
yeah.
nice talk. Yeah,
Thank you.
Achha, I have a quick question. This is you have done. I think this initial part, the TLR1 and this hybrid TLRs or heteromers. Those were compared using BMDCs and BMDMs.
Right.
And given that we have those data available for mouse colon, intestinal dendritic cells and intestinal macrophages, and
Right.
or even human colon with human dendritic cells present in the colon versus human macrophages present in the colon where it matter. Would you mind to check, or have you checked if the distributions of those TLRs are similarly different in those settings? So as to lead to altered heterodimerization.
I have not checked like whether this TLR distribution is different, but what I can, so that is like kind of like our hypothesis, like why, or maybe like what you can say like we think could be the availability could be one of the reason why different cell type they are engaging the same ligand in different manner, right? But this TLR14, this novel assembly, which we are first time describing, so we have seen that in case of neutrophil also, like I have not presented that data, they also engage this TLR14 heterodimer for VCC for induction of the inflammatory responses. And as I have shown, in case of in vivo responses also, this TLR14 is playing a very important role even when we are feeding them orally. So that suggests could be the gut associated dendritic cells. They are also probably getting affected by the same way.
Any more question?
Fantastically kind of interesting talk, So several questions. For example,
this light is like, yeah.
Yeah,
Okay.
this is Dipavan.
Yeah, I can see.
So
Okay. I think like that's better.
So when you find one ligand which binds to one TLR heterodimer, just differential availability may not affect this kind of phenotype, right? So that's kind of counterintuitive. For example, if that's the case, then definitely you will get the overlapping data as you have got. But in the knockout, when TLR2 is not available, they will bind to TLR14 because they are available, right? And because then that differential availability situation is not there, right? That's one confusion I have. And second thing is, if there is a TLR heterodimer, which is not really reported widely, now is found to bind this bacterial product, bacterial protein, must be other ligands also. There must be other ligands for which this is happening, because
so far it is not reported. That doesn't mean this is the only exclusive ligand which binds to it. It cannot be. So it is like first time we are reporting that that can also happen. And another thing what I am trying to say here, so it doesn't mean that because the basal level availability of TLR14 is there and any ligand can induce this TLR14. That must be also ligand dependent. So not all the ligands. So then in case of dendritic cell, for all the ligand we will see this, right? So it has the, what I feel that VCC is recognized by TLR1, and then this is bringing the TLR1 to the TLR4, something like that. Like ligand binding is happening via TLR1, signaling mediation is happening via TLR4. But the interesting part here is that upon ligand activation, so this TLR dynamic, you mentioned about this is a dynamic thing, right? Like it's not key always like surface TLRs will remain same. Ligand also directs like what ligand, what receptor will go inside and what will come out. So in case of ligand, upon ligand activation also we have seen that TLR1 level increases and TLR4 level increases in dendritic cell.
But if you get rid of
and TLR2 level gets decreased.
No, if you get rid of TLR1, say, or TLR4 in dendritic cells.
In case of VCC, we have seen that it does not induce pro-inflammatory signaling or cell death.
But then why TLR2 and TLR6 cannot perform that?
TLR26, their expression level is less in case of...
but still it's there.
It is there. Expression level is low, and upon VCC activation, their expression is becoming almost zero.
So there must be some other regulatory mechanisms also, right? Because otherwise it cannot be explained.
Yeah.
Thanks. Thank you so much.
Maybe I have missed point in continuation of what Gopal was asking. Just a simple query that as the TLR2 is lower in this DC.
In dendritic cells compared to the macrophages.
Have you ever tested by increasing TLR2 in DC and see whether it will then form the TLR1 and 2, not TLR1 and 4? Introducing.
So by using the HEC293 heterologous system, we saw that it can induce signaling via both 2,6 and 1,4. Okay, but propensity towards 1,4 is much higher. Like the liking, like for induction of signaling for TLR14 is much higher for particularly for this ligand than TLR26.
Runika, VCC is a Vibrio cholerae toxin.
Like cytotoxin, cytolysin. Okay. It's extracellular, like it is secretory toxin.
Secreted. So basically, we call it cholera toxin.
No, it's not cholera toxin.
So it's exotoxin.
It is like cholera toxin. Cholera toxin is also an exotoxin.
Okay.
This is also an exotoxin, but what it does, it forms spore, and also it induces death signaling via different manner and kills the cell.
So like cholera toxin, whether this exotoxin has affinity for rafts.
Rafts. We are doing some work on
because
that
your
aspect.
HEC2 cell, they are devoid of
rafts.
TLRs,
right.
Then does it interact with or it transduces signal in HEC2 cells?
Without the TLR transfection, it does not induce anything.
By itself, it does not.
By itself, it does not.
Okay.
Only like when we are transfecting with the TLR, and we can look at individual TLR signaling, how it happens, or how the interaction of the TLR can happen. Only then, like.
Okay. Thank you.
It kept my mind that please started by telling that the surface, it resides in the surface, the TLR. So
TLR resides on the surface also, and it also remains inside in the endoplasm. So different TLRs.
The cells where the surface receptors are not, TLRs are not present, is there any possibility that the intracellular receptors are present there?
In HEC293, there could be intracellular, can be, but the surface receptors are not there.
And if there is no more questions, again thank Veronica.
So that is if we talk about intracellular receptors, these are not TLRs. TLRs are generally, but those other receptors like Regai-like receptors and all those can be present, which are present almost in all lymphocytes. Okay. Okay.
One thing is that you are treating the HEC cells with the VCC, right?
What we are doing, like on HEC cells, there is no expression of this receptors on the surface, right?
That's a TLR.
So that for the TLRs. Right. So we are transferring cells. Now these cells are expressing the TLRs.
Yeah.
So
you
once these are
TLR constructs,
TLR
1 and
constructs,
4 or 2 and 6.
Either individually or
right.
together.
Right.
We are overexpressing
But
it.
you are adding the VCC.
Then we are adding the VCC.
So the question that Shamulda asked is whether the VCC can interact with membrane receptors other than the TLRs. So that is not clear from that experimental data. It might still interact with
so what I can say, like it can interact with other receptors, but it is not activating the NF-kappa B. Maybe I should say in that manner, like for NF-kappa B signaling. Thank you.
So the last speaker of this session, Imroz Khan from Ashoka University. So he's an evolutionary biologist with special focus on understanding the selective forces that determines adaptive evolution of the immune system. So his research combines diverse approaches, use of experimental evolution and life history analysis. So he did his bachelor's in chemistry from St. Xavier's College, Calcutta, master's in biochemistry from University of Calcutta. He did his doctoral studies in evolutionary biology at Indian Institute of Science Education and Research, Kolkata. Before joining Ashoka, Imroz was a postdoctoral fellow at the National Center for Biological Sciences, Bangalore, and a visiting researcher at the Free University in Berlin. So Imroz, your floor is yours.
Thank you. Okay, so okay, this doesn't work. All right, thank you so much. And just wanted to express my heartfelt gratitude towards organizers. So my name is Imroz and I'm a evolutionary biologist and who happens to be having some sort of Miltonic affair with immunology. I'm not an immunologist at all, right? It's all about implications. What I try to do is to understand the evolutionary basis of host-pathogen interactions. And in a stark contrast to most of the talks, well, almost all the talks that's happening since the morning, right? I don't work with molecules. Rather, my scale is slightly different, although intimately linked to what people are talking about, because if you think about evolution, because evolution doesn't see molecules. It looks at phenotype, it looks at organisms, it looks at populations, right? And then molecules, they try to reorganize, reprogram themselves to cater to the organismal need. So my question, my science starts from there. And today I'm going to talk about an aspect of host-pathogen interactions that almost all of you be aware of is called co-infections. So, and I don't think I need to really talk a lot about what it means to, why should we look at co-infections in biology? Because if you look at the ecological world, it's really difficult to find a situation where organism is infected with one pathogen, right? It might start with one, but eventually it might be hijacked with many others. So eventually host has to grapple with multiple pathogens at once, right? So our immune responses has this immense task of keep on deciding that what to use, when to use is all about the timing, right? But then the question is, if you think that our immune system, how it has been shaped by co-infecting pathogens over the years of evolution, because that is something that we're not aware of. Historically, most studies in immunology has been focusing at one pathogen, one host system, right? And we do have a lot of information already. Last 100 years has seen an extraordinary advancement in the field of immunology in terms of discovering cells, molecules, signals, but we don't yet understand how those molecule cells have come all this far. Why do we see our immune responses the way they are? So that's a big question that we all kind of grapple with. So that's my, one of the interests that we follow in our lab. And again, I kind of summarized, co-infections are widespread in nature. If you think about disease burden in the world, right, most of diseases might have been caused by multiple pathogens, bothering an organism. In humans, for example, over 1/6 of global populations is affected by co-infections, and examples are many. Right, some of the landmark diseases like HIV and all, they're also known to be getting complicated by introduction of several other infections while the patient is grappling with HIV. And then, as I said, that an overwhelming number of studies all these years have been looking at one host and one pathogen system, but we don't yet understand that how our immune response has been influenced over an evolutionary time scale by multiple pathogens that we all grapple with. So that is one thing. One of my students, Trijon, was kind of getting interested into and he asked this very, very fundamental questions. Well, it cannot be more fundamental than this. Like, how might mixed infections influence the evolution and development of the immune system? So that's Trijon. He was my second PhD student, and now he is a postdoc at Utah. And this is his PhD work that I'm going to summarize today. All right. So if you think about what are the possible trajectories for evolving immunity against multiple pathogens, how do you see hosts are grappling with multiple pathogens? So one possibility is that if you have different microorganisms affecting you, and you need to have different immune responses, has to activate against all these pathogens at once, it just puts in the risk of increasing the cost of activating multiple arms of immunity at once. So that could be inherently heavily costly, right? In the morning, we have been talking about how immune responses that serves us can also be super detrimental. Now, activating too much of immune responses against multiple pathogens could be really, really problematic, right? And one could understand if you are grappling with multiple infections, your ability to evolve immunity against multiple pathogens could be slower or maybe less effective. Second possibility could be determined by the intra-host, within host interactions between microbes, right? There could be interference between different pathogens. And if one pathogen kind of eradicates another one, then you have potentially running into the problem of handling only one. So your immunity might evolve as per maybe one pathogen's pressure. This is a possibility too. But the third possibility is possibly a bit more complicated, a bit more nuanced. Now, most often the pathogen that infects us, they might have very, very different, within host growth and clearance dynamics, right? They grow at a different rate, they get cleared out, or they kind of behave within a host in very different ways. What if two pathogens that are infecting us, they're growing in a very different growth kinetics. They're following very different growth kinetics. So let's say the two pathogens, right? I hope that some of you from the back can look at this bugs here, and then think about the temporal dynamics of these pathogens within the host. And one pathogen suddenly grows too fast compared to another one. So your body now, well, intuitively, it's very intuitive. There's nothing, very deep science in it. If you are suddenly seeing a burst of one kind of pathogen, your body's immune response might want to grapple the fast-growing pathogen one, right? So your immune response is likely to tuned as per the temporal dynamics of within host pathogen dynamic, the temporal dynamics of within host growth. So likely that immune response will target these pathogens, and this puts us in a situation like this, where your immune response, well, the pathogen growth can show such kind of temporal dynamics, and immunity might want to attack this bug first, with respect to the one that's going to cause later infection. And if you think about the existing models of theory, it shows that is the initial stochastic variation of pathogen burden is what determines how our immune response is going to act, and also the survival success of the host. If the host can tackle the stochastic increase of one kind of pathogen of another one at the initial phase of infection, right, it might survive. If it cannot, it might die. I'll show you how. Right? So, but you see that when we talk about multiple infections, we are dealing with a very, very complex situation, because no two pathogens could be same. If I want to use one model, one pathogen, or maybe two pathogens, all the pathogens will be very different, even within the same bacteria, even within multiple strains of the same bacteria could be very different. So it's important that we began this problem with building a theoretical model first before we taking a stab at an experimental level. So this is where Dipendra joined my lab as a Simon's Ashoka Fellow, and he wanted to tackle this problem. So we wanted to model growth dynamics of different pathogens first. For instance, look at this right panel. You can think of 4 different kinds of pathogen, broadly classified into a pathogen which could grow rapidly towards causing acute infections. You see this gray dots, which denotes the pathogen load of the individuals which dies, right? And then you have some individuals who could clear the infections to a level that the infections, where the pathogen burden is no longer problematic for them. So you can broadly say that gray dots are the dead ones, and the blue dots are the surviving ones. And then you can keep on forming a few more classifications like the other one. Let's say one pathogen can grow slowly and then until it reaches acute infections, and then it can also clear the infections. And then there could be third and fourth category where they can either grow rapidly, towards an acute infection phase, but then, and this gray dots representing the individuals those died, and then some individuals, they don't clear the infection completely, but in those individuals, infection gets into a chronic infection phase. The pathogen load gets into a, latent or, this pathogen burden, they remains constant within the individual, but they're no longer problematic for the host. So they kind of survive with this chronic infection load. So these are the 4 different kind of pathogen categories that you could come up with in the model. And then you can mix these individuals, you can mix this, we can pair this pathogen based on contrasting growth and clearance dynamics. And this is what we did, because as I said, that it's entirely possible that 2 pathogens that are infecting our host could have, most likely to have a different growth dynamics in the body, right? So, this is a limited view, of course, but then you can think of many situations which kind of adhere to this kind of stuff. And when we model the host, post-infection survivorship against such pathogens, we largely, by and large, what we see that it is the rapid-acting pathogens, the pathogens which grows very fast. They're the one which drives the fate of co-infection. So here, the blue lines that you see, this is a simulated survival curve against rapid-acting pathogens, and the red lines are the ones which are, infected with slow-acting pathogens. And you see the orange-colored line; these are the co-infecting, individuals which were infected with multiple pathogens, and their survival curves roughly mimics the survival pattern of individuals who are infected with rapid-acting pathogens. So this is what we could summarize. That this is this kind of summarizes fair number of possibilities. And then subsequently, what we asked is more evolutionary, right? What if a population keeps on happening, keeps on being exposed to such pathogens over and over generations' time? So, and then how do you see, right, pathogens adapting against such pathogens? So we want to simulate that situation as well. Again, I'm not getting into mathematical details. If some of you are interested, I'm happy to explain later on after the talk, right? X-axis gives you the proportion survival, right? If you infect a bunch of individuals, what proportion of them survive? This is what this y-axis denotes. And in the x-axis, you have the generations, And very likely, when you are infected with a pathogen, your survival success is very low at the beginning, but eventually, as evolution happens, they keep on gaining the survival success. Now look at what's happening, right? These blue lines are the individuals, the populations which have been infected with co-infected pathogens for many generations. And again, if you see their adaptive trajectory, right, which again kind of reflects the fast-acting pathogens. R means stands for rapid acting, right? And in all the cases, the survival success of the host populations against the co-infected pathogens is determined by the dynamics against the rapid-acting pathogen. So this is what we could start off. And this is, again, as I said, these are very complex situations, and there could be n number of situations, but this is what we can kind of roughly generalize what's happening. Okay, so the next big question that we were grappling with, well, again, one of the predominant assumptions that we had at this stage, right, what exactly the evolution is working. Now, if you think about rapid-acting pathogens, right, if it's growing very fast in a host body, and if it is killing very fast, it is entirely possible that host doesn't have enough time to even induce a suitable immune responses. So it's again the speed of infection which literally outperforms your ability to induce an immune response. And this is what is, what we think is happening, that if you are, and in all these cases, right, you see that our pathogens are always evolving at a slower rate, right? And again, this is one possibility could be that, their ability to modulate their immunity against those pathogens is relatively less effective. So this is the assumption that we began with. And and this is what Dipendra came up with. And subsequently, Srijan chipped in, and then he took a momentous task of finding for mathematics. And this is where we took resort to the tools. The major tool that we have in our lab is not about instrumental techniques that we really are. This is a brute, kind of labor we try to handle with populations comprising of hundreds of individuals, and which we maintain for generations and for generations, right? These are not one or two individuals comprise the population. These are hundreds of individuals in each population, and they're being exposed to infection for generations. And this is what he started doing. So we use this model system called Tribolium castaneum. These are flower beetles. You must have seen these beetles, in your atta maida, whatever you keep in a stored food, these beetles will pop up, right? And these are excellent model system. Right after Drosophila, this is the most well documented model system for insects. And we chose two pathogens, again, I'll come to the reason again. So these are the two insect pathogens that we use. These are natural pathogens found in these beetle populations. One is Bacillus thuringiensis. This is a positive bacteria. And again, this is a Pseudomonas entomophila. Another pathogen that is again known to infect insects. But those two, they differ entirely how they grow inside the host. This is precisely mimicking what we found in the model. We wanted to select pathogens which will have very, very contrasting growth and clearance, clearance pattern within a host. So Bacillus thuringiensis, if you see, BT, I'll call this one as BT and the Pseudomonas as PE. If you think about BT, they grow really fast in a host, right? Within 6 to 7 years, you'll see that number of cells can amplify to maybe 100 to 1,000. That fast, but entomophila took some time, right? It takes around 20 or 24 hours, after infection, so that it can reach to a level where it can cause an acute infection. And you can see a precise reflection in there how they cause the mortality in the host. If you beetle populations with a BT infection, you see there's a sharp decline in the mortality, sharp decline in their survival within maybe 6 to 10 hours. Whereas the red lines, which represents beetle populations infected with entomophila, they die in a continuous speed. It's not there's no dramatic decline in survival, but it happens over a prolonged period of time, right? And then we had the third infection conditions, which is a co-infection, where we mixed BT and P in a 1:1 combinations, and you see their survival pattern roughly comes intermediate to both BT and P, but yet what is really remarkable to see that their survival pattern at the initial scale is very, very similar to how BT drives host mortality patterns. And this is the within host growth dynamics of the pathogens. If you look at the Bacillus thuringiensis, it grows rapidly at the beginning, and then suddenly after a while this is a phase where most of the mortality happens. And after that, host whoever survives, they can clear the infection pretty efficiently. You see that infection load literally touches down to 0. It's a complete clearance. Whereas in contrast, the Entomophila, it grows to some extent till 24 hours, 30 hours, and then eventually you see the host starts clearing the bacteria to some level, and then it stabilizes, right? And this kind of load you can find even after maybe 15, 16 days. So bacterial load stays. So inherently, these two bacteria differs quite dramatically, both in terms of growth dynamics as well as how they get cleared or how they handle within a host, right? And then what we could now keep on hypothesizing, right? If you are infected with a pathogen which grows so fast, your body needs to induce fast-acting immunity. Because if you don't have the fast-acting immunity, you cannot tackle fast-growing pathogens. But in contrast, if you think about those persistent infections, you have to have a long-term surveillance being continuously being active in a host, right? So in all probability, if host is trying to control both the situations at once, it has to invest on fast-acting immunity, while it also has to invest on immune surveillance going on long term. And this can collectively increase the cost of immune responses pretty much high, right? And if this is so, one can argue, because now your investment is so high, you cannot possibly evolve as effectively as you could do against those single pathogens, right? So possibility one is that immunity might evolve much more at a slower rate against mixed infections. Second possibility, again, coming down to the difference in the growth and clearance dynamics, right? If they're growing and killing at a different rate, hosts might invest in temporarily segregated immune strategies. And in all possibility, this possibly be a more optimal strategy for hosts. In that sense, it doesn't have to activate all the immune responses together. So it can, go as it happens, right? And again, we come back to the survival curve that I was showing, and if you see that the mortality pattern within 6 to 8 hours, right? If you collect the beetles which died at this early hour, and 2 minutes, okay, yeah, I just maybe in 2 minutes I'll just summarize, right? If you look at those jet individuals and extract the bacterial cells, you'll see that most of this beetle death has happened because of Bacillus thuringiensis. This is a blue dot, are the BT cells, right? And so it's entirely possible that the host has to evolve an effective immunity against such mixed infection case. It might want to tackle the BT cells first. So, you automatically, your immune response can be segregated. And that's what we assume could happen. So this is where we wanted to find an empirical evidence. As I said, we created this different beetle populations infected with this different pathogens, both with BT and P and a mixed infection case. And we literally simulated a natural selection in the lab. That means you take up a number of populations, you unleash infections to this beetle host, and whoever survives, you take those individuals to constitute your next generation. This is how natural selection progresses, right? And this is what we call experimental evolution. And look at the result that we find. Maybe I'll end here in the interest of time. So as expected, you see that these red dots, this is again the percent survival, and x-axis gives you the number of generations. And with generation, you see that beetle populations which infected with Pseudomonas entomophila, they could increase the survival success, pretty rapidly, compared to the other two. Whereas the blue lines depict the populations which were infected with Bacillus thuringiensis. You see their evolution is quite slow, and mixed populations which were evolving against the mixed infections, they were mimicking how they have been also might have done in case of single infections. So this is what we predicted from our model as well, right? You see the mixed infection was predicted to mimic the rapid acting pathogens, and this is where we can see this is a very tight overlap between the empirical, the theoretical predictions and our empirical data. And next thing that we just briefly wanted to check whether this again is a case for limited scope for immunomodulation against a fast-acting pathogens, right? Or conversely, the reason why these beetles which were infected against entomophila, which is slow-acting pathogens, could they afford to modulate more kind of immune responses? That's why they're more effective. And this is where we took a lot of multi-omics platform, and I'll just briefly show you one result. Right, if you, so this is where we are, in looking at a metagen expression profile of different immune categories. Here we are not looking at the genes because individual genes are always problematic. So we divided our immune, all the immune molecules that we receive from our RNA-seq analysis, right? We divided them broadly into the different categories. For example, the group of molecules which are like antimicrobial peptides or induced immune vectors. We also looked at molecules which are regulators or molecules which part in maybe this bunch of molecules are the molecules involved in looking at this is an inducible molecule. This is a regulators. These are two different categories. Various other molecules. And if you look at these panels, I'm not getting into this anymore. So y-axis gives you again metagen expression profile, and x-axis gives you the phenotypic, the cumulative phenotypic score. And we wanted to find a correlations. And you see that in there are at least 3 different categories of immune molecules are involved. So expression profile changes in receptor, regulator, and inducible effectors are explaining the phenotypic variations. And in contrast, if you look at Bacillus thuringiensis, there's only one category of molecule explaining. So it just shows that overall availability of immune molecules to this fast-acting populations evolving as fast-acting pathogens are really limited, and populations who evolve against a slow-acting pathogen, they had a much more wider scope of the selection to act upon their immune responses. So in summarize, what we found is that the fast-acting pathogens, they can limit the adaptive success during co-infections. We found a mathematical prediction that co-infection will closely make pathogens. We found an empirical support, and when we looked at the different immune categories, if you look at the immune signaling cascade and divide them into these broad categories, we find that at least 3 different categories is where the selection can work in order to improve the survival against entomophila, this dark blue thing. Whereas in contrast, BT is more limited. And mind that this is an arm of immunity which is really cytotoxic. So if you change these molecules over the due course of evolution to increase your effectiveness, you might as well pay a high degree of immunopathological cost. So even though this might explain the phenotypic variations, but this arm of immune molecules are dangerous to use, right? Maybe I'll stop here, and I'll quickly acknowledge my group. It's a growing group, and of course, I want to thank both Abhirup and Gautam who has been helping us. This is also first time we were trying to do some sort of modeling, and Gautam has been very instrumental in terms of giving important suggestions. Lastly, all my funding sources, SERB, Ashoka, Alliance, and Axis Bank, who have been generously supporting such fundamental work without asking me to do more translation stuff. So I'm thankful to them. And with this, thank you. Okay.
Thank you. Thank you, Imroz. Some pressing question? Oh, okay, Avik.
Imroz, a thought-provoking presentation. So I have a very simple question. So you talk about single infection and the mixed infection, but have you considered the subsequent infection, right?
As I said, this is an incredibly difficult empirical paradigm to do, yes, but that's something, as I said, there's innumerable number of situations. But what I can make a quick intuitive stab at your question. I feel it still might adhere to the mathematical prediction that I had, because again, it gives you a scope of who comes first, right? Which pathogen is going to challenge the host immunity first. So it might drive the immunity substantially in favor of the first pathogen, whereas the second pathogen might have residual effects, and also it might have its own immune response as well. We don't know, actually.
No, I completely agree. But the example you gave probably in your second slide about the HIV, even for the HIV, during the HIV infection, no co-infection.
Exactly. Absolutely.
It happened much later on.
Right? Absolutely. I completely agree with you. But this again, maybe I would say that this is just one instance out of maybe millions such examples.
Imroz, fantastic thought-provoking work. Now, at least, I know your experiments are really difficult, right? Transgenerational tracking. But at least on mathematical model level, what if you take the populations which were which evolved in response to a fast-acting pathogen, and now after 30 generations or after 60 generations, or go other way around. Okay, so someone which has, I mean, one population which has evolved in response to a slow
Cross immunity
pathogen,
win.
after 30 generations, they encounter a first pathogen. I mean, what will be the prediction? I mean, maybe you can give it a try, because that will be something which will be really closely related to pandemic preparedness, you know. So now, if you, I mean, we are all kind of surviving because of all these slow pathogens. Most of the pathogens we encounter are kind of slow pathogens because now if now some zoonotic disease comes up, which is really a fast growing pathogen, what will happen?
I think that's a very, very topical question. And this is where we were, one of the students in the lab, she's trying to track down cross reactivity against, multiple pathogens. And she's clubbing pathogens based on their phylogenetic relatedness, right? Strains, which are most closely related. Then you take the larger classes, right? The gram-negative, gram-positive, and again, the kind of pathogens which are likely to induce similar kind of immune responses versus dissimilar kind of immune responses. And I'm yet to analyze the data, but this is the question that is definitely, that we are looking at.
So thank you so very much for your such an illuminating talk. And we are now at the end of this session. So all three talks were really, really fascinating, wonderful, excellent. What else? I don't know what can be said. All thought-provoking. And again, for me and also from on Shyamulda's behalf, we thank the organizers, especially Professor Chattopadhyay, for inviting us to chair this wonderful session. Thank you all. Enjoy.
Hello. So after this wonderful session, we will proceed towards the felicitation of our chairs and invited speakers. May I request on stage Dr. Arunika Mukhopadhyay and Dr. Imroz Khan, and I request our chairs, Dr. Shamul Roy and Dr. Tanya Das, to kindly felicitate our speakers. And also I request Dr. Shomen Boshak, our keynote speaker, to be on stage for the felicitation. So sorry. I now request on stage our co-conveners of this conference, Dr. Momita Das and Dr. Anindita Banerjee, to kindly felicitate our chairs for this session. I request all the delegates to come up on stage for a group picture. Now we break for a short tea break and quickly come back for the next session.
non-killing effect as well. So we have been studying, or we started studying some of those responses like programmed cell death mechanism and inflammatory cell death response, and as well as inflammatory response generation. And in previous talk, one of the previous talk, Arunika mentioned mostly about inflammation mechanism, how one of the pore-forming toxin VCC can trigger that kind of inflammatory response that Arunika has discussed. Now, in my talk, I will focus on another pore-forming toxin, bit atypical pore-forming toxin, thermostable direct hemolysin, produced by Vibrio parahemolyticus, and it's a more recent study, and some of it is like ongoing study I'm going to talk about. And what I'll be speaking today is mostly the collaborative work which have been done in collaboration with my long-term collaborator, both from professional and personal point of view, Arunika's lab. And I'll mostly talk about these two aspects of TDH functionality, thermostable direct hemolysin functionality, that is programmed cell death induction and immunomodulatory response generation by this toxin. I will not show much raw data. I will show some data, and I'll basically give a brief overview of the idea or hypothesis that we have, and I'll try to keep more time for discussion or questions if you have. So, as I mentioned, that thermostable direct hemolysin is produced by Vibrio parahemolyticus. It is basically a seafood-borne pathogen, and it causes acute gastroenteritis in human, and it can even lead to septicemia. Now, thermostable direct hemolysin, TDH, it is one of the major virulence factor of this pathogen, and it is a pore-forming toxin by nature. It damages cell membrane, eukaryotic target cell membrane, by punching hole. That is basically its biochemical description, the way it can kill a target cell. Now, from pathophysiological point of view, TDH can evoke various distinct responses, which came out from mostly in vitro studies and some ex vivo studies as well. For example, it leads to hemolysis, that is lysis of erythrocytes. It can lead to cardiotoxicity, particularly if mouse injected with TDH, it can have cardiac shock. Also, it has been demonstrated that TDH generates massive enterotoxicity in animal model. So based on these observations, TDH has been described as one of the major virulence factor of Vibrio parahemolyticus. Now, how can it kill target cell? Obvious explanation can be membrane damage due to TDH pore formation, which will make the semi-permeability barrier function of cell membrane abortive. As a result, target cell can die. However, in recent time, it has been shown in some other studies by other groups that TDH can generate more programmed cell death response. Also, TDH can elicit inflammatory immunomodulatory responses. But until recently, we did not have any understanding how these responses can be generated and what are the mechanistic basis, what are the signaling mechanisms. So in our study, we have tried to explore some of these issues. So in the first part of my talk, rest of the part of my talk, I'll talk about the mechanism of that programmed cell death, and we have discovered some of the unique features that TDH-mediated programmed cell death is having with them. So, for these, we mostly utilized epithelial cells to understand cell death mechanism of TDH-induced cytotoxicity. What we found, and it is consistent with our earlier observations, that TDH can trigger very potent cell death or cytotoxicity in the target cells. And these cytotoxic responses are associated with some of the hallmark features of programmed apoptosis-like cell death, such as membrane phosphatidylserine flipping, DNA aggregation, etc. So there is nothing exciting about it. Another toxin causing apoptosis-like feature, and these are classical signatures. In the same direction, we found that during TDH intoxication, there is caspase activation.
Now, caspases are another hallmark features of apoptosis. So we can see that TDH intoxicated cells, when we probed them, we found many of the initiator and executioner caspases are getting activated. Their proteolytic maturation is happening. Again, nothing exciting. Now, the exciting point came when we found that when we treated these TDH intoxicated cells, prior treatment with caspase inhibitor still led to cell death induction. That means even under the condition of caspase inhibition, TDH could trigger cell death. This phosphatidylserine exposure was happening. So this was some kind of an intriguing observation that we are seeing caspase activation, but at the same time, if we block caspase activity, still TDH is triggering cytotoxicity. So we searched literature, and then we got some insight that just caspase activation is not sufficient. That means protein processing of caspase is not enough for their participation in the apoptosis. Because even after caspase cleavage, they remain associated with some of the class of inhibitory proteins that are termed as X-linked inhibitor of apoptosis. Now, during actual apoptosis, another class of protein, that is SMAC diabolo, they get released from mitochondria, then they interact with XIAP, and then they release XIAP from caspases, and active mature caspases get relieved, and then they participate in the apoptosis process. And in our study, we found, I'm cutting long story short, we found that during TDH intoxication, this SMAC diabolo release from mitochondria is getting compromised. As a result, XIAP is still remaining attached to active caspases or politically cleaved caspases, and that is why caspases are not participating in the actual apoptosis response. So despite caspase activation, we found caspase-independent cell death. Now, how it is happening? We found that mitochondria played a very important central role in TDH-induced cytotoxicity. TDH triggered mitochondrial damage, which resulted into release of various mitochondrial factors like cytochrome C, AIF, that is apoptosis-inducing factor, and endonuclease G. And upon mitochondrial damage, these AIF and endonuclease G, they get released from mitochondria, and then they translocate to nucleus, where they trigger and execute DNA fragmentation activity. Endonuclease G participate in that DNA damage and DNA fragmentation, which results into large DNA fragment formation. And it is commonly known that AIF endonuclease G activation and their nuclear translocation plays very prominent role in caspase-independent cell death. So that means, in the context of caspase-independent cell death, mitochondrial damage and AIF endonuclease G is doing caspase-independent apoptosis-like programmed cell death. Then we've had another very interesting observation. So TDH, as I mentioned, it is a classical example of pore-forming toxin. And until recently, it used to be known that as a pore-forming toxin, journey of a pore-forming toxin should end at the cell surface. Bacteria will release them, it will attack plasma membrane, and its journey will stop because it will damage cell membrane, and done. But we found in our study that TDH treatment, after TDH treatment of the target cell, TDH can actually translocate itself to the target cell mitochondria. And this process, we don't know the exact mechanism how it gets targeted to target cell mitochondria, but we find that lysosome is possibly involved in this mitochondrial translocation of TDH, because TDH intoxication also results into lysosome damage. And if we block this lysosome damage of the target cell by baphyllomycin treatment, which is an inhibitor of or blocker of lysosome function, then mitochondrial targeting of TDH is also getting perturbed. That means that lysosomal functioning is possibly involved in mitochondrial targeting of TDH. So take home message here is TDH not only damages target cell mitochondria, but it also goes to target cell mitochondria, which possibly involves participation of lysosomal function, but mechanistic details we still do not know at present. Now, another aspect of TDH cytotoxicity is involvement of PARP1. And our data shows that PARP1 activation also contributes to this caspase-independent cell death. Now, PARP1 is basically a nuclear protein involved in DNA repair pathway, and it can mediate cell death by two different mechanisms. One is caspase-dependent classical apoptosis, but it can also participate another caspase-independent cell death mechanism, which is a recently demonstrated mechanism and given terminology as parthanatos. So what happens during that process, that AIF gets released from mitochondria during caspase-independent cell death. AIF goes to nucleus, causes DNA damage, which triggers PARP1 activation, and that PARP1 generates more PAR molecule, which then goes to mitochondria, activates AIF more, and amplifies AIF production and more mitochondrial damage and more caspase-independent cell death. And indeed, in our study, we found that TDH intoxication also activates PARP1 activation. 2 minutes. Yeah, I'm almost done. So that means altogether, we come up with this mechanism that mitochondria plays a very important role during TDH intoxication, and it is doing a caspase-independent apoptosis-like cell death mechanism, but which is not a classical apoptotic pathway. Now, the another aspect we discovered during TDH intoxication is that in the context of innate immune cells like macrophages, TDH treatment leads to not only cell death, but also triggers production of various pro-inflammatory mediators like IL-6, IL-1 beta, ROS, and nitric oxide production. And using various macrophage cell types, macrophage cells, different TLR transgenic mice, as well as using in vivo model, we found that this TDH-induced pro-inflammatory response generation is specifically TLR2 and TLR4 dependent, but independent of TLR1 signal. Now, whether TDH engages combination of TLR2 and 4 or it works independently of TLR2 and 4 pathway, that we don't know, but we know for sure that these two processes are happening there. So that means TDH triggers on one hand inflammatory response, on the other hand it triggers cell death mechanism. So how the balance between these two responses are there, we still don't know. So I'm posing questions now which we don't have the answer yet. We are trying to answer those questions, and how the balance between two such responses are contributing to TDH TDH pathophysiology during Vibrio parahemolyticus infection, that aspect still remains unknown. Now, we have a very recent observation. Again, we don't have the mechanistic understanding about it. If we do peritoneal injection of TDH mice in a very sublethal concentration, based on earlier studies, even under those sublethal concentration, which people used to believe that that can be a sublethal concentration, but which we found that that kind of TDH treatment results into multi-organ failure in the mice. We see hemolytic blood serum. There is a hemolysis in blood serum. It produces blood urine in the mice. It causes intestine, kidney, spleen, heart, and liver. So it's not. So basically what we're trying to say here, that TDH treatment, its inflammatory response cell death mechanism is not only just limited to certain cell types, but it is causing a multi-organ kind of pathophysiological defect in mice, and thus it can possibly contribute to Vibrio parahemolyticus pathogenesis process. So with this, I would like to conclude my talk, and before I finish, my sincere thanks to all my past and present lab members, my collaborators, Arunika, Somnath, with whom we do a lot of cryo-EM based studies, Shashi, we collaborate for bioinformatics studies, and thanks for all my granting agencies, and thank you all for your patience. Thank you so much.
Hey, Kaushik, very nice talk.
Thank you.
Just a couple of areas. It has been shown earlier, though, that if you inhibit caspase-dependent apoptotic pathway, cell would switch to MLKL-dependent necroptosis.
Necroptosis,
And if you
yes.
block both, then you might see that, that does not say that cell was not using caspase for apoptosis. It says that in presence of inhibitor, it switches to alternate sulfate. In that area, I find one confusion. It's a fantastic work, but that area I think needs to be a bit consolidated. We even tried both the inhibitors of MLKL and caspase to as certain things.
Yes.
And I will come to that. This other part is that inflammasome. These porins are likely to cause iron imbalance in the cytoplasm. And typically they would trigger inflammasome activation. And that ties up with what you see in vivo with cell death and inflammation together. It's a telltale signature of inflammasome activation. So any thought about it? So
Right.
The first one is...
Yes. So the first question is, in absence of caspase participation, whether other kind of things are happening. Now, MLKL mediated pathway, Gasdarmin mediated pathway, we can expect that, I'm not saying, we are not ruling out those possibilities, but cell death mechanism we studied mostly in the epithelial cells. So I doubt whether MLKL or pyroptosis, necroptosis kind of phenomena will be happening in non-immune cell types or epithelial cell types at all or not. It happens in fibroblasts, but epithelial cells, we have to see whether those are operational or not. In fact, we have Gasdarmin knockout mice. MLKL we don't have. We have some studies in pipeline. So that is, of course, we'll be venturing into that one. So we don't have the answer yet. That's a possibility exists. Good for us. Another student can work on that direction. Now, your other question was inflammasome. Yes, absolutely. Inflammasome could very well be operational, but again, and we believe in the context of macrophage, for example, without even doing those experiments, I'm sure inflammasome will come into the picture. Now, the important aspect about the work that we are we have done, these are the concentrations where we are using very sublytic dose of concentration of the protein, which earlier people used to use much higher concentration, which will really form pore and iron influx, because ultimately when pore formation happens, then of course iron imbalance will happen inside the cell, and that can evoke any kind of cell signaling pathways. But the protein concentrations we use in our study, for these particular studies, we keep protein concentration at such a low dose, pore formation would possibly be restricted. That is why we try to work in a sublytic concentration at which erythrocyte lysis and pore formation would not possibly happen. But again, all possibilities exist.
Kaushik, excellent talk. So one thing is I'm wondering, because you are telling that your mitochondria is affected. Have you seen that mitophagy is happening or not?
No, we did not
because
check.
the lysosome, you are telling that lysosome is involved here. So mitophagy during that lysosome is also involved in. Other course of autophagic pathway maybe involved.
Yeah. Autophagy, we are working on this. I mean, we have started some work in that direction. Mitophagy, we have not explored yet. In that connection, I'll just mention one point, very interesting aspect we found, that here we found that we have intact cell, we add TDH, mitochondria gets damaged, and cell death pathways are triggered. Now, if we isolate mitochondria from the target cell without TDH treatment, we take pure mitochondria, then add TDH, TDH can still bind to that isolated mitochondria, but then it cannot damage mitochondria. That means many other factors. In fact, I did not show the data. We have shown in our study, the published work, that actually BAX, BCL balance is also important and required for TDH recruitment and functionality. So alone, TDH can bind to isolated mitochondria, cannot damage mitochondria. BACs must be participating in the process.
So have you checked over time, 2 hours, 6
Yes,
hours?
yes, yes.
And,
We have monitored till 6-hour time point. We have seen, isolated mitochondria, right? Yeah, we have not seen any kind of mitochondrial damage in that way.
thank you.
Huh.
So when you took at the different doses of your toxin, starting from 0.1 to 5 to something to
1.1 micromolar.
So apparently I did not see any dose response.
Yes, yes.
So what does it tell you?
Yes, that's another very enigmatic observation we found in a cell type different manner. That data I showed for epithelial cells.
Okay.
There we don't see any kind of, we don't have answer. But you correctly pointed out, in epithelial cells, we don't see any dose response.
Like
but
go to the half the dose?
Yes, we have even gone to much lower dose. Whatever dose we can go to a detectable level for further binding studies, etc. We did not see much difference. But in case of macrophage, for example, there is very nice dose dependence. So we concluded that, in fact, reviewers asked us the question, why don't you see the dose difference? So we had to kind of hand wave saying that maybe macrophages are more susceptible compared to epithelial cells, but we don't have the answer, but we see that difference exists.
So what type of language you expect between lysosome and the mitochondria?
Crosstalk, to be very honest, we are still at the learning phase, what is happening there. So mitophagy can be one possibility, what Dipanjan was mentioning. Now, what we think, I think we don't have the solid, robust data, reproducible data yet. We have tried exploring clathrin-dependent pathway. We tried exploring caveolin-dependent pathway, but mostly using inhibitors. None of those things could give us any conclusive result. So what we think it is happening here, some kind of mixture of receptor-mediated endocytosis could be happening, and then some kind of partitioning of lysosomal degradation and fusion with lysosome mitochondria. We need proximity. Yes, yes.
That the proximity is needed?
We could not resolve this, you mean binding, that's what you are talking about.
And also subsequent events.
Yes, I mean, we could not resolve it in a very, I cannot confidently tell anything in that direction. So that's what I can tell at this moment.
Any questions from the students?
I have a very nice talk, Kaushik. I have a very fundamental question, just for my knowledge. You said these toxins are soluble, right?
Yes.
So when they insert into the membrane, just like other transmembrane,
yes.
how they stabilize? I mean, do they have a stretch of hydrophobic region or something?
Yes. So sometimes there is a confusion because of two things. There are a class of proteins which are called porins. These toxins, they are not porins. Porins are outer membrane protein present in mitochondrial membrane, or in case of bacteria, bacterial outer membrane contains porins. These pore-forming toxins, these are actually exotoxins. They are well-behaved soluble protein, and the point you are asking, when they are released, there is a particular motif and stretch that are varying from one toxin to another, but there is an amphipathic stretch present in any of these molecules which remain kind of sandwiched in the core body of the protein structure. Whenever they find target cell membrane, that triggers structural reorganization in the molecule, cooperativity. As a result, this membrane inserting segment opens up, refolds, insert into the membrane. And that is one of the things that we have been studying for last 10 years or so.
There are no more questions. We'll move up and move on. And our next speaker is Dr. Moumita Bhaumik. She's from Division of Immunology, ICMR NICET, and she did her PhD from IICB, CSIR Indian Institute of Chemical Biology, followed by an assistant professor in molecular biology in Sripad Singh College, and finally joining at ICMR, National Institute of Cholera and Enteric Disease, from 2017 onwards. Okay, she has multiple awards and honors like Indo-French Provis 1 fellowship from CEF-IPRA and AK Memorial Medal for first class, first in being zoology from Kolkata University of Calcutta. Her topic is of presentation is very interesting. She's going to tell us physiological networks and disease functions of RNA binding protein AUF1 in inflammatory bowel disease. And being RNA binding proteins are playing very important roles in various things. So I'm really curious to know how this AUF1 plays a role in the inflammatory bowel disease, in the disease pathology. Technical problem.
Okay. Okay. Hello, everyone. So I started my journey in NICET 7 years back. Now it has metamorphosed to NIRBI, National Institute for Research in Bacterial Infections. So I work on okay. So I work on inflammatory bowel disease. Briefly introducing the disease, this doesn't cause mortality as such, but it causes enormous morbidity. So it has two types. One is ulcerative colitis and Crohn's disease. As you can see from this diagram, ulcerative colitis, it's in the colorectal region, and be patchy inflammation throughout the alimentary canal. And this is the present scenario of in India, how the disease is rising. And if we consider the disease, the pathophysiology of the disease, it causes intestinal dysbiosis, means the gut microbes get disturbed, and obviously the metabolites that are produced after fermentation of by these gut bacteria through the fibers that we eat, one of them are the short chain fatty acid that is also reduced. A lot of studies are being carried out on the T regulatory cells and how the inflammation, it causes the inflammation. So my lab focuses on the RNA binding proteins. And by RNA binding proteins, I mean the class of RNA binding proteins which control the stability of the mRNA. When either it binds to the mRNA and causes decay, or it binds and make the mRNA stable. So if you imagine IBD as the elephant, and in each arm, my students and my team, they are trying to look at different problems of the IBD in different angles through this RNA binding proteins. Today, that one I'm going to talk about is the metamorphosis of a cell. So I give the reference of Franz Kafka's famous story, Metamorphosis, where the hero, Gregor Samsa, wakes up in the morning and finds that he is metamorphosed into an insect. So like that, we are studying the B cells, and these B cells, sometime after some conditions on them, they are metamorphosed into the beta-10 or IL-10 producing B cells.
So how this metamorphosis occurs will be my story. So we took baby steps and we started looking for the IL-10 producing cells in the splenocytes. Whole splenocytes we collected and treated them with three different short-chain fatty acids, the butyrate, acetate, and propionate, and we got an enhancement in the total IL-10 production. So we focused on two types of B cells, B1A, which is characterized by CD5, means CD19, CD5 positive cells, and B2 cells, CD23 and CD19 positive. And when we get at this population and look for the IL-10 productions, we found in the B1A cells, we found with butyrate treatment, there was an enhancement in the IL-10 producing B cells, whereas we did not find any change in the B2 cells. So since butyrate was more effective in producing IL-10, for the further studies, we took butyrate for the further studies. Now, it was already reported that how T cells were stimulated to produce IL-10 by a particular cytokine called IL-27. Now, what is this IL-27? IL-27, this belongs to the family of IL-12 family. Now, it has two subunits, P28 and EBI3. This complex of IL-27 together or independently, only P28, it binds to the IL-27 receptor and it signals to produce IL-10. In T cells, it is already being reported. So we verified this finding that was reported, and yes, in CD4 T cells, when we charge them with with recombinant IL-27, we found an increase in the IL-10 production in the gated population of CD4. Same thing happened with the B1A cells, but not in B2 cells. Now, the obvious questions that came in our mind, whether butyrate can increase this IL-27 production or not. So we treated those whole splenocytes and checked for the IL-27 mRNA and the protein level by ELISA and by Western blot. So we got an enhancement, a dose-dependent enhancement in P28, but EBI3 did not have any change in their expression. Moving on to and correlating whether butyrate can drive IL-10 production from the B1A cells, we took the cells, the whole splenocytes, and treated them with either the green one is with recombinant IL-27, the red one with butyrate, and the violet one, we treated the cells with butyrate and neutralized with anti-IL-27 antibody. And we get it for CD19, CD5 positive cells, that is the B1A population, and checked for the IL-10 production from this. And not only that, we sorted out the population of B1A cells and treated this with this following, and we got, there was also a reduction in the IL-10 production in the supernatant. So as the cell was turning to produce IL-10 and becoming regulatory in functions, it started developing or producing certain appendages like PDL2, PDL2 team 1 and others also. So I have shown here two, but there are other appendages, means other molecules also we have checked, and we found that yes, the cell was turning to be a B10 in nature as well as in function. So what butyrate was doing? Now it was reported that butyrate can regulate the many cytokine mRNA expression. So already we worked with butyrate and we have seen that it activates lot of RNA binding proteins. So coming to butyrate and the mRNA expression, we checked whether it changes the turnover of IL-27 P28 mRNA or not. So what we did, we treated the cells with actinomycin D to stop the transcription, and we checked the T-half of this mRNA. So it was already reported that TNF-alpha, what the mRNA of TNF-alpha was decreased by butyrate. So we took it as a positive control, and we found here, in case of IL-27, with butyrate treatment, the half-life was increased, and GAPDH was the control. So I have made this just a metaphor. That's my favorite painter, Gustav Klimt, who shows the death and life in his painting. Similarly, butyrate can also dictate the life and death of the mRNA. So now what butyrate was doing or activating which RNA binding protein? Now there are a lot of reports and we were working on a RNA binding protein from previously, which is known as AUF1. Now this RNA binding protein, it binds to the 3' UTR of mRNA and can cause stabilization or decay parallelly with this. This is a kind of funny protein. It has 4 subunits, isoforms, and each isoform has an RNA recognition motif in singular form or in multiple form. What happens when it binds to any mRNA, it forms in a dimer form in one isoform. Say, for example, this one is one isoform, it binds and then it calls for another isoform and it can change its conformation, and by this way it can dictate whether it's going to stabilize that particular mRNA or destabilize that mRNA. Now how butyrate increases this AUF1 expression, that was a separate story I will not discuss here. But what we see that when we treat the cells, this is splenocytes, with a dose-dependent manner of butyrate, and we get an increase in all the isoforms of AUF1. So having found that AUF1 increases on butyrate treatment, the question comes whether AUF1 actually binds to IL-27 mRNA or not. So we did an RNA immunoprecipitation, and we found that in that immunoprecipitation, we found that IL-27 mRNA was associated with AUF1. Now, which of the isoform? It has 4 isoforms. So we have EGFP construct for each isoform. And so we transfected each cell. We took the raw cell line and transfected it. And then we did an IP with EGFP. And we found out that it binds to P42 and P40 of the isoform. Now, at which part of this mRNA it binds? So we cloned the 3' UTR of IL-27 with EGFP, and here also when we knock down AUF, we get a reduction in the fluorescence, and which does not come back even with butyrate treatment, whereas in the mock, we get an increase in the fluorescence with butyrate. So now it was clear that butyrate, IL-27, and all this phenomena was at par. But now, since we are a very new and I don't have that capacity to do a knockout or pre-flox system, so we came to an already known system called Morpholino, which is already an FDA-approved therapeutic used in Duchenne muscular. Luckily, I got a collaborator from CS who developed this Morpholino, and which is cell-penetrating form. He used it already in the cell line. The first time with him, we tried in the system, and we published it together, and we found that although it's not quite a good model, it has no specific organ where it knocks down, our target can be achieved as it knocks down fully in the liver, in the colon, and to some extent in the kidney and heart. Now we looked for in the spleen also. So we found here in the spleen also, it was knocked down in all the isoforms. So we standardized the treatment, and the treatment, and we knocked this AF gene in the model, injecting this Morpholino in 2 doses. And to prove our hypothesis, we fed this mice only right. Now, when this SMO stands for scramble or the mock control. So if we look, it was here in the first blot, the AUF1 is knocked down when we treat with Morpholino AUF. And here also, you see, there's a reduction in IL-27, which was not restored even after butyrate treatment. What happens when we knock down in the model. Does it affect on the IL-27 mRNA decay? So we took the splenocytes from there and looked for the number of IL-27 mRNA. And there also, in one knockdown, we found just a reverse that mRNA half-life was decreased of IL-27. And this was true with IL-10 producing B1A cells. As you can see, there was an increase with butyrate treatment, the IL-10 producing B1A cells, which reduced on B1 with AUF knockdown and also did not restore with butyrate treatment further. Now, to prove that IL-27 mRNA turnover is being affected, it must reflect on the protein synthesis. So what we did, we took the cells and stopped the translation with cyclohexamide, and we made a density gradient. And as you can see, in the lower region of the fractions, you tend to get more polysomes, and as you go higher up, you get more free mRNA. So when you see, compare between the scramble and AUF1, and if you see the pattern of polysome, and this is the actual pattern that we should get, here we get in the lower fractions, the polysomes, then the subpolysome, and this is the free mRNA. That is similar almost to what we got in both scramble control and the AUF knockdown mice. And in IL-27 mRNA, from each fraction, we did a qPCR and found out how mRNA is associated whether with the polysome or in the free form. And we found a shift towards the free mRNA, means the protein synthesis also was affected. And the gap age was the control here. Now, to prove the full pathway, what we did, we collected or we sorted the population of B1A cells from the wild type mice and the AUF knockdown mice, the B1A cells, and in vitro, we collected those cells and treated them with either butyrate or recombinant IL-27. And with recombinant IL-27 and with butyrate, in case of control, there was an increase in IL-10 production. But when we knock down, the butyrate treatment cannot enhance the production of IL-10, whereas recombinant IL-27 can. Only 2 minutes, sorry. So I will go faster. So the thing comes like that. Butyrate, it stimulates AUF1, IL-27, then IL-10 production. So when we were working with the AUF1 knockdown, what we found, we were taking the tissues and we found that in the colon region, found a colitis being developed in this AUF knockdown mice. And we found that the gut length was reduced and the histology of the gut also were like colitis. And we also found inflammatory cytokines. Now, to prove our hypothesis, we were collecting samples from the human IBD patients, from the IBD patients in the colorectal biopsy tissues. While collecting that, we also collected some non-inflammatory from the control, means non-IBD patients. So, we started developing organoids from both the tissues, and this I show the data of the control from the tissue that we took, and we have made the organoids, and in the organoids we AF1 even with morpholino. The first time we showed that with morpholino you can knock down a gene expression also by using morpholino in the organoids. So we got a reduction in the AUF1, and when we reduced the expression of AUF1 here, we also found an increase in the inflammatory cytokines, reduction in the IL-27 and IL-10. And what happens to this pathway? We were talking about spleen, and now when we found that that AUF knockdown mice was developing colitis, and when we knocked down in the organoids, it also starts producing inflammatory cytokines. We wanted to look what happens in the IBD patients, and here also what we see, there was a reduction in the AUF1, IL-27, IL-10, and increase in the TNF. And that was with the immunohistochemistry. Show here that the butyrate rescues with TSS-induced colitis. I will not talk about much with this data, just for the time's sake. And here we show that not only spleen, but the same thing was happening with the colon tissue, or we have tested with mesenteric lymph node cells and also the colon tissue. The hypothesis of AUF1 IL-27 beta production was found to be restored with butyrate treatment. I come to this interesting part where the immune cells, we took the explants from the colorectal tissue, and with those explants, we treated them with butyrate. When we treated them butyrate, we found that there was an increase in AUF, IL-27, IL-10 as expected. But the important thing that we observed, we wanted to look for the betaine cells in the explant tissue of human by doing FACS. While doing that, we did not find a correlative recovery with the butyrate treatment, and that where we stuck. Now we became inquisitive more why it was not restored to the original level. Now when we map that with the score of the IDBD patient here, and each individual point represents that particular individual, where it is correlated here, and with increasing the score of ID, more active the disease is, the betaine population production, betaine synthesis, or the butyrate response to this population is is not found. Sorry. So if we look at the higher score, the butyrate response to this particular patient's explant was not there, whereas we could find a response in the lower scores. So just last slide, I want to show that to prove all this theory, what we did, wild type mouse and the AUF knockdown mice, we collected and sorted the B1 populations, and there we treated it with butyrate. And we call it B1A wild and B1A knockdown. And we adaptively transferred the colitis mice and looked for the recovery. And what we found that in wild type was a recovery from colitis, but there was no recovery when it is put knockdown mice. And that was shown by the inflammatory response, the GART histology, the disease index, and also by the survival curve. So we have two phenomena that I have spoken today about the activation of IL-27 by butyrate. That is, when butyrate comes, it activates AUF1 with some IL-27 mRNA and stabilizes the IL-27. Now, it's being produced by the antigen-presenting cells. So it can come from the B1A cells by itself, or it can come from macrophage or DC. And it binds to the IL-27 receptor, it signals the B1A cells and polarizes the B1A cells to produce IL-10. I thank my students. The work is mostly done by Oishika. She independently developed this organoid culture in my lab. These are my collaborators, the clinical collaborators, Dr. and Dr. Rajiv Sharkar from SSK. My funding. Thank you.
Okay, although that talk is open to discussion, we might not have much time, probably for two questions. Yes.
Very nice, Doc. Quick question. This model, that in vivo model you have developed, that there is a lack of clarity on the cell type specificity, right?
Yes.
Where exactly this is, I mean, one would expect dendritic cells to be important for the entire pathway. But AUF1 being an RNA binding protein which binds to RNA rather promiscuously, Many other target genes in many
Yes.
other cell types would be affected.
Yes.
And the concern remains that this might be in part due to T cell effects, right?
Yes, it has T cell effects.
So
Because
how
we are finding that it affects ROR gamma T and the FOXP3 regulation, FOXP3 transcription factor. In one case, it is positively, means it causes stability. In one case, it causes decays. So by this way, it can affect T and Treg balance.
So the effect that you see, how would you sort of figure out exactly it is because of the regulatory B cells and not because of lesser regulatory T cells?
It is there. It regulatory T cells are there for particularly focusing on B cells. I should have got some mice which are devoid of T cells or B cells. That facility I do not have particularly. So this is a problem. It's a general problem, but AUF is not restricted to the B cells. You are right. It is the same thing happens in the T cells. And IL-27 is also present in the antigen presenting cells and including the B cells. Of course, it's an antigen presenting cell, but it's not present in T cells. So IL-27 effect is not present in the, means it cannot be produced in the T cells. But yes, of course, it is with the T cells and the B cells.
One more question. Students from back, any question? Okay, doesn't look like any more question. Actually, I have a question.
Yes, sir.
Very nice talk, illuminating talk. Do you know how AUF actually induces mRNA decay? I mean,
We are studying that. In one case, it is decaying, as I said, it is stabilizing. There can be a lot of many factors. One of the factors that we got recent data that it acts sponging out the microRNAs. So the microRNAs, which, yeah. So this is one of the mechanisms that we are, and means we are working on that.
okay. Okay, thank you again for your illuminating talk. And with that, the session is kind of closed, and both the talks are very, very illuminating and interesting. So with that, I would like to thank both the speakers for their excellent talk. And before ending, I would like to share my thought. We are both honored to be here. And for that, at the outset, I would like to thank the organizing committee and especially our beloved DJC sir. And it's an honor to be here chairing the session in front while he is in the audience. Thank you, sir, yeah. Okay. So over to the organizers.
Wonderful session. The speakers are requested to come to the stage once again for felicitation. I request the chairpersons to felicitate the speakers. Hey. Yeah. There is memento. Now I request the joint conveners. Okay, the tray is missed. Now I request the joint conveners to come up on stage and felicitate the chairs. Thank you. Before going to the next session, which is the panel discussion, there is a short announcement for the volunteers to gather upon the stage to take a group photo. The volunteers, can you please come on stage for the group photo? All the volunteers, can you please assemble on the stage for a group photo? Then we'll be quickly moving to the panel discussion, and that will be the last scientific session of the day, before the valedictory session. This event wouldn't have been possible without the volunteers. Their efforts are really remarkable. All our students, big thanks to you. আমরাও থাকবো। I will request the volunteers again to replace the table for the next session. Table in the middle. Thank you. Thank you. That's fine. ঠিক আছে সুজয় কুমার দাশগুপ্তা ওদিকে থাকবে হ্যাঁ ওদিকে থাকবে আর ওনারা দুজন একজন মডারেটর তো আর দুজন Okay. Thank you. Now I'll request the moderator for the panel discussion, Dr. Sujoy Kumar Dashgupta, to come up on the stage. Dr. Sujoy Kumar Dashgupta is an emeritus scientist in the microbiology department of Bose Institute. He is a chairman of the microbiology department. He is a recipient of West Bengal Academy Fellowship and is also a CU visiting faculty. His research interest is in Mycobacterium tuberculosis. He will be moderating the session. The topic of the discussion is microRNA and its role in post-transcriptional gene regulation. And the panelists are Dr. Gopal Chakrabarti from Department of Biotechnology, University of Calcutta. Sir, please come up on stage. Dr. Dipanjan Ghosh from National Institute of Pharmaceutical Education and Research. Dr. Ghosh, please take your seat. Dr. Nirmalya Dey from Amity University, Kolkata. Please come up on stage. Thank you. Over to you, sir. You can speak from here also.
It is pretty late in the today. I'm very happy to be here to moderate this session on microRNA. And I have my co-panelists over here, starting from Dipanjan Ghosh from Naipar, Kolkata, Nirmalya Dey from Amity University, and my good friend, Professor Gopal Chakrabarti from the Biotechnology Department of Calcutta University. And so this is a panel discussion. And apart from the experts who are here, I can see many of our scientists who are also very experts in microRNA. So as we discuss the issue, you're free to interact with us and give your inputs about the latest developments in the area of microRNA. Can I? Yeah. Yeah. So I'll just start off with a brief introduction since it is pretty late. And the reason why we are here is 2024, this year's Nobel Prize has been awarded to Victor Ambros and Gary Ruvkun for their discovery of microRNAs and its role in post-transcriptional gene regulation. And to give you a brief introduction, okay. So 1953 was a monumental year for humanity, and the structure of the DNA was discovered, and from the structure of the DNA, it became evident that how the centrality of the DNA in all living processes and the central dogma of life, which is DNA makes RNA makes protein. So everything was centered around the DNA, but in due course, many other interesting developments happened. For example, David Baltimore and Howard Temin discovered the reverse transcriptase, which means that the central dogma had to be modified in that the RNA can be reverse transcripts to DNA. So in the modern era, the central dogma of molecular biology is pretty complicated with the participation of various more elements. Somewhere, I think, in the 1980s, Thomas Cech and Sidney Altman discovered that the RNA can itself function as an enzyme. And with these concepts, various concepts, gradually it became clear that RNA has much more important role to play, just not for acting as a messenger RNA, but for doing many kinds of things. And from that, the concept of the RNA world has evolved. Now coming to, okay, so at a particular time, it was believed that yes, DNA is present in all the cells, but the question is, why is some DNA not expressed in some cells and expressed in other cells? So the Jacques Monod and Francis Jacobs model suggested that a gene is selectively expressed because a gene is selectively expressed because of the involvement of transcriptional factors. So because of these transcriptional factors, some genes are expressed, some genes are not. But so that was acceptable for a long, long time until the early 1990s. It became clear that it is not just the transcription factors, but these small RNAs called microRNAs, which have a significant role to play in manipulating gene regulation. So this story of microRNA starts from this worm called the roundworm Caenorhabditis elegans. And this was Victor Ambrose, they observed a particular small RNA in this called LIN4, which was regulating a gene called LIN14. So that was the time it was discovered, but people could not understand the implications fully until another microRNA called LET7 was discovered. And it is interesting, initially people thought that this is something C. elegans specific, but with the discovery of LET7, it became clear that LET7 is present in all organisms and it is evolutionarily conserved. So the discovery of LET7 convinced us that this is a much more fundamental thing than some kind of an isolated phenomena. So from backstage to front stage, what was initially thought that RNA plays a role just as a messenger RNA, now we can see so many different types of RNAs. And so as many of the speakers were emphasizing about the RNA, centrality of the RNA, and the RNA binding proteins, for example, with the RNA binding motif, there are some of the very ancient motifs. So now we have so many different types of RNAs, starting from messenger RNA, transfer RNA, ribosomal RNA, snRNA, SNU-RNA. And the topic of our discussion today is on miRNA and also the related phenomena called siRNA. And Dr. Dipanjan Ghosh will be talking about another related RNA called the long non-coding RNA. And of course, we have the CRISPR RNA. So, there is some kind of a evolutionary conservation in the whole process. So the nature uses RNA and this ability to hybridize with messenger RNA or DNA to either degrade them, to activate them, or so many other processes. So we will now go into the panel discussion, and I invite Nirmallo to give his insight about how microRNAs control, and particularly he will be talking about the biogenesis of microRNA, which is another very exciting area. Please, Nirmallo.
Thank you, sir. And I also would like to thank the organizing committee for letting me the opportunity to be a part of this revered panel. So without wasting any further time, let me talk a little bit about the biogenesis of these molecular Davids, that's how we can call them, which have a very huge potential to find out, target and bring down their target Goliaths. Now, talking about the microRNA biogenesis, they can be transcribed both by RNA polymerase II and RNA polymerase III in the nucleus, in our gene. So they can come from the exonic part, intronic part, or the intergenic parts. Okay. Initially, they are going to produce a huge transcript, maybe of like 5 kb of 6 kb of size transcript. So that is one is called a primary microRNA. Now then comes to the step of the maturation. Now into the step of maturation, it can either have a canonical maturation or a non-canonical maturation. In canonical maturation, it goes by a drosha-dependent pathway. So this is one type 3 type of RNAs, which is specific to crop the double-stranded RNA structure. Cuts in some staggered cuts it's used to produce. So these primary microRNA, which is, as I mentioned, that it is a little bit huge, have a lot of hairpin kind of structure, a stem loop kind of structure. So this drosha RNase enzyme, what it does, that it cuts and crops the extra parts, the linear parts, and only leaves the stem, which is sizing around maybe 20 to 22 bases, with a terminal loop. And this one is called the premature microRNA. Apart from the drosha-dependent pathway, it can also be processed in a non-canonical pathway, as I just mentioned, which utilizes and which mostly acts on the midtrons or the intronic microRNAs, which are coded from the introns of the genes, which utilizes spliceosome and the Laria debranching enzymes. So that doesn't include the drosha. Whatever the thing, be it a canonical, be it a non-canonical, be it a drosha dependent or independent, they both ultimately comes down after the cropping, after the initial trimming to a level of around 70 to 90 bases product, which is called the premature microRNA. Now this premature microRNA comes out from the nucleus to the cytoplasm utilizing the RAN-GTP and exportin pathway. Once outside from nucleus to the cytoplasm, now it goes for the next layer of processing, next layer of maturation. And that utilizes another ribonuclease RNase3, which is called dicer. So this dicer is not now going to crop that loop part that is associated with the stem loop or the hairpin kind of structure, and now going to leave only the stem part, the double-stranded part, which is sizing around 20 to 22 bases. Now, this is, as far as the RNA molecule is in concern, is the mature part, but this is not at all, as of now, is the functional molecule in terms of mediating the gene expression regulation. In order to make it functional, it needs to interact and it needs to recruit its protein partner. And that one is Argonaut. Argonaut and its adapter GW182, which is a 182 kD protein rich in a particular type of motif with glycine and tryptophan. So these Argonaut going to choose from that double strand one particular strand to be its interacting partner to produce the RISC, RNA inducing silencing complex. That one is called a guide strand, whereas the other one is called a passenger strand. Now, there is certain pathways, certain thermodynamic features, certain characteristic traits they used to find. The one that is thermodynamically less stable among that double-stranded structure is actually chosen by Argonaut to be recruited to make the final RNA-induced silencing complex. Previously, it was idealized that passenger RNA's work is done as of now and is going to get degraded. But recent work also has found out that passenger RNAs may not always go for degradation. That can also be recruited with Argonaut to produce another different kind of RNA-induced silencing complex as well. Once this microRNA is loaded with the RNA-induced silencing complex, the Argonaut and GW182, then it is the functional molecule. And its uniqueness lies in the fact, if we compare with these double-stranded RNA molecules, it usually works as a defensing molecule, okay, as a defender, protector. But in terms of the protection, because generation of the siRNA, silencing RNAs, they also follow the certain similar kind of or overlapping kind of procedure, overlapping kind of pathway. But their critical characteristic is that they used to completely degrade their target messenger RNAs. Whereas microRNAs, in many cases, because they used to do that because siRNAs always used to produce a perfect complementarity with its target. Whereas microRNAs do not usually or characteristically make a perfect complementary structure. It always used to leave a bulge. It always used to make a perfect complementarity for couple of, from position 2 to 8. That one is called a seed region. That actually determines its activity, but it has to follow a bulge, follow a perfect kind of a incompleteness or imperfect complementarity from position 13 to 16. So that's why, apart from making the degradation, causing the degradation of the target messenger RNA, they always, in many cases, used to produce a lot of translational repression, either by distorting the ribosomal assembly, or by distorting the ribosomal structure itself, or by decapping, or by deadenylating the 3' end of the tail part of the messenger RNA. It can either cause the degradation of the messenger RNA as well as can cause only protein repression, keeping the mRNA or the target molecule intact. So that is about the biogenesis and getting to the functional molecule. Now I'd like to hand over to Gopal sir for his further words.
Yeah, thank you, Nirmala, for presenting in such a lucid way the biogenesis of the microRNA as well as siRNA. And he clearly gave the distinction between microRNA and siRNA, although they are processed almost in a very similar way. So, so now I would request Professor Gopal Chakrabarti to give his idea on the application side of microRNA. And so, please.
I'm going to show few slides. Okay. So which one is this one, right?
So just to point out another distinction between microRNA, if I correct me if I'm wrong, that microRNA acts on the RNA of the own cell, whereas siRNA is targeted to various viruses and other extra DNA coming from outside. And yes.
Yes,
So,
sir,
so
right,
with
right.
that little bit addition, go ahead, Gopal.
And that's why mRNA are more in the development and other kind of part as well, whereas siRNA is more like defensive, more
Yes,
like a
so it's
protective.
a more defensive and the other one is more regulatory.
So thank you for organization, for inviting me. Also special thank to Professor D.H. Chakrabarti, who was also one of my colleague in Calcutta University and my neighbor lab, basically. So every day we were interacting lots of things about science and other things also. So I am not directly RNA person actually. I worked on basically moving. Not moving. Okay, this one. No. So I worked on basically anti-cancer drug development that again, so drug protein drug interaction. Okay. So it's a anti-cancer drug development. That is the major field of in my work. So how I came to the microRNA area. Just very briefly, I'm in the interesting story. So actually, next slide. Okay. This is not moving. So actually, hello. I working on drug development, and also one of the interesting thing is to me that the drug resistance actually how drug resistance also is very important area of work develop, because you know that in cancer certain time after treatment there's a resistance occur. So that is the for successful therapy you need to also address the resistance mechanism also resistant area. So we are working on that field last say more than 12 years. So one of the area you could see that the defined area of drug resistance. So one of the two area very important, that is the cancer cell actually increase the upregulation the autophagy phenomena. Autophagy phenomena is basically that if you give a stress, it actually some stress is it destroys some the organelle or even protein. They actually reuse, they recycle the process. It's a different process. Steps are the lysosomal degradation, and that actually use cancer cell to develop the resistance. So we working many years on that field, and we found that autophagy is definitely a different important step for development of drug resistance. So then actually one of the ideas come that how autophagy is increasing or using the cancer cell for development, whether the mechanism, one of the protein is that's the Becklin one. Becklin one is actually upregulated during autophagy. It's the initiation state of autophagy. So we then try to do to understand whether microRNA is involved or not that. So I actually did have the idea, experience in ION. So I went to my neighbor, Professor D.H. Chakrabarti, whether we can collaborate he can help us to little bit about RNA work. So he agreed and we started the work. And you can say that I'm just moving. Okay. Hello. It's coming. That thing is not coming.
Do you have a video?
emphasize on this.
That is the result I want to show. That is the my array. That is the data basically. That is the data for this real. So not this coming not. Why not established? That time it was M.
Anyway, you can just summarize.
Something resolution problem is.
Me another. Okay. So that is actually I want to talk that area. So what is that is very I don't know. Otherwise, meaningless. So we develop the
we can see
resistance.
some upregulation and downregulation. Some microRNAs like
So
HFB103.
what is no, just wait.
285 to 204. Some
So you can
microRNAs
suggest visibly I'm showing that. Now trust students, I know that. But actually, for student scholar, I like to show the array. So this array actually at that time when we did that in India there is no facility. No company actually is a 12 years back. So we had to send to outside. So one company, Oxygen company for Europe. So they have the agent. So I contacted with them, they're Delhi based. So I sent the RNA purify. One time RNA said that quality is not good. Then again purify. That's a long story. So I sent that. Then they did the array. So you can see that there is a 4 actually sample. 2 are control. That is a sensitive cell. Paclitaxel is one of the highest using clinically drug. Okay, and another that is the red one is control C1 CR TX1 and TX2. That's a duplicate sample for taxal resistance. Okay, paclitaxel resistance cell. So here actually there are several microRNAs upregulated. Blue means downregulated, red means upregulated. Okay. So we found that one of the microRNA, that is the microRNA 17, that actually checked for software. There is a software the target you can say, and then we tested some real-time PCR and other method. That is actually directly target the Becklin-1. Okay. And another microRNA that is very important. I show you that apoptosis process. That is the apoptosis. Downregulation apoptosis also cancer cell actually used for development of resistance. That is the microRNA 16 actually. That is also downregulated and apoptosis basically BCL2. This targets the BCL2. So we did a lot of work, all established and then published the paper, to executive paper. And one thing now is working after that. So you can see that they are actually not showing them. There is several microRNA upregulated and downregulated in the resistance cancer cell. So there is definitely a network. So you know that in a particular cell, not single event is not happening. There is a multiple event, multiple thousands of protein are present, thousands of gene are expressed in any event. So it should be something just trigger the resistance. So we actually, what we develop, the sequentially resistance, we develop the sequentially resistant cell, and definitely next stage. So which microRNA or which part company group of microRNA is triggering the initiation of resistance, that we can understand through array method. Okay. So I'm stopping, and maybe next I will go after question.
Well, yes, this is a real demonstration how microRNA can play a significant role in the development of cancer or development of drug-resistant cancer. And to mention that there are maybe, how many, there are about 5,000 or how many microRNAs have been reported in human?
As of the latest Mir-based Sanger database, the number of prematures are like around 49,000.
So 49,000 microRNAs have been...
No. So there in database around, but predicted is around 2,500, and the database is 1,100. That is annotated.
So, okay.
Say
So
human. I'm talking about human microRNA. So
talking
before
about all,
total-
yeah.
total-
It's
total-
from around 470 on organisms. If we look into from
Yeah, so
470
we
on
are human.
organisms,
we
have
are
a,
talking about
uh,
human. So even
position.
if you take a human, it will be around 11.
11.
So that's a
11.
huge number of
11
microRNAs. And I believe that there is a particular procedure to for the nomenclature of the microRNAs. And isn't it? Probably. And yes, and what his experiments show that there is a possibility to have a microarray panel and try to find out which microRNAs are induced under which conditions. And I believe there are many in the audience who have done a lot of work in the microRNA also. But due to lack of time, we will now go to Dipanjan. Dipanjan will talk about a very interesting topic. The microRNA is no longer micro, it has become very long. So there is another group of RNAs called the long non-coding RNA, which are also have a lot of impact on the gene expression. And Dipanjan will take us through the long non-coding RNA story. Please.
So most of these students are here, so I want to talk concerning to the students specifically. So I believe that you understand the basic difference between the DNA and the how many of you don't know the chemically how they are different? I believe all of you know that, right? So can you tell me what is the actual our genome size? Human genome size? So 3.5 billion is the size. How many of this sort of genome is encode for the protein? mRNA. How many? How many percentage of our genome?
Dipanjan, I believe they have just completed one semester. Please don't stress them.
Okay. It is only 3% of it, less than 5%. So rest of them people say junk. Today it's not junk, right? So most of them from these, what you said, the junk, the intergenic DNA, all these non-coding RNA comes from, or right? And so next, people used to think non-coding is they don't. The literature, see, with the invent of the RNA-seq, cDNA sequencing and all this thing, their number came exponential. Now, can you just guess what's the number of the LNC RNA genes now present? I was just searching yesterday. What is the number? What can be the approximate? So I was surprised even. It was 95,000 genes are present. Their transcripts are 3.25 lakhs. 3.25 lakhs transcripts that come from the 95,000 genes. All right? Then Where from this LNC RNAs come? Definitely from your genome, it is transcribed. And where from it is transcribed? Can be transcribed from the exons, introns, as Neha already pointed out, intergenic sequences, or from the pseudogenes. More than 10,000 LNC come from human, from the pseudogenes. Okay, so now you know they come from your genome, but not from only exons or introns, from also intergenic sequences. And now they have some similarities between the mRNA. Scaffolding, okay, decoying or sponging. So what does guide do? When they act as a guide, they bind your transcription factor, they guide the transcription factor to bind to the operating site. So they can bring your activator or inhibitor of the transcription, this way they can regulate your transcription. Another way, they can be part of being a scaffold, they can integrate all the complex molecules that have involved your epigenetic modification. Okay, it does the epigenetic modification of your important genes, and therefore gene expression they can regulate. And I told that decoying means they can bind a microRNA or mRNA and they can cleave it, or sponging. I think the previous speaker also talked about sponging. They can bind and they can inhibit some of the mRNA or microRNA. These are the more or less 4 options they do, the way of them mode of actions. But most importantly, nowadays you see, you name a disease, there should be some of the micro long non-coding RNA. They have been there. You can see they are very much important. Okay, you take any disease like cancer. You got any cancer, you see many. And this list is going on day by day. All right, even for diabetes, metabolic disorder, or inflammation, IBD, these things, or you can go for neurodegeneration, Alzheimer, all right? So now, interesting thing, if you get to so many LNC RNA, but you see 4 or 5, they are involved in many diseases. People have done the part. Okay. So I will just give you the 2 or 3 examples. These LNC RNAs are involved. Most important is one LNCRNA, they have a one role in one disease model and different role, same RNA, LNCRNA, in the other disease model. Okay, just like I'm giving one example, if you see, you will find everywhere there is some LNCRNA called MALAT1, hot air, NEAT1. These are very common, H19. And they are involved in many diseases. So just like one example I'm giving, MALAT1, it is called metastasis associated long adenocarcinoma transcriptome. Then suggest in cancer, it is involved in the metastasis. Okay. But in Alzheimer's disease, it is in actually anti-proliferative. Stop proliferations. Okay. And just hot air, in cancer, it is actually blocks the microRNA involved in gene suppressions. Okay, but for Alzheimer's disease, it actually methylates the amyloid beta chains. So these are the things when you even in your disease model in the situation when it changes, its role also changes. Right. And another last one I want to tell you, NEAT1. That NEAT1 is a very important one, interesting one, because not only we are working on this, because it makes a parasiticles. In the nucleoplasm, this LNCRNA, they bind some of the nuclear protein. And this 3D makes a proper 3D structure without okay. And this RNA protein complex, they have some biological role and they are placed in the nucleoplasm, in the nucleus. Alright, and they can recruit various transcription factor and they can mediate splicing also. Okay, and most of that various viral infections also, this parasiticles plays an important role, especially influenza virus, HIV. Okay. One, not only this disease, your mood, okay, you are aggressive. There are, I have to read this, there are some LNCRNA that are upregulated, that is called Myeloid. This LNCRNA methylates your chain, which is called the gene, this MOCA gene, sorry, MAO gene, okay, monoadenine oxidase this gene. And interesting, last one I need to tell, there are some people who died of suicide. Their LNCRNA analysis shows there are some LNCRNA like HFOX1 or IRF2. These are downregulated. They gave it the suicidal marker. No, what they did, they analyzed from the people who died out of the suicide. Then from the brain cell, they have done it. And they showed that these are the downregulated. There are differential expressions of the LNCRNAs. So why it is very important? The take-home message is that it is your next gen drug target. Okay, as Sam was telling, it was DNA. Now the RNA has become very important, and these RNA, as you know, they are more in number, and they are involved in very physiological conditions in disease. They are involved, and therefore they are believed to be the important drug target. Okay, the problem in the working in this field is the LNCRNAs are expressed quickly, not too much. Okay, and most of the LNCRNAs, they are found in combination binding with the cognate proteins. So you don't see many of the seen in the free. Then most of the cases they bind with the protein. So getting these LNCRNAs in free form when you isolate the RNA, it is a very challenging. Okay, therefore, so I believe this you can overcome in various modification, but in future, what but I believe within 10 years, this the whole transcriptomics of valence RNA profiling can give you a diagnostic or prognostic.
Well, thank you. Thank you.
So in normal cell also, yeah. So normal, it has a role in your transcription modification in normal cell also. In disease cell, it's at a different role also. Okay. So even in normal cell, their expression is so low. So yes, the one is that the epigenetic modifications, right? So normal, I forgot that happens in a normal cell, normal condition. There are sets of LNCID involved in your normal cells epigenetic modification, but their relative expression changes. Even in the context dependent, their activity also changes. And one point is I should mention, because they are most of them are intergenic, their sequence is also different. Many of them.
So GDN is a guide, it's kind of guide RNA that really controls the mitochondrial protein expression in Leishmania. Leishmanial mitochondria. So anything equivalent that you see? Okay, it's very established in Leishmania system.
It is expressed in Leishmania, not
Yes, yes. No, it exists only in Leishmania.
Okay, so I think time is running out. Otherwise, we could have a much longer discussion on this RNAs. Thank you, Dibanjan, for you are a good teacher also. Okay, so coming to starting from Victor Ambrose, I think when he discovered the lean-4 locus in C. elegans, he was wondering how it functions. And it so happened that the lean-4 locus did not code for any protein. So everybody was wondering how this lean-4 locus is functioning. And ultimately, he showed that it doesn't code for a protein, but it codes for a microRNA. So now, as Dipanjan was mentioning, so only 3% of the genome, 3% of the human genome or any other genome codes for proteins, and the vast majority of the DNA actually codes for various kinds of non-coding RNA. So the area of non-coding RNA is open, and maybe very soon we'll be expecting a Nobel Prize on non-coding, long non-coding RNA also. And so we come to the end of this panel discussion, but anybody, those who are more experts in this area, want to throw more light on the microRNA, we can have a brief discussion if time permits. And the students also, if you want to have anything to say on this topic, please raise your hands. Okay, they are all tired, so I think with that we can wind up this panel discussion. So I thank all my co-panelists for giving such a very interesting discussion, and let's see what happens in the area of non-coding RNA in future. Thank you. Please, hold on.
Thank you. I'll request Dr. Shujoy, sir, to felicitate the speakers. Panel. Our volunteers will promptly shift the tables, and then we can go ahead with the felicitation. May I go? May I request Dr. Shuja Dashgupta to kindly felicitate our panelists? Wait, that's it? Okay. And then I will request Professor Shonku Bose, our group CEO of Techno Group, to please come up on stage and felicitate Dr. Shujoy Dashgupta, moderator of the panel. Thank you. With that, I thank all the dignitaries. And that brings us to the end of the scientific sessions. And now we are approaching the valedictory session, the much awaited session for all the students. And I have the names of the winners with me also. But before that, I'll call upon stage our program coordinator, Dr. Rajot Pal, to deliver the welcome address or the valedictory session.
Good evening, all. So, at last, we have come to the end of 2 days scientific discussion of BioTalk 2025. For last few months, few days, for day and night, hard work from all the volunteers, all the faculty members, all the staff members of this Department of Microbiology, Department of Biotechnology, and Department of Applied Nutrition and Dietetics, all have done a splendid job and hard work for which we can come to this end of this scientific session of BioTalk 2025. BioTalk, this event has started just last year. So by these 2 years, I'm very happy that we can reach up to this level, that so many eminent scientists from Kolkata, outside Kolkata, from the other cities of the India, even from abroad, they are trying to come and attend our BioTalk, SNU-BioTalk. So for that reason, I'm very happy. And just while discussing there, before coming here, me and Momita had a discussion that আসছে বছর আবার হবে. So in Bengali, I'm literally trying to say that sure আসছে বছর আবার হবে. And in the next year, BioTalk 2026. So actually, I am not welcoming only for the valedictory session to all of you. I'm also welcoming in advance BioTalk 2026. So I hope that in the next year also, we will enjoy and we will have some very good scientific discussions. And I'm also very happy that within 2 years, we have come to a level that last day I was watching the posters. So many very good research works have been presented, and so many eminent scientists has visited this BioTalk 2025. So that is a challenge for us also to maintain this level and to achieve more level for the scientific discussions and research work. So with that, I am welcoming you all to the valedictory session of BioTalk 2025. So I'm requesting to MC to kindly take forward to the valedictory session.
Thank you, Dr. Pal. Next, I'll call upon stage Dr. Anindita Banerjee, the co-convener of the conference, to summarize the conference events.
Good evening. So as we come to the conclusion of SNU-BioTalk 2025, it fills me with immense pleasure and gratitude to reflect on the success of this international conference themed Symphony of Cellular Signals in Metabolism and Immune Response. Over the past few days, we have gathered to explore, discuss, and expand the horizons of scientific knowledge, and it has been nothing short of inspiring. This event has brought together leading scientists, researchers, faculty members, and students with a total of 250 participants, including eminent national and international delegates who added immense value with their expertise and perspectives. Their presence underscored the global relevance of our theme and highlighted the importance of international collaboration in scientific research. The academic backbone of this conference was built around 3 significant sessions, each addressing a critical aspect of cellular signaling and its broader implications for metabolism and immune response. Session 1 was intracellular signaling processes of cellular metabolism. Session 2, signaling pathways in physiological and pathological processes. And session 3 was signaling in innate and adaptive immunity. A key highlight of the conference was the enthusiastic participation in the poster presentations and model exhibitions by the students and other participants, which showcased the creativity and dedication of our participants. Poster presentation session featured 36 posters on a wide range of innovative topics. Each poster reflected rigorous research and fresh perspectives, providing a platform for meaningful discussions and valuable feedback. The student-led model exhibitions were a testament to the creativity and ingenuity of our younger scientists. These models brought complex biological processes to life, providing visual and interactive learning experiences for all attendees. The enthusiasm of our students shone brightly through their work,
making this segment a memorable and enriching experience for all of us. One of the most engaging features of the conference was the panel discussions on cracking protein complex structure using AI and other computational tools, and microRNA and its role in post-transcriptional gene regulation. Another intriguing session was conducted by the Indian Science, Technology and Engineering Facilities MAP, the ISTEM Government of India, that hosts a dynamic and interactive program linking researcher and resources. Adding to the vibrancy to the event was the cultural program, meticulously organized and presented by the talented students of Sister Nivedita University, Department of Microbiology and Biotechnology. This program was a delightful interlude blending traditional and contemporary art forms. It provides a moment of joy and relaxation amidst the intense academic schedule and reminded us all of the importance of celebrating arts alongside scientific pursuits. As we reflect on the many insights gained during this conference, several themes emerged that includes interdisciplinary importance, technological integration, collaboration and innovation, engagement, and creativity. So, looking ahead, as we conclude SNU-BioTalk 2025, let us carry forward the inspiration, knowledge, connections we have gained here. The ideas sparked during this conference have the potential to drive groundbreaking discoveries in cellular biology, metabolism, and immunology. This conference has been a testament to the power of collaboration, creativity, and scientific inquiry. Thank you all for making this event a resounding success. We definitely look forward to welcoming you to future editions of SNU-BioTalk, and best wishes in all your future endeavors. Thank you so much.
Next, I would request our co-convener, Dr. Moumita Das, to project the reflections from participants. I would request few of the participants to come up.
Well, good evening, everyone. As we conclude the session today, we had some great discussions, some great exchange of scientific ideas. But to move forward, we would like to have some words of reflection from the participants or the dignitaries who are here so that we can have a scope of improving ourselves. So if anyone voluntarily participate in this, we would be very glad. Please, someone from the participants, from the students, or from the delegates, if someone is interested. Can we have some words of reflection so that we can improve? We always believe that there is scope of improvement. So please feel free to raise your hand. We will reach out to you. Anyone from the dignitaries, sir, please? Sir, some words of reflection.
I was here for only a small time. I just came in the afternoon, but I was very excited about the lectures. But one thing to note is that how SNU organizes such meetings. I haven't seen such nicely organized meetings, particularly in biology area. So I think this should be a model for other institutes and universities, how to organize such a topic on such a, no, it's true. I mean, it's true. The student participation and the way you conduct the meetings and everything matters while organizing a meeting. So that is a lesson to learn. And so I enjoyed it very much for whatever short time I was here. And I surely hope to come back. I don't know whether this is an annual event or not, maybe.
Yes.
Okay,
yes.
so please invite me next time also.
Thank you so much, sir. So now Nirmala, sir.
Yeah.
So it's been a real fantastic experience for me, and thank you all for very much for inviting. I especially like to thank Professor Dhruvajyoti Chattopadhyay sir. He's like my. I think fantastic word is self-sufficient. That's what also all the players and everybody used to say, fantastic experience. So I like to make it really crisp and short. So thank you very much, and au revoir, and so long. Thank you.
Okay, so with these words, we now move forward to the prize distribution. Gopal, okay, Gopal sir.
Okay, thank you again. And I also came this afternoon actually, and I had several talk, 4 or 5 talks probably. And what I noticed that your organizer, so cordial reception, that is the great. So very cordial reception, the program, and I enjoy much. The students, scholar, faculty are very, very cordially organized, definitely, and all the body are enjoying the thing. So that is a good thing to me. Thank you all, all of you. Okay, conducting this.
Thanks. Okay, so sir, can we move forward to the prize distribution now for the poster sessions? Amit sir and Sudipta sir.
So I will request the conveners, the joint conveners and the co-conveners to deliver the prizes. But before that, I'll just announce now the ones sitting in the back. This is for you. The second runner-up, Jeet Maiti from Department of Life Sciences, Presidency University, Kolkata. In the poster section, first runner-up, Dripa Kumari, research scholar from Institute of Life Sciences, again, Presidency University, Kolkata. We'll take pictures, so please stand by. And the first prize. Congratulations to the second runner-up and the first runner-up. And the first prize goes to Sudipta Sarkar, research scholar from ISCS. Please come up on stage. A big round of applause for all the 3 winners and all the participants. A big round of applause for the first prize winner. In short, you are requested to project your posters. So first, yes, one at a time. Sudipta will come first. Yes, you can take the hand mic.
Hello, my name is Sudipta Sarkar and I am from the group of Professor Sujay Sinha. We are mainly working on the field of morpholino. We synthesize morpholino and we check whether there is any in vitro, in vivo effect. So we are new in the field of morpholino and there are other downstream processes. So particularly my work is in the field of immunotherapy. So we are trying to think about the other effects rather than the regular siRNA mediated therapy. So here I present some of my research work. So in vivo model, we recently find some interesting results. In morpholino treated mice, they shows effective in cancer therapeutic research. So here I conclude that because thank you very much.
Okay, congratulations again on your work. Keep up the best. The next one, Ripa Kumari. This projects the highlights of your work.
Hello, I'm Ripa Kumari. I work under Dr. Piali Mukherjee at IHS, just the beside neighboring institute. So my work is with Chandipura virus. It is an negative sense single-stranded RNA virus, and basically our work involves how mitochondrial innate immune regulation is involved during an RNA virus, during a neurotropic RNA virus infection, and how virus survive the host innate immune defense mechanism. So we are trying to figure out the mitochondrial innate immune regulation during Chandipura virus infection. Thank you.
Thank you and congrats again. Jeet, if you could reflect upon your findings.
Hello, I am Jeet from Presidency University Department of Life Science, and I am work under Professor Prabir Mukhopadhyay and Dr. Ranjana Pal. And basically, I am working on arsenic-induced leukocyte transendothelial migration, and I perform mRNA sequencing from leukocyte to understand arsenic-induced epigenetic modulation and after effect of inflammation. Thank you.
Great job. Congratulations again. Now we move on to the Best Model Awards. The second runner-up, Working Model of Neuron. The team members are Shreyoti Ghosh, Pragati Jaiswal, Sharnali Datta. Where are they? Come up. Ananya Chaudhuri. A big round of applause. The first runner-up team, okay, there is a joint first runner-up. One is from the structural model of GPCR, and the other is signaling in innate and adaptive immunity. Structural model of GPCR, the team members are Borsho Roy, Momita Shaha, Naushin Das. Please come up on the stage. And signaling in innate and adaptive immunity, the team members are Suraj Shah, Priyam Basu, and Sutirtha Maiti. Big round of applause. And the first prize goes to the model on common mycorrhizal network and defense responses in neighboring plant, Aditi Bagh, Deboshmita Dhar, Shubhra Jyoti Paul. সবাইকে দেখে ছবি হবে তো all the winners. So all the winners, please come up on stage for a group photo. Even the poster winners. The poster winners, please come up on stage. Congratulations to all the winners. All the participants may collect their certificates from the registration desk. The SNU participants will get the certificates later on. And before that, before you leave, there is a little left for the session. I call upon stage Dr. Amit Sarkar, the joint convener, Dr. Amit Sarkar and Dr. Sudipto Shaha too. Okay. So the certificates can be collected from the registration desk, but after the session ends, we are asking all the participants to come back from the registration desk. They're all coming back. All the participants, please take your seats. I call upon stage now Dr. Amit Sarkar, the joint convener, to deliver the vote of thanks.
Thank you, Damayanti. And very good evening, everybody. I know you are tired and you are very much eager to go back home after collecting a certificate, but thanking people is very, very important part of any event. First of all, as we come to the end of this today's compact sessions, I have a feeling that I has attended a master class of cellular signaling, but as I was an organizer, I couldn't attend most of the talks. So I was kind of scared. I was feeling what will happen if there is a test. But thankfully, there is no exam. So I'm saved. But jokes apart, I'm honored and privileged to get this opportunity to propose a vote of thanks for SNU-BioTalk 2025. As you can imagine that to organize any event of such a scale, we require dedicated support from different, several individuals and also organizations and institutes. So when I was recollecting, the list is quite long, obviously. But first and foremost, I'd like to express my sincere gratitude to the Honorable Chancellor of Sister Nivedita University and also the Chief Patron of this event, Mr. Shottam Raichodhuri, for his continuous encouragement and support, all possible support. Next, or very next, I would say that I want to express my regards, deepest regards and sincere gratitude to our Honorable Vice Chancellor, Sir, Professor Dr. Tuberculosis Chakrabarti, for his vision, for his leadership, and his, of course, for his dedication for this particular conference. This conference is actually his brainchild. We are that mere executioner of his orders. I can recall that for the last 4 months of time, I think, despite of his extremely busy schedule, we the organizers met him almost every day, discussed everything from planning to execution in minute details. It was kind of a learning experience for all of us. Thank you very much, sir. A special note of thanks to the administrative leaders of this institute, our Registrar Sir, Professor Shuman Chatterjee, our Group CEO Sir, Professor Shanku Bosch, and our Chief Financial Officer, Mr. Shubir Rokhit Sir, for all possible administrative and logical supports. I also want to extend my sincere gratitude to Professor Mala Mitra, Dean of Science, for constant encouragement. And a special note of thanks also goes to Professor Arun Lahiri Majumdar and Professor Ashok Ranjan Thakur for their constant inspiration and encouragement. And then I would like to extend my deepest gratitude to all the esteemed keynote speakers, Professor David Goldsman, Dr. Andrew Karafelis, Dr. Venkatesh Balan, Dr. Shomen Bashak, and Dr. Dippaman Ganguly, who delivered a very inspiring and thought-provoking keynote addresses. Our deep appreciation goes to our esteemed speakers, distinguished chairs, and also our panelists, sharing their expertise and their thoughts about the current research in cellular signaling. I would also like to thank all other participants and attendees. Most of us are our students, more than 100. You contributed significantly for this event to make this a memorable one. We loved the way our AIMSs conducted this program. Dr. Damayanti Das Ghosh and Dr. Shreyoshi Haldar, a special note of thanks for both of you. I'm deeply grateful to Dr. Rajat Pal, Assistant Director, Microbiology and Biotechnology, and also Program Coordinator for SNU-BioTalk 2025, for his insight and expert advice throughout the journey. A big thanks for the heads of the departments of Microbiology and Biotechnology, Dr. Fatima Kalkattawala and Dr. Shamshubra Thakur Chaudhary, for the deep interest and dedicated contribution as a coordinator of the steering committee for this conference organizing committee. I must take this opportunity to express my most sincere gratitude to my fellow convener and dear friends, Dr. Sudipto Shaha, Dr. Anindita Banerjee, and Dr. Momita Das, for the relentless, untiring effort for last more than 4 months of time. This team shows an example, sets an example of dedication, coordination, and collaborative work at its finest level. I feel privileged to be a part of this particular team. I thank all members of the organizing committee, there's the faculties and staff members of microbiology, biotechnology, and applied nutrition and dietetics. Everybody tried their best in all possible ways. But I have no words to thank Dr. Surjit Singh and Dr. Rachishnu Datta for their effort and meticulous planning, which was instrumental for the resounding success of this event. The same goes also for two of my junior colleagues, Ms. Smita Shah and Ms. Yoki Bhattacharya. I also like to make special mention Ms. Neha Chanda and also Prashanjit Shud and Ranjit Bhomik's contribution. We're deeply grateful to Anushandhan National Research Foundation, or ANRF, for providing support in terms of finance. I'd like to thank Dr. Dibyendu Panda, who was a liaison, for looping in Andrew Carafellis and David Goldsman to this particular event. iSTEM National Coordinator, Dr. Harilal Bhaskar, I thank him for giving a very engaging talk yesterday. Thanks to the participants of the cultural program and their mentor, Dr. Shamshobra Thakur Chaudhuri. Your creativity and dedication added an extra dimension to this particular conference. A heartfelt appreciation goes to the faculty and staff members of the Mass Comm and Journalism Department, led by Professor Minal Parikh, for their contribution, creative contribution, and effective communication, photography, videography, live streaming, everything. I'd like to mention especially the names of Udit, Dibyendu, Shuman, Mayuri, Shemanti, and here our light and sound supports, Narayan and Nayan, and also Dipankar. We are grateful to Ina Mem, to Professor Pradeep Dattagupta, Colonel Dhrubajyoti Chanda, Ms. Papia Vasu, Ms. Shukanna Majumdar, Mr. Abhay Sharma, Mr. Shorab Rajak, Mr. Shubhamay Bonik, Mr. Tulsi Laik, and several other support staffs who worked relentlessly in the backstage for successful of this particular event. Lastly, we also thank our sponsors, Imperial Life Sciences and Spinco Biotech, and our vendors for invaluable contributions and collaboration, which greatly supported the success of this event. So as we conclude, let us take the things we learned here in terms of the cutting-edge research, modern day research about the cellular signaling, and try to contribute in our respective fields based on that. Thank you. Thank you all. Sir.
With this, I just want to declare this SNU-BioTalk 2025 a close. But definitely, whoever present over here, let us give a big hand to the team SNU-BioTalk. And that's the spirit which we have to keep in our mind to take us for better and better and better BioTalk seminars in the coming years. Thank you, everybody. And thank you, our Amit Sharkar, because Professor Amit Sharkar actually thanked all of us, but we should thank Amit. So let's give a very, very big hand to Amit Sharkar.